Dear participants, thank you for joining us today. My name is Niels Fertig. I'm CEO and founder of Nanine Technologies, and it is my honor and great pleasure to welcome all of you to our Cardiac Safety Symposium 2021. For all of you participating today, be it from a research background or from a drug development perspective, we all have one thing in common. We share an interest, we share a partly professional interest in cardiac safety. It's simply close to our heart, so to say. Cardiac safety is the key to the overall safety profile of any new chemical entity. And cardiac liability is the number one reason why a drug candidate fails in clinical trials or even is withdrawn from the market. Hence, there's such a focus on cardiac safety in drug development and regulation to protect people's health and physiological integrity, and also to avoid costly drug development with late-stage failure due to safety liabilities. As you're all aware, there's an ongoing effort to refine regulations within the so-called SIPA initiative, where SIPA stands for Comprehensive In Vitro Proarrhythmia Assay. The initiative intends to provide guidelines for avoiding proarrhythmia risks for any new drug candidates, and actually has been initiated with steering teams from ESI and the FDA and several other international governmental institutions, as well as industry leaders. Nanyon is strongly supporting SIPA, and we actually have been involved more or less from the inception of the initiative. For example, my colleague Sonja Stölzle-Feix is a committee member of the Ion Channel HTS working group and also a participant in the cardiomyocyte working group. You can see her here on the picture, which was taken in 2018 at the Safety and Pharmacology Society meeting in Washington. Well, that was at a time when face-to-face meetings were still commonplace, and we all hope that will soon be the case again. And the photo was actually taken from a dedicated myocyte working group get-together. And I guess some of the faces will be familiar to you. Many of the people in that group are actually participating or even presenting today at the Cardiac Safety Symposium here. Like on the right, I see Ralf Kettenhofen, or in the back, Kanda-san from Japanese NIH. And well, SIPA mainly addresses these four components displayed here, which I'm sure you're all very familiar with. And Nanyon, as a provider of advanced cell analysis instrumentation, participated with two automated patch clamp platforms in the phase 1 and phase 2 study of the Ion Channel HTS group. And also we participated with our impedance technology in the cardiomyocyte working group, phase 2 study. Nanyon has been in the field of automated electrophysiology since the company was founded almost 20 years ago. Yes, that's right. Nanyon will turn 20 years old pretty soon, next year. And we clearly have been involved with the cardiac safety community very intensely over the years. We actually do strive to advance tools and methods to analyze and assess drug safety, which is among the main reasons why we decided to organize an invite for the Cardiac Safety Symposium. And well, the overwhelming response to the symposium really manifests the interest and the relevance. And we are extremely grateful for all speakers joining and contributing to this important topic. Thank you so much for joining today. I'm really delighted by the exciting program, and I'm very much looking forward to the talks and discussions during the symposium. I wish all of you a great meeting. Please enjoy. And with that, I would like to pass the word over to my colleague, Sonja.
Thank you very much, Niels. And hello, everybody out there. I'm Sonja, and a very big welcome also from my side here. So I just want to add a couple of points to the introduction here. Niels has just mentioned the relevance of advanced cell analysis platforms being used in the SIPA initiative. And later on, we will hear about cell-related aspects quite a bit. So the point that I here want to make is that the connection of technology providers or also stem cell providers to their customers is a really strong basis for a reach into the cardiac safety community. So the knowledge, for example, that we gain from being an active member in the Safety Pharmacology Society or within the SIPA is directly transferred to the work being done at the bench. So we provide the adapted experimental protocols, for example. The newest technical aspects are being made available as well, and also the correct data formatting for any cross-industry workflows are realized. So when it comes to the challenges of the people that are being involved in the different HESI and SIPA working groups, you can see here we have the following challenges. So completing projects with increasing demands on our time and resources. Also sharing our work to ensure the larger community is aware and can benefit. And also embracing the next generation of scientists, technology, and more. So this is a slide that actually Brian Barrage has shared in the last HESI meeting in February. And really the last 2 points were a very strong motivation for me and also my colleagues to engage in organizing this symposium here. So I would say, let's get started. And I just wanted to motivate everyone here also to join our roundtable discussions later on. We will have 2 parallel discussions going on the first day and 2 parallel discussions tomorrow. You have very interesting topics here that you could join. So for example, you could discuss about to use or not to use serum-containing media today with our stem cell experts, or you could discuss about automated patch clamp best practices tomorrow with Jim Kramer. So during these roundtable discussions, you can choose if you want to unmute or share your camera, and really you don't have to, but I would say please don't be shy. We are amongst us, and this is your chance to ask any question you have on these topics. All right, so with that, I am extremely happy and very proud to present Dr. Yasunari Kanda today as our keynote speaker. So Kanda-san currently holds the position as Division Head of Pharmacology at National Institute of Health and Sciences in Japan, which is the NIHS. His research area is regulatory science, which seeks to apply new science into the review process and bridge the gap between scientific innovation and patient safety. He coordinates NIHS research activities within JICSA, which is the Japan IPS cardiac safety assessment. And he has also collaborated with FDA and SIPA. In addition, he has contributed to several OECD guidances as an expert. He's also a program committee member of Safety Pharmacology Society and also an international scientific committee member of World Congress on Alternatives and Animal Use. He also serves as the editorial board member of Scientific Reports, and he himself has published over 100 peer-reviewed articles and book chapters related to assessing the efficacy and safety of drugs. So welcome, Kanda-san. We are all excited to have you here today, and the stage is yours now.
Okay, thank you very much. Thank you very much for your kind introduction, and it is a great honor for me to have a presentation at the great cardiac safety symposium this year. I'm very excited to talk with you today. And also, I cannot see my camera onto my computer, so I'm not sure. I can't see you from the computer. So anyway, let's get started. Today, I'd like to talk about current status and future perspectives in cardiac safety assessment using human iPSC technologies. Okay. So today's content is, at first, I'd like to share our summary of the JIXA study for arrhythmia risk assessment. Then we are now working on cardio oncology field using iPS cardiomyocytes. And also, based on this current COVID-19 pandemic, I'm also working on SARS-CoV-2 project using iPSC models. And finally, I'd like to talk about some personal future perspectives in this field. So as Sonja-san told you before, I belong to the regulatory agency in Japan, and I work with PMD in Japan. And basically, we are focusing on regulatory sciences, which means new science or new assay methodologies should improve the predictability in human. Of course, also ensure the product safeties. So we are always trying to see the innovation to understand how to obtain the better predictable models or a predictable way to assess drugs and chemicals and so on. So how can we bridge the gap between the innovation and drug safety issues? And one of the key issues that I'm working on is iPSC technologies. As you know, iPS technologies can provide mechanism-based approach to ensure patient safety as well as product safeties. And also, hopefully, it should provide us a better predictability in humans. And also, in terms of animal alternatives, it should facilitate 3 R's principle: replacement, reduction, and refinement. And at 2008, at the time iPS was developed, I am working on this project at NIHS in Japan. And at that time, we expect that human iPS cardiomyocyte, which express multiple ion channels, should be useful to assess arrhythmia risk assessment. And then, over 10 years have passed to work on this project for me. And 2014, this shows a medical innovation strategy in Japan. And in the section of regenerative medicines, our project regarding the proposed method for cardiac safety issues using iPSC cardiomyocyte can be incorporated into the government offshore strategy in Japan. Then we have started to work with SIPA, FDA, HSC to make a better model using iPS cardiomyocytes for prediction of cardiac safety issues, including TDPs. And based on the OECD Guidance 34, we developed some schedule of the JICSA study to make the testing methods. The OECD Guidance 34 is basically regarding the chemical safety issues, but we apply this concept to drug safety issues. So we have started to make protocols, small validation studies, large-scale validation studies. Then we have worked with CEPR groups to for international validation studies. Then we have now discussed with possible guidance Q&As now. So now I'm working on, I have to talk about some comparison between the JICSA and CEPA. So using the CEPA grid model, regression models, we try to apply the serum-free concentration for the regression models. And we found that serum-free might be better to see the AUC. So previous model is about 0.87, and when you use a serum-free concentration, you can obtain the better AUC shown here. So that is a collaboration with FDA SHIPA to understand the better models to assess TDP liabilities. Also, now we are working on some cardiac ion current using human iPS cardiomyocytes. So we have now obtained the iPS cardiomyocyte as well as overexpressing cell lines such as HEC or CHO and so on. So it would be interesting to see the differences between the iPS cardiomyocyte and overexpressing cell lines in terms of ion cardiac channels. So we like to characterize the cardiac ion channel using human iPS cardiomyocytes. This is because now we have obtained a better, more mature human iPS cardiomyocytes. So it will be interesting to see the differences with without maturation to understand the better predictability in humans. So we have collaborated with Nanion Japan to measure late sodium current using human iPS cells, including homemade academia cells, as well as iCell cardiomyocytes. And also, we are trying to see the ion channel current using iPS cardiomyocyte with some modifications. So based on this discussion, we are now working on some ICH guidance document to harmonize approach and to reduce data variabilities. And also, we like to obtain a better model for translation to clinical settings. Also, in the case of iPS cardiomyocytes, we like to see some drug effects on repolarization studies using some new iPS cardiomyocytes or human primary cardiomyocytes. And this is also a work with an unbiased company to compare the differences between the human iPS cardiomyocytes and primary human cardiomyocytes. So based on this pluralism risk assessment, we are moving on the cardio-oncology, which is a multidisciplinary approach for cancer longer survivalorships. And in order to proceed with the cardio-oncology field, we have had a first cardio-oncology workshop in Japan on February 11, 2020. This is just before the COVID-19 pandemic in Japan. We brought experts, clinicians, basic scientists, regulatory scientists for the discussion about cardio-oncology field. This is basically about clinical information for non-clinical assessment. And we have discussed clinical information and biomarkers, including BNP, and also we are trying to bridge the gap between the non-clinical clinical settings. And also another thing is Japanese Oncocardiology Society has just launched 3 years ago. So this is the only clinical aspect for cardio-oncologists. So we have worked together to obtain the better models, better protocols, or better biomarkers for patient safeties. Okay, so as you know, cardiotoxicity can be observed by anti-cancer agent such as QT prolongation, arrhythmia, and also heart failure, cardio damage, and so on. So we have various types of cardiotoxicities in humans. So we have to understand what we should do for non-clinical settings. Also, this shows the incidence of left ventricular dysfunction that is associated with anti-cancer drugs. So as you can see here, various types of anti-cancer agents, such as anthracyclines and alkylating agent, or a small molecule TKI, show some cardiotoxicities in humans. So we have to translate this clinical information to non-clinical protocols. And based on this situation, we have discussed the in vitro evaluation system of left ventricular dysfunction using iPS cardiomyocytes. And basic question is, how can you assess functional and structural toxicities caused by anti-cancer agent in vitro? To answer this question, we have developed some imaging-based techniques to monitor cardiac contractility as well as relaxations. Based on this spectral tracking system, we have developed motion image-based technique to see the cardiac contractility as well as cardiac relaxation phase quantitatively using this motion video imaging. Also, we are trying to make the better, more mature models using iPS cardiomyocytes. The basic idea is that we need to use the fit-for-purpose cells for each assay systems. And for example, when you culture the cells using the normal plate, the cells can move randomly, and the shape of the type is very small and round. But when you culture the cells using the line pattern dishes, there is a dig onto the culture plate like this. As you can see here, the cells can move along one direction. This is totally different from the conventional plate. So also, we are trying to analyze gene expression patterns using the normal plate or line pattern plate. So as you can see here, some key genes such as cardiac myosin heavy chains and KCNJ2 and SACA, all of these gene expression levels are upregulated using the line pattern plate, suggesting that the cells can acquire some phenotype with line pattern plate. In addition, we see the effect of isoproteinol using this line pattern, using this mature iPS cardiomyocyte onto the line pattern plate. As you can see, the left panel, isoproteinol did not enhance the contraction significantly. In contrast, when you culture cells with line pattern plate, as you can see here, you can see the dose-dependent increase of contraction in iPS cardiomyocytes. So, like this, my suggestion is that you would use the models for your own purposes, such as polarismia or conductivity or some other things such as mitochondria and so on. So we need to assess, we need to characterize the basic iPS cardiomyocytes, whether or not you can see the toxicity or not toxicity. Okay, so and this shows a case with normal plate. So we like to see the effect of anti-cancer agent such as toxic doxorubicin and less toxic erlotinib using these imaging techniques. As you can see here, treatment of doxorubicin inhibits the contraction and relaxation in a dose-dependent manners. You can see the effect at 0.3 micromolar or 1 micromolar, which is equivalent with human blood concentration in patients. In contrast, in the case of erlotinib, there's no big differences with or without drug treatment for contraction and relaxation. So what I can tell you here is that motion imaging technique can distinguish the toxic compound and less toxic compound in terms of conductivities. Also, we examine the effect of this compound on cell viabilities in iPS cardiomyocytes. This is also iPS cardiomyocyte with normal plate. As you can see here, treatment of doxorubicin at 0.3 micromolar for 8 days, chronic treatment, the cell undergo apoptosis like this. And also, this data was confirmed by 7AD stainings. And based on this data, as you can see here, cell viability was decreased in a dose-dependent manner at 8-day exposures for doxorubicin. In contrast, in the case of erlotinib at 1 micromolar, there's no significant differences between the cell viabilities during the culture period. So it is necessary to establish an in vitro assay system to predict functional as well as structural toxicities using iPS cardiomyocytes. Also, I like to share another examples drug candidate for hepatitis C viruses, HCV. So upper panel shows a compound BMS 286094, which was developed as a prodrug, as a nucleotide analog for treatment with HCV patients. But it was discontinued at clinical phase 2 due to the cardiotoxicity, especially conductivity impairment measured by LVEF. Also, I like to show the data about sofosbuvir, which is also developed as HCV treatment, and it was already approved. Using these two types of compound, we like to compare the cardiotoxicity assessment using imaging-based techniques. As you can see here, left panel shows a waveform, motion waveform treated with BMS 98094 at 0.3 micromolars. As you can see here, conductivity was reduced after treatment of this compound in a dose-dependent manner. In contrast, in the case of sofosbuvir, the decrease of conductivity is smaller compared with this BMS 98094 compound. And this is the quantitative data using image-based analysis. As you can see here, contraction velocity was inhibited by this compound in a dose-dependent manner like this, and relaxation is a similar data. In contrast, in the case of sofosbuvir, contraction velocity as well as relaxation velocity are very similar with or without treatment. So I think that this contractility parameters such as contraction velocity or relaxation velocity might be useful to distinguish the toxic compound with less toxic compound in terms of contractilities. Then we have checked the effect of this compound on calcium transient. As you can see here, this compound did not affect calcium transient at 48 hours. But after a long-term culture, this compound inhibited contractility at day 4 or at day 6. But in this case, in the lower panels, sofosbuvir did not affect contractility parameters or calcium transit data up to 6 days. Then we have decided to check the effect of gene expression patterns with or without this compound. And as you can see here, gene expression of myosin heavy chain or troponin was reduced in the for the treatment of BMS 986094. However, like this. However, sofosbuvir did not affect the gene expression patterns of myosin heavy chain and troponin 1. So this means that contractility impairment as well as cell toxicity should be important to understand the comprehensive cardiac safety issues for assessment. I'm not sure, to be honest, I'm not sure the relationship between the cytotoxic effect and contractility impairment for this case, but for the based on the time course study, cytotoxicity should happen prior to contractility impairment. So based on this technique, we are trying to attend the healthy mouse site validation studies. That is an ongoing project. So this mouse site validation study has been developed to identify chronic treatment-induced cardiotoxicity. And we are now working on some to get some confidence in individual assay systems, including contractility or mitochondrial toxicity, biomarkers, and so on. So I don't want to tell you more about this HT validation study, but one thing that I can tell you here is that mouse site study is very expected to reveal how to use iPS cardiomyocytes for drug development, drug safety issues, including cardio oncologies. Okay, so let's move on to the next topic, cardiotoxicity with COVID-19. So, as you know very well, COVID-19 SARS-CoV-2 virus binds to its receptor AC2 and then go into the cells and replicate RNAs in patients. And one thing from clinical studies suggests that patients have some severe arrhythmia or myopathy or myocarditis and sometimes cardiac arrest after infection of COVID-19. So as a regulatory agencies, we are trying to see some suitable model for patient safety or disease modeling. And we have used iPS cardiomyocytes to see whether this SARS-CoV-2 can be infected into the cells. As you can see here, after infection of SARS-CoV-2 for 48 hours, as you can see here, N protein of the SARS-CoV-2 and spike protein of the SARS-CoV-2 can be seen using the iPS cardiomyocyte. Suggesting that iPS cardiomyocytes might be a model for inspection of SARS-CoV-2. Also, we have checked the effects of infection using the MEA system. This is milestone data, but after infection of SARS-CoV-2, as you can see here, there are some EAD phenotype of the field potential recordings, suggesting that iPSC cardiomyocytes can be a model to assess arrhythmia caused by infections. Also, we see the effect of contractility measured by muscle motion software. And as you can see here, contractility and relaxation can be inhibited by SARS-CoV-2 infection. Also, using the spike protein, we have examined the effects of oxygen consumption rate measured by C-force apparatus. And after addition of spike protein, as you can see here, oxygen consumption can be reduced by spike protein like this. And when you use a SARS-CoV-2 inhibitor treatment drugs, the inhibitory effect of oxygen consumption can be recovered. So I guess these are good model for disease mechanism study and so on. And also we have worked on some drug repositioning data using this assay system. And there are several types of drug repositioning, such as remdesivir and dexamethasone, are approvable drugs for COVID-19. And we are focusing on some AC2 internalization after adding to the spike protein. So we try to search a drug to inhibit this internalization, because theoretically, spike protein with mutation also binds to AC2 and undergo internalization. So we are trying to see the suitable drugs to prevent internalization. And yes, so this is a screening system for drug repositioning, using the AC2 overexpression cells. We have used screening of the studies. And as you can see here, before adding the spike protein, the AC2 is localized at the cell membrane. And after addition of the spike protein, this AC2 was internalized like this. In contrast, if you use anti-COVID-19 drugs, the internalization was inhibited, shown in GFP signals. So also, we have checked confirm the effects using the Brett signal. I'll skip this data. And then we found that clomipramine antidepressant inhibits the RNA replication of the virus using the Veros E6 cells. In contrast, other cells did not affect the RNA copy numbers in the Veros E6 cells. Also, this clomipramine inhibits the SARS-CoV-2 RNA copies in a dose-dependent manner with IC50 4 micromolars. Yeah, of course, this drug is basically work on the brains, but it has an ability to inhibit the AC2 internalization in Veros E6 cells. And also, we confirm these data using iPSC cardiomyocytes. In addition, oxygen consumption rate can be recovered using this clomipramine, showing in green. So also we have obtained the data with a synergistic effect of clomipramine with remdesivir. So based on these investigations, we found that iPSC cardiomyocytes can be a model for drug repurposing for COVID-19. So, okay, so I'll speed up my presentation. So also, we have worked on COVID-19 drugs for cardiac safety assessment, especially we have obtained some data for hydroxychloroquines. And treatment of hydroxychloroquine induced EAD in iPS cardiomyocyte at 3 micromolar or more. So IC50 against HERG is 2.5 micromolar, and this is basically based on the HERG inhibitions. In contrast, hydroxychloroquine, remdesivir or favipiravir, camostat, and ivermectin did not affect fetal potential duration in iPS cardiomyocytes. In addition to fetal potentials, hydroxychloroquine inhibit the conduction velocity using iPS cardiomyocytes, and also it decreased the contraction phenotype using iPS cardiomyocytes. And based on these parameters, we have just checked the parameters electromechanical window in iPS cardiomyocyte. We have just defined this electromechanical window, which is the time differences between at the end of the fetal potential durations and at the end of the relaxation phase. So we just define the time between the fetal potential and contraction. Some people use a calcium transient CDD90 instead of contraction relaxation durations. So here we have just used data from contraction relaxation durations. And as you can see here, application of E4031 decreased the electromechanical window in iPS cardiomyocytes. In contrast, in the case of moxifloxacin, electromechanical window did not decrease after treatment with the drugs. Also, as expected, hydroxychloroquine inhibits the electromechanical window in the cells. So, suggesting that this electromechanical window might be useful to assess arrhythmia risk assessment. So, finally, I'd like to talk about some future perspectives in my talk. And one of the things is that cell types. So basically today I have talked about the 2D monolayer models to see the cardiac safety issues. Also, we are working on some 3D models, spheroid models, or microphysiological systems for future cardiac safety issues. Hopefully, the maturity of the cells should be improved by 3D or organoid. However, you would obtain the lower throughput using the more complex, more advanced models. So we need to think about which cell type should be useful or should be suitable for the purposes such as arrhythmia or contractility or mitochondrial dysfunction and so on. And this is one of the medical innovation projects in Japan now that is devised several years ago. So hopefully we like to find a better models for cardiac safety issues. But one thing that I can tell you here is that we do not standardize our MPC model for safety issues, not only for cardiac safety, but also other toxicities such as hepatotoxicity, nephrotoxicity, or other type of toxicities. So it is important to discuss how to standardize models of MPS, but we already have various types of MPS available all over the world. So it is not easy to discuss how to proceed with standardizations. So in the case of cell types, we have to solve the problem regarding the diversity of iPS cardiomyocytes. So also we have made some homemade iPS cardiomyocytes by myself, but it is not easy to compare our own cardiomyocyte and other types of cardiomyocyte developed by other sources, other protocols, and so on. So we need to discuss the diversity or 3D or co-culture models, and some conditions might be reflected, such as diabetes or inflammation, and so on. Of course, you can obtain the healthy donor or disease models using these iPSC technologies. We have to think about the purposes such as anti-cancer agent or acute or chronic study or drug combination such as drug-to-drug interaction or drug combination such as hydroxychloroquine plus azithromycin and so on. And based on this technique and assay system, you will obtain some confidence for individual assay systems. So today, I don't discuss about the simulation data, but it is important to combine the wet data, including iPS cardiomyocytes, with dry data, such as simulation or physical chemical properties, and so on. So we need to discuss more about the future safety pharmacology using these iPS cardiomyocyte techniques. And these techniques should be categorized as new approach methodologies in safety field, as well as other type of assay system. And this idea is also applied for chemical safety assessment, such as developmental toxicities and so on. Okay, so let me summarize my talk. So today, I have to talk about the cardio oncology field for future iPSC field. Also, hopefully, MPS or simulation would be required to ensure patient safety as well as product safeties. Okay, thank you, Ben, for your discussion. And this is my collaborators in Japan and also in outside Japan, such as FDA, HESI, and so on, and SHIPA members. Thank you very much for your attention. And I apologize for the delay.
Thank you so much, Kanda-san. It was a really great presentation, and I think you handled the technical difficulties very well now by reducing some of the slides. So we are almost in time, and I think we have time for one question. So there was one question coming in from the Q&A here, which is, I think, very interesting. So the question is, Kanda-san, your observation that SARS-CoV-2 spike protein can reduce oxygen consumption rate in iPSC cardiomyocytes is very interesting. Why do you think this is happening?
Oh, you mean the mechanism of the lower oxygen consumption? So
yes, exactly.
Actually, I don't have a specific answer for that, but one thing that I can observe is that mitochondria can be damaged by SARS-CoV-2 infections. When we see the iPS cardiomyocyte with SARS-CoV-2, the shape of the mitochondria is going to go to the punctured dotted structure. Mitochondria is basically a fusion type, but in the case of SARS-CoV-2 infection, the shape has been changed. Morphology has totally changed, and you can see the dotted structure. So I guess that mitochondrial function should be decreased after infection.
Okay.
Yeah.
All right. So I think then we should move on to the next presenter, just to stay in time. So Kanda-san, thank you very much again for your very interesting presentation. Lots of insights here.
Yeah. So please don't hesitate to email me about my presentation, if possible.
Very good.
Thank you very much.
And we will also not miss answering all remaining questions or sending them to you and to the other speakers as well. All right, so after these outstanding insights now, we continue with a presentation from Dr. Ralf Kettenhofen. So since 20 years, Ralf has been involved in development, standardization, and production of pluripotent stem cell-derived cardiomyocytes and other cell types. And Ralf obtained his PhD and continued with postdoc research at University of Bonn. Since 2001, Ralf has been a remarkable expert in iPSC-derived cells, firstly as head of lab at Axiogenesis, then as a senior scientist at Encardia, and now as a senior scientist and business development specialist at the Fraunhofer Institute for Biomedical Engineering in Würzburg. So Ralf's expertise contributed enormously to the community, and I'm very happy and excited to hear your insights today, Ralf. So Ralf will talk about processes from iPSC banking to drug development and cardiac safety. So welcome, Ralf. The stage is yours now.
Yeah, thank you very much, Sonja. It's a pleasure to be here. For all these years we have been working together, it's a pleasure to participate in the Cardiac Safety Symposium. I hope you all can see my screen.
Yes, all fine.
Perfect. All right, and I will continue. Oops, work. Slides not moving still. Just going on some difficulties here. So slides are not moving. Where do I need to click? Ah, now it's good. Okay. Thank you very much for the invitation. I want to talk today about our processes at the IBMT and introduce the institute I'm working for, and as well as talk about the second part. And I will introduce the EBSC, European Bank for Pluripotent Stem Cell, as one of the biggest projects, as well as the second one, which is the R2UTOX project. And the second part, I was asked to speak about the changes in the integrated risk assessment. And to my point of view, this is really a change in the paradigm of the regulatory guidance, the cardiac safety assessment. But starting with my institute, so I'm working at the Fraunhofer IBMT, it's the Institute for Biomedical Engineering, and we are part of the health group at Fraunhofer, and we are focusing on drugs, diagnostics, devices, and data, and this is the 4D of medical science for us. Just a brief overview for those of you who don't know Fraunhofer in total. So the Fraunhofer Association, or Gesellschaft, is a non-for-profit organization, and our mission is actually to support and utilize private and public enterprises for a wide benefit to society. We are about 29,000 staff in total in Fraunhofer, and we have 75 institutes all over Germany, and also internationally we are very engaged. We have about 2.8 billion euros of financial volume, but this is a little bit different to Max Planck. Actually, we are only funded by 30%, only by 30% by government. The rest we have to cope actually by interaction or projects with industry or direct interactions with industry. Yeah, so today I will introduce a little bit of the work I'm doing. So we have several locations actually. With one of them is definitely the project center in Würzburg, where I'm working at. Our headquarters is still at Sulzbach. It's in the Saarland in Germany, in the southwestern part. And we have a subsidiary also in Berlin, where we are working mainly with the with data sciences, which became a really prominent, important part of stem cell work. I won't go into that today, but I'm happy to explain that offline with you. Okay, so what are we doing in Würzburg and in Sulzbach mainly? So this is stem cell process engineering for drug development and regenerative medicine. So starting with a donor supplying somatic cells, all of you know that we can reprogram those cells into induced pluripotent stem cells. And these can be genetically manipulated, either to induce a special phenotype of the cells or to include reporter cell lines, whatever you can think about. And yeah, we are also integrating the data science behind it, which is, as mentioned before, a really important part of it. And recently we have integrated the human pluripotent stem cell registry into IBMT, which was formerly at the Charité in Berlin. And all this is integrated into a large project, the eBISC project that I've mentioned before. And I will come to this later on what we are doing there. So in Würzburg, we are focusing on induced pluripotent stem cells, the automation and production upscale of these cells. And we're also moving forward to differentiate those cells into the variety of cell types, including cardiomyocytes. So this is actually the part where I'm most engaged in as a scientist, and I'm still interested in these cells. So all these cells are then used for tissue engineering, and we are also introducing 3D printing and material sciences to bring new materials into this stuff to get matured cells. And we use those also for assay development. And these can then be introduced into drug development and drug safety, as we're talking today, as well as cell therapy and regenerative medicine, to hopefully bring some benefits to the donor or the patient actually back. And around all this stem cell derived science, we have a lot of know-how in the area of cryotechnology and biobanking. So as ISBIX already is mentioned, and GMP is one of the features that we have as well. And coming to eBISC, the European Bank for Induced Pluripotent Stem Cell, this is supposed to be a sustainable source for relevant human cell models in drug testing. And Fraunhofer is here the first non-industry partner coordinating EU-funded IMI project. So I'm really grateful that IMI is supporting this project here. And we have 10 pharma industry partners from FBPhS, the European Federation of Pharmaceutical Industries and Associations. And together with our partners, we are setting the industry standards for iPS expansion, biobanking, and quality control. And all this, hopefully, we managed in the second part of the funding of eBISC to make this really a sustainable cell bank, which is supposed to distribute not only iPS cells, but also derived cells from donors that are healthy or diseased. So we can see in red that marked here, actually, what kind of services we are also providing within eBISC. So this is something we can collaborate on with you as well. I want to into detail. The second project that I want to introduce is our ready-to-use tox assay. So we're utilizing iPS cell technologies together with new breakthrough cell freezing technologies to bring ready-to-use plates into industry. How do we do this? So we use, for instance, patient-specific cell lines, iPS cells, differentiate them into the cells of interest, like cardiomyocytes or neurons. And we scale up the production of these cells. And then we use a process called vitrification, which brings the medium immediately into a glass phase state that avoids the crystallization and therefore is very smooth to the cells. And it's thought that we put the cells onto the assay plates already and vitrificate them on the plates so they can be stored for a long time and can be then sent to our customers and collaborators as ready-to-use assay plates to be used when needed. And these can be produced in large scale, can be introduced into phenotypic screening, as well as safety pharmacology. And safety pharmacology is already the part to come to my next topic of my talk, which is actually the new integrated risk assessment in the drug development part. And as I said before, I'm convinced that this is already a great step forward. So it's a real change, the paradigm of the regulatory guidelines for cardiac safety assessment. Okay, the drug development process, just to have an overview, I think everybody of you is quite familiar with that. It splits into two parts, which is actually the preclinical phase as well as the clinical phase. And you have to screen about more than 100,000 compounds to finally bring one drug into the clinical state, and hopefully can bring it also to approval by the FDA or regulatories. The risk assessment itself here is an integrated one, and the complexity of the assays, costs and time increase with the progression of the different stages of the drug development phase. And there have always been made progress and new innovations to de-risk compounds with new assay types. And for this, actually, there will be a lot of improvements around this one. But cardiac safety assessment is done mainly in the area of preclinical development and clinical phase 1 and 2. And these are regulated by the guidelines ICH S7B as well as ICH E14 for the clinics. So the current cardiac safety assessment in the preclinical area is mainly regulated by ICH S7B, which is GLP-HERG assay, ion channel assays, patch clamp assays, and GLP in vivo animal ECG studies. And these studies have been only used to make no-go decisions for the clinical phase. In the clinics, and I'll just make a slight step here to the clinics as well, we have the clinical phase 1, which is mainly used to assess drug safety with healthy volunteers. And this is QTC assessment with dose dependent. And the clinical phase 2 is the dose finding part of the clinical phase. And here we have larger studies to do, which is thorough QT studies, including high quality ECGs with 3 times therapeutic dose to make sure that there is no side effect. And it has to include a placebo and positive control group as well. And up to now, there is no communication between the preclinical development results and the clinical phase, only between the clinical phases themselves. So it's a clear separation between those. So there have been now new amendments and substitutions to the clinical phase already, because there are several situations where you can't really conduct thorough QT studies. So there's one amendment in the question and answers process. This is one of the processes that is used by the regulatories to introduce changes in the guidelines. And the first one is Q&A 5.1 that was finalized in 2015. And the aim of this was to waive positive control groups. Sometimes it makes sense to do that, right? And this is, for that, actually, the requirement is that you have to introduce high-quality ECG recording already in clinical phase 1. And there has to be a concentration to dependent QTC determination. And it has to be a supertherapeutical dose treatment with 6 times therapeutical dose. Then you're able to waive positive control groups. The Q&A 6.1 is the second amendment that was finalized in 2014. Is actually the aim is here to substitute thorough QTT studies when they are not feasible, like if you have to check a combination of drug products, biologics like large proteins or oncological products. And here, actually, of course, you can imagine that it's difficult to get to manage a high exposure scenario of 3 times therapeutic dose, and also no placebo, for instance, in oncological drug application is not feasible. And here, drug assessment is only done by normal QT recording, and an assessment where the delta of the QTC is not allowed to be larger than 10 milliseconds. So these are the current settings, right? And in the meantime, actually, the HESI Cardiac Safety Committee has conducted the SIPA initiative. We have already heard of this by former speaker Kanda-san. It actually was started in 2013 already, and combined the 4 different groups, the I-channel high throughput group, the in-silico group, which we're communicating a lot, the stem cell myocyte group, as well as the human ECG group. They have produced a lot of data, generated, the protocols were established, new studies were validated, and a lot of publications have come up recently, just to mention a couple of them here. So in 2018, the validation study by first author is Xenia Blinova here was published, as well as the application of impedance assays and field potential recording by Bott et al. Kanda-san has published as well on the human stem cell, induced pluripotent stem cell with the Jigsaw group in collaboration with the HESI. And Warong Liu from Janssen has published data using calcium imaging. And this all ended finally in the publication in 2020 by the best practice manuscript where Gary Jenkinson is first author. And all this has now ended up actually finally in the last process where these findings and validation have are implemented into new risk assessment. So the ICH is currently updating the current guidelines with another question and answers process. And yeah, this is an ongoing process, and hopefully this will be finalized in the first quarter of 2020. As you can see, this is a 9-year process, long process, but I think finally this was worthwhile to go for it. So what is the new integrated risk assessment now? So now it's called a non-clinical development for some reason, as we can see. So we have now currently is integrating the Q&As, the best practice HERC ion channel and in vivo ECG studies. There is an attempt to add safety margins definitions, and this will include also that these preclinical or non-clinical studies need to include a 3-time full Cmax study with metabolites and from the parental drug as well. And if all the data that is obtained from these 2 core studies, the drug is actually called double negative, which means now that this is able to support, to inform the clinical phase. So now we have a direct information that goes into the clinics. This will be able to support lower clinical dose exposure and wave positive controls, as well as to only use lower doses and QTC prolongation risk assessments in the clinical phase. This is really a change of paradigm. So no real thorough QT will be needed then. But what happens if the best practice HERC and in vivo animal studies are not double positive? So, or a pro-arrhythmic signal is detected in the clinics. So the new feature of the risk assessment now contains the so-called follow-up studies. And here are included in silico studies as well, and then finally the human stem cell cardiomyocyte polarization studies. And these can bounce back and inform the clinics again. So we have a forth and back between clinical and non-clinical development. With this, I want to summarize my talk a little bit in regards of the non-clinical and the question answer amendments. So we have the integrated risk assessment, so, still containing the current ICH guidelines, for in vitro HERC assay, in vivo assay, and other information that we need, so relevant non-clinical studies and going into the risk assessment as well as clinical information. The follow-up studies are integrated here, and all this ends up in the evidence of risk, what it's called now. And the amendments that are currently integrated is here in the integrated resistance. So we are currently integrating the general strategy and recommended methods to calculate HERC safety margins. We have best practice considerations for our patch clamp assays in Q&A 2.1. Q&A 3 are containing the best practice considerations for in vivo QT studies. And finally, we have the question answers 2.2 to 1.5, which contain the best practice considerations for in vitro human myocyte repolarization assays. And the final summary of my entire talk as take-home message. So Fraunhofer IBMT is engaged in and offers actually standardization of culture expansion, biobanking, and differentiation of iPS cells, development of in vitro models for drug development and drug safety based on iPS cells and the derived cells. And we offer collaboration in the stem cell sciences. And for the risk assessment and the change here and the paradigm change, can take up message now. We have the in vitro assays informing the clinics. Best practice HERC in vivo QT recordings will be integrated via the Q&As as well as the follow-up studies integrated into the guidelines in silico testing as well as human stem cell cardiomyocyte assays. With this, I want to thank you for your attention, and I'm ready to take some questions.
Brilliant talk, Ralf. Thank you very much. I think we all enjoyed these in-depth explanations of the risk assessment and all the Q&As. And definitely, it's also very interesting to see what is going on at Fraunhofer and EBISC. So I have one. The first question here incoming is, let me read that to you. So will the Q&A amendments from 2.2 to 2.5 allow to submit iPSC-derived cardiomyocytes data to the FDA for the approval of clinical studies?
Yeah, that's a really good question because actually the current status is that it's supposed to be used for follow-up studies. So if the double negative are not occurring in the preclinical core studies or in the clinics, so it's actually not supposed to be directly, but I've heard from with direct communication with scientists from the FDA that stem cell cardiomyocyte assay data is welcome to be submitted as well. So I think this will be not a problem. Since they are validated and they are now in the guidelines, there shouldn't be, to my point of view, it shouldn't be a big issue to submit data from stem cells.
Mm-hmm, mm-hmm. Another question here was, which assays with iPSC-derived cardiomyocytes can be used to assess acute drug-induced effects on repolarization?
Yeah, also a good question. Actually, I think we are going to hear quite a lot of applications of stem cells in the next talk, so I won't go into deep here. But actually, I can refer to the publication by Gary Jinton in 2020, where the best practices are all mentioned. And they propose to have transmembrane potential assays, for instance, patch clamp of stem cell cardiomyocytes, or microelectrode-based transmembrane measurements like the LEAP assay, for instance, that can be used. Field potential recordings, impedance and contraction assays can be used, as well as optical recordings like voltage-sensitive dyes, as well as calcium transition recordings. So Kanda-san has already shown a lot of those kind of data.
Yeah. Okay, so now I see it's time for a networking session. Jason, I think you have some explanations here.
Yeah, thank you, Sonja. Ralf, maybe if you could just end the screen share, then I would just share a couple of slides just for everyone in attendance to see what we have planned next for everyone. So you should be able to see my screen share now. And I draw your attention to a nice little session that Hopin allows for. So on the left-hand side of your screen, you should see a tab called Networking. This will be a de facto interactive mixer where Hopin will match you with a fellow meeting participant by default. So meet someone new or rekindle familiar face. By default, each meeting is set to 30 seconds, but feel free to extend your talk if desired. The platform will automatically connect you with the participant. So we're going to reconvene after 2:45 p.m. Central European time for Bettina Lickiss, who will be presenting, and a little bit of also due diligence on Hopin, some of the features that you're seeing on your screen. So on the left-hand side, you see the stage. So whenever we have an upcoming talk, whether we have a breakout room or whether we have a networking session, you could always navigate back to the stage ahead of a scheduled presenter that we have. You can also take a look at the expo. That basically is a summarized consolidation of all the affiliates and institutes that are presenting here. And with that, I end my quick little screen share and direct your attention to networking and welcome you back at 2:45 for Bettina's presentation.
Hello, everyone. Welcome to session 2. My name is Elena. I'm a Senior Sales and Alliance Manager at Nanion Technologies. And I hope you have enjoyed the little networking session. So it was something like a scientific speed dating. It can be interesting, and one can meet a lot of people that one usually maybe not get in touch voluntarily. So I'll keep this short and sweet. I would like to say hello to everyone. Welcome to session 2. And I will start with our first speaker. So her name is Dr. Bettina Lickiss. She is from Innovitro, and there she's responsible for scientific communications and business development. Bettina holds a doctorate degree in neurobiology, and she also gained 5 years of experience in stem cell biotech industry and in the field of human iPSC-derived cardiomyocytes before she started in Innovitro. In her today's talk, Bettina will focus on advanced cardiac contractility assessments with integrated mechanical and optical stimulations on cardiac iPSC-derived or iPSC-derived cardiac cardiomyocytes. Sorry for this. Bettina, welcome. Maybe you start sharing your screen, and then we start with your talk.
Thank you very much, Elena. Yes, I'm going to share my screen. Hold on. So, can you see my screen?
Mm-hmm. Yes, maybe you go full. Yeah, perfect.
Yeah.
Perfect. We can hear you. We can see you. Enjoy.
You can hear me. You can see my presentation, so I can just start. Perfect. So thank you very much for the invitation and also for the very kind introduction. I'm very happy to be here today to talk about data of mechanical and optical stimulation. Hold on, I'm going to use my laser pointer. Of mechanical and optical stimulation of human iPSC-derived cardiomyocytes using the FlexSite technology. So here you have a little teaser trailer of the FlexSite plates where you can see the cardiomyocytes beating nicely in a dish or in the 96 well. But before I go too much into detail, I would like to tell you why it's actually good to have an alternative cardiac contractility assay for preclinical drug development studies. So you know that we have a big problem with drug attrition rates throughout the entire process, actually, of the drug development. And we know also it's actually a multi-factor problem, but when we think of preclinical stages, we know that one of the reasons why I have high drug attrition rates is because we're still working in with animal models trying to predict what's going to happen in humans. So one very good way to improve the situation is by also introducing cell-based assays that are using human iPSC-derived cell types, for example. And when we are looking at the organ systems that are involved in drug attrition rates, we know that there are basically 3 major ones involved. It's the cardiovascular system, it's the nervous system, and it's the hepatic system. And again, when we have a look at the border between non-clinical and clinical phase, we see that there is that we have the cardiovascular system drug attrition rates due to cardiovascular system slightly reduced in phase 1. So it's giving us a good hint that we're doing something quite right in the preclinical stage. So let's have a look at what we are doing there. So the preclinical endpoints that we're using to address the cardiovascular system preclinically is, on one hand, the action potential, so the electrophysiology part. It is the calcium transient, as well as the contractility. So these are the endpoints that we are using. And now let's have a closer look at the gold standard methods that we are using to assess these endpoints. So for the electrophysiology, we usually use patch clamp techniques and MEA techniques. We have, for the calcium, a diverse set of calcium imaging techniques. And for the contractility, we usually use the Langendorf heart, which is still an ex vivo animal model in comparison to the other ones that are mainly using cell-based assay systems. So having a closer look at the Langendorf heart, it's basically a technique that is 100 years old, and mainly rodent or frog hearts are used for this model. So we have a different species we're working in. We have a rather low predictivity due to that. We have a rather low throughput because, as you can see here in the picture, we use an entire heart for one test. And obviously that drives up the cost in the lab work we have to put in. But it also has a very good advantage, and that's the physiological relevance that we get along with an entire heart we're working with here. So the FlexPsych technique that we are working with is basically, well, I like to call it the modern version of a cardiac contractility assay. So we're using human iPSC-derived cardiomyocytes as a human model. So the human model, we're basically in the same, we're therefore in the same species. So we have a higher predictivity. We're using a 96-well system, so we scaled up the throughput, and therefore, obviously, we have an impact on lowering the costs and also the lab work that goes in. And very important in this case is that we are also physiologically relevant. So why is that? When we look at this video here, which is basically the video that you've seen before, but this time you see the FlexPsych plate from below, you see that instead of stiff plastic and glass surfaces, we have membranes. So we have ultra-thin silicon substrates, membranes that serve as substrates for the cardiomyocytes. You can see that here, that are then plated onto the membranes. And this gives the cells the mechanical environment that they usually experience in the native human heart. So we create quite a physiological condition for them. So the measurement happens by when the cells are contracting, as you can see here, and actually down in the video, you can also see the electrodes. There's a change in deflection taking place. And this change in deflection is then quantified by capacitive distance sensing. That's what we try to depict here in the schematic view. And this is how we get the raw data. And we also have a specialized software for it so that you don't just have raw data, a bunch of raw data that you then have to evaluate somehow. No, we basically do it end to end and also come up with a good software to analyze it. So talking about the parameters, I'm pretty sure most of you are very familiar with the amplitude and the beat rate that you're focusing on when it comes to talking about contractility. But with the FlexPsych technology, we're also able to basically take the beat apart into its little bits and pieces. We have, as I said, the amplitude, but you can also focus on the durations. For example, the upstroke, downstroke duration. You can focus on the entire beat duration. We have the upstroke and downstroke slopes, so the velocities, and also the upstroke and downstroke integrals, so basically the area under curve. We are focusing on the frequencies, so beat rate variations, and of course, arrhythmic events. So since all of the data, basically I'm going to show you later, is going to be analyzed data, I thought it's also nice for you to see how the raw data is actually looking. So in this case, you see a regular beat rate with no added compound that will change the beat rate. But when you then add compounds that have a cardio effect, you see that there can happen like beat rate irregularities, like in the second panel. You can see after contractions popping up, like here highlighted with the red arrows. Or you see proper fibrillation events, like in the lower panel. So since I compared the FlexPsych technology to the Langendorf heart, you know already that we're using it a lot for preclinical cardiac risk assessment of new drug candidates. So we're doing acute, but we're also doing chronic safety and tox measurements. But there are also lots of other cardiac contractility related endpoints that we can address. And I just want to point that out here. For example, we can also focus on cardiac maturation, because we already know that the one drawback that we have with the cell type is that it basically expresses a rather juvenile phenotype. And with the physiological environment we created with the Flexide, we support the maturation in vitro and thereby getting more native-like data, especially regarding positive inotropic compounds. Another topic that we can assess is, for example, different medium compositions. So if you have, in this case, serum versus serum-free medium you want to compare, it's a possibility to use the Flexide with on the functional level. If we have isogenic cell line comparisons, healthy versus disease phenotype characterization, for example, or if we characterize in-house cell lines, or as Ralf mentioned before, like cell bank cell types, that's also possible. So before I actually go to the topic that I mentioned in the title, so the light and mechanically induced pacing, I would actually like to focus on the first three topics here as well, that you have a better insight of acute and chronic safety. You see how the cardiac maturation takes place, and also a little outlook in how medium assessment can be done. So starting with a little combination of acute functional analysis that also shows the pro-maturation effect. In this case, we chose three positive inotropic compounds, known compounds, isoproterenol, SBA, and omecamtiv mecarbil. So all three compounds that are working molecularly on a completely different level. But it's usually not with all cell-based assays, but it's sometimes hard to create positive inotropic compounds reactions with human iPSC-derived cardiomyocytes. So in this case, we were able to show that with the Flexide technology, we kind of push the maturation in the way that we can see upon increased concentrations of isoproterenol, SBA, and omecamtiv mecarbil, there's also an increase in the amplitude. If you look at the shape of the amplitude, you see that there are differences, and therefore it's also good to have the other parameters to assess. So thinking of the contraction relaxation cycle, we can refer to the upstroke and downstroke durations. So for isoproterenol, you see that with increasing concentration of isoproterenol, there's a decrease in the downstroke duration. The opposite effect we see when we added SBA to the cells, where you see that there is definite increase in the downstroke duration, so basically an increase in the relaxation phase that you can see here as well. And for omecamtiv mecarbil, you can see that the contraction relaxation cycle stays very stable unless you go into really unphysiological conditions with 1 micromolar. So this is the pro-maturation effect I wanted to show you on an acute basis. And the next topic I would like to address is the chronic toxicological analysis of human iPSC-derived cardiomyocytes. For this, we have chosen, for showing you detailed analysis, we have chosen 3 different compounds, all 3 basically of the cardio-oncology background. We have erlotinib and vandetanib as tyrosine kinase inhibitors. We have idarubicin as an anthracycline. These 2 are very cardiotoxic compounds, and erlotinib is known as a cardiosafe compound. So we evaluated the data over 5 days as a chronic analysis, and we chose the amplitude here to show you that erlotinib is indeed working cardiosafe with even very strong concentrations of 10 micromolar, only affecting the amplitude mildly. In comparison to vandetanib, where you can see here indeed a very dose-dependent effect, but not a time-dependent effect, because if you, for example, look at the 1 micromolar, you see that there's an immediate decrease of the amplitude down to 50%, but then over time, over 5 days, it stays pretty much the same. And if you look at idarubicin, you see that there's a dose, but also a time-dependent effect. So not really when you look at the 10 micromolar, because then you just knock out the cells completely. But if you go to milder concentrations, to, for example, 10 nanomolar, you see that there's a progression profile, that there's a slow decrease in amplitude over time. So this was a detailed version. We have actually done a chronic cardiotoxic profile of a lot of compounds. So of in total 15 tyrosine kinase inhibitors and 3 anthracyclines. And we made a heat map out of it to make it a bit more accessible. So for this, we chose the parameters amplitude, beat rate, and beat duration with the 4 different compounds, compound concentrations, and also the days, like 1 day, 3 day, and 5 days after a compound administration. And you can see here in the first 3, for the first 3 compounds, all 3 anthracyclines with a cardiotoxic effect, that there are indeed strong seizing effects of the amplitude, the beat rate, and the beat duration highlighted in deep red, especially at the strong concentrations and also at day 5. The opposite is visible when you look at the negative controls, in this case allotinib, where you see stable conditions highlighted in yellow, and also the last 3 compounds served as negative controls, where you can see also very stable conditions or only mild seizing effects upon higher concentrations and longer durations of the analysis. So in total, we were able to show nicely that the Flexside technology serves well as a chronic cardiotox analysis method. And I think I won't go too much more into detail because there's still something else I would like to show you. And the next step in this case is the comparison of human iPSC-derived cardiomyocyte contractility cultured in serum versus serum-free medium. This was a project that we've performed with CDI, so we used iCell cardiomyocyte squared and also the provided serum-free medium. We cultured the cells as usual onto the Flexside plates for like 6 days, and then switched half of the plate to serum-free medium, measured for 24 hours before we then also did a compound effect analysis for an additional 3 days. So in total, the cells were in half of the plate of the cells were in serum-free medium for roughly 4 days. So having a look at the mean beats that we generated out of the data, you can see the comparison of the serum-free cardiomyocyte mean beat in blue and the maintenance medium, so the maintenance medium containing serum in red, and you can see that they're very similar. But if you look at this, the serum-free medium, you see that there's a slight increase in amplitude. And this is also what we could see with the analysis here in the diagram, where we basically had to focus on all the parameters we're usually looking at. So you can see that there's a mild increase in amplitude and the beat rate, or there's a downstroke slope, which is a bit higher. And generally speaking, is still to say that there's still very similar results that we received with mild changes. So starting with the cardio-safe tyrosine kinase inhibitor erlotinib as a negative control to see how the cells work in maintenance medium. So we see here that even at higher concentrations, the amplitude basically stays the same. When we look at erlotinib on cardiomyocytes cultured in serum-free medium, you see that there's a very similar effect of the amplitude staying pretty much the same, unless if you want to be, if you want to have a closer look, you see that with erlotinib at 10 micromolar, so the strongest concentration, there's a slow decrease in amplitude. The opposite, we wanted to check with doxorubicin, so cardiotoxic anthracycline. And in this case, you can see again the effect of doxorubicin in cardiomyocytes cultured in maintenance medium. And you can see that there is a definite decrease in amplitude depending on the higher concentration of doxorubicin. And again, we were able to show a very similar effect of doxorubicin in serum-free medium, with again a slightly more prominent decrease in amplitude in comparison to the maintenance medium. So in total, we were able to show very similar results of cardiomyocyte contractile behavior in serum-free medium also compared to the maintenance medium containing serum. So finally, we're reaching the topic I already told you about in the title. So we are going to the contractility studies with light and pressure-induced pacing of human iPSC-derived cardiomyocytes. Both of the studies were proof of concept studies. And I would like to start with the optical stimulation of iCell squared that we transfected with channel rhodopsin. So we basically plated the cells onto the Flexside and onto the Flexside plates, and then 4 days later, transfected the cells with channel rhodopsin, and the day after we performed the measurements. What you can see here is the sol heads, so basically the optical stimulation system, which is 96 LEDs integrated into the lid. So you basically have one LED for each well. And the optical wavelength we were using was 470 nanometers for activating channel rhodopsin. And each sweep was 30 seconds long. So basically the duration of the stimulation was 30 seconds. So this is the data we created. So starting with the pre-stimulation situation, where you can see that the cells were beating roughly around 60 beats per minute. And then when we started the simulation, we were able to push or to stimulate the beat rate up to 150 beats per minute before we stopped the stimulation, and then saw that the beat rate went down nicely to basically pre-stimulation situation with roughly 60 beats per minute. So that was a very successful proof of concept study showing that it's easy to transfect the cells with channel rhodopsin onto flex site and then perform simulation studies. The next stimulation was the mechanical stimulation we used of ISA squared with pneumatic pressure. So in this case, the pneumatic pressure was built into the lid so that the pressure was come from above and then stretch the cells downwards, basically. There are three different ways we can use the pressure. So there's, from one hand, the static pressure that we could use for a simulation of stress, for example, hypertension. We could use dynamic pressure as a sort of mechanical training, because it's also possible to create another pro-maturation effect on top of it. And the ramp pressure, so basically increasing pressure to try to replicate the Frank-Starling mechanism. And this is exactly what we tried here, replicating the Frank-Starling mechanism, where we applied stronger strain to see whether that has an impact on the amplitude. And we had cells cultured in maintenance medium, as well as cells cultured in serum-free medium. And you can see here nicely how more pressure induced the higher membrane strain and also resulted in the higher contraction amplitude, as we were hoping to see. We also tried to create the pro-maturation effect out of constant cycling stretching over 3 days at 1 Hertz. And after that, we evaluated the data and you can the cells, and you can see that after 3 days of stimulation, there is an increase in the amplitude in comparison to the control situation, which you can also see here in the analyzed data in this box plot, that the amplitude was definitely increased. So a data that we actually quite liked seeing, because that was sort of literature-wise associated with a more mature cardiomyocyte. But what we weren't expecting was that we also got an increase in the beat rate. So usually the beat rate in the control situation is around 60 beats per minute. But with the stimulation, we had the beat rate rising up to, let's say, roughly 90 beats per minute, which is quite fast and certainly not very adult-like. But so we're still a little bit puzzled about it and have to find a solution for that. But nevertheless, we were still eager to show you the data. And with this very interesting data, I would like to finish my presentation and thank you very much for your attention. And also would like to thank Nani, and especially our partners in crime, Marta, Ulrich, Sonja, and Elena, as well as Ravi, Christian, and Wissam from CDI, and Jin from Nexel for cooperation events. Thank you very much.
Thank you very much, Bettina. That was wonderful. Thank you so much.
Thank you.
And we already have some questions for you. So I will just start right away. So Prasad is asking about how many cells are per well. So, or do you have any recommendations to perform cell counting per well at the end of the experiment?
Generally speaking, you mean?
Yes.
This is something that is definitely due to the actual commercial cell line you're using. So cells can vary a little bit, and we definitely have evaluated the typical commercial cell lines out there, all of them, and they all work nicely. But yes, the cell numbers vary a little bit and are a little bit different, but we usually give the instructions on how many cells per cell type then and help out.
Okay, thanks. Then we have some more. Let me see, a lot coming. So Chloe's asking, how long can we keep the cells in a flex site plates?
That varies as well, depending on what you want to do. So since we are a contract research provider, we are doing what our customers would like to do. So most of the data is performed on an acute level. So they're in total with pre-culturing time, like roughly 7 days. So we also can go to chronic, then we're staying with 14 days. So this is the usual frame we are working in, but we also know from collaboration events and data that it is indeed also possible to culture them for longer times.
Mm-hmm. Okay, then one more. So from Stephen, was the medium also protein-free, since there's a major concern with regards to compounds that are highly protein-bound?
Yes, I know that there's always a concern. So we used a different, for the medium with the CDI. I actually have to ask Ravi about that later. I don't know exactly if there's a protein in it or not. For the data we have performed with the Frank-Starling mechanism, it was a very reduced assay buffer, and I think there was no protein in it.
Okay. And one last one from Beatrice before we continue. Can you comment on the evolution of contractility over maturation and the differences with human myocytes? Is the amplitude very different and duration and velocities?
Indeed, as I mentioned, especially with the last data, there is a difference in the amplitude. There is an increase in the amplitude that you would expect with overmaturation. That's why we were happy to see that in with the last data I showed you. But then again, I think we have to perform some more to see whether we can also deal with the beat rate.
Okay, perfect. Thank you so much, Bettina.
Thank you. Thank you.
And I will just make a small reset and join again with Khuram.
Yes.
A myriad of people who have contributed to the work that I'm going to share today. And I don't think I'll be able to thank every single one of them. But just briefly, this list includes folks who've contributed to the work I'll share today, both internally and externally, including some of our collaborators in biotech and startups. So just to give a little background, the electrophysiology of the heart begins by depolarization of a spontaneous depolarization of a region known as the SA node in the right atrium. That wave of depolarization spreads through the heart and is captured by the electrocardiogram. But what we know is that the electrocardiogram is actually dependent on the cellular action potentials of each individual myocyte in the heart. And the three major contributors to the action potential waveform include sodium, calcium, and potassium channels, specifically the hERG channel. Much of regulatory guidance as well as preclinical development emphasizes the effects on hERG because we know that those drugs that prolong action potential duration due to hERG block also tend to prolong QT interval and can lead to proarrhythmic events known as early after depolarizations. These EADs can manifest a potentially lethal arrhythmia known as torsade de pointes. However, the role of the cardiovascular system is actually to pump blood to the vital organs and to convert electrical activity into mechanical force. And what's important to recognize is that there are additional factors related to cardiac physiology and pharmacology that have the potential to have adverse events, two of which include conduction and contractility. So to begin, we know that prolongation of the QRS interval, or slowing of cardiac conduction, is associated with an increase in mortality. We also know that patients who present in the emergency room due to poisoning or overdose often have a slowing of cardiac conduction. Again, antipsychotics, antiarrhythmics, and antiepileptics have been known to have off-target effects on cardiac sodium channels and have led to conduction abnormalities. With respect to contractility, cardiac output is an influencer of hemodynamics. And so we know that hypotension consistently ranks in the top 10 adverse drug events in hospitalized patients. We also know that control of hypertension leads to a decrease in mortality. So patients who are hypertensive often take medications to reduce vascular resistance and lower blood pressure. We've learned over time that chronic treatment in cancer patients with drugs including anthracyclines and tyrosine kinase inhibitors have been associated with irreversible cardiac damage and dysfunction, including reduction in ejection fraction and development of heart failure. So it's important to understand cardiac physiology as a whole and not just focus on repolarization risk when it comes to drug development. So what I'll do now is I'll share with you some of the technologies that we've evaluated and implemented at GSK that have helped us to provide safer compounds for the clinic. First, I'll go over a simulation that we implemented in conjunction with Gary Mirams, formerly from Oxford, now at University of Nottingham, and he'll be speaking later today. So this AP Predict tool allows us to dive into high throughput screening data within GSK, use that data to simulate what might happen in a cardiac action potential. We can simulate over a broad range the effects of hERG, sodium and calcium channel blockade. And the reason we've implemented this is to allow us to visualize a more complex phenotype of our drugs. Our drugs are often not just hERG blockers. And so we've used this simulation to help us refine the use of animals in drug development, as well as to predict clinical outcomes. Here we show how we've implemented this paradigm analogous to the SIPA paradigm, in which the simulation uses our sodium, calcium, and hERG data, and we then support that with a stem cell cardiomyocyte platform using a multi-electrode array. And you can see quite nicely that we're able to mimic empirically that simulation or prediction. What we lack, though, is a better understanding of the kinetics or binding properties of molecules. We know that drugs such as class 1 antiarrhythmics, known sodium channel blockers, fall into 3 subsets of categories which confer safety. Those are class 1A, 1B, and 1C. Class 1B molecules tend to behave as anesthetics. They have very rapid onset and offset kinetics and tend to be clinically safer. Class 1A and C have been associated with conduction abnormalities as well as repolarization risk. So what we did was we recreated these biophysical properties using classical voltage clamp techniques. Here we show reference compounds of various classes and how by changing the stimulation rate, we're able to capture the frequency dependence of class 1B molecules. Again, this has been published in the past, and we were able to recreate this using our own patch clamp rigs, basically. What you'll notice, though, is that these class 1Bs, again, behave very much like anesthetics in that they have marked frequency dependence. We wanted to understand whether our preclinical assays were able to pick up this biophysical property without having to do rigorous, labor-intensive voltage clamp electrophysiology. So we tested our hypothesis in the rabbit ventricular wedge preparation. And what we found was, for the most part, class 1B antiarrhythmics do in fact show a shift in the potential to prolong QRS interval in a frequency-dependent manner. And the same was true for the class 1A and C, which showed less of a response in a frequency-dependent manner. However, we did find a disconnect in lidocaine, which is a known reference agent that has anesthetic properties and should show that frequency dependence. What we do know about lidocaine, however, is that it is also a strongly voltage-dependent inhibitor of sodium channel and also has a significant amount of tonic block at rest. So again, we're left with having to use labor-intensive voltage clamp techniques to really understand the kinetic properties and thereby the cardiac risk of our drugs. So what we did then was, again, wanting to recreate those properties and understand whether we could capture those, we used classical voltage clamp protocols and this very complex protocols to look at both on and off rate kinetics. And what you'll see is that the class 1B molecules had very rapid onset and rapid offset kinetics of binding to the channel, whereas the class 1A and 1C molecules had intermediate and slow kinetics. Again, requiring the use of voltage clamp techniques to really understand the biophysical properties. But there are more technologies and assays that we have available to us now, especially over the last few years, to understand this potential biophysical risk. So first, I'll talk about the stem cell cardiomyocyte MEA assay. you're probably very familiar with the spike amplitude measure and this sort of trace. And what you see is that a molecule such as quinidine causes a reduction in spike amplitude over a concentration-dependent manner. We were able to recreate that same property or endpoint in using various class 1 antiarrhythmics from A, B, and C. And again, you see a very characteristic reduction in spike amplitude. What we found very interesting about this assay, though, is that class 1B antiarrhythmics, which we know have rapid onset and offset and are voltage dependent, tended to cause a reduction in spike amplitude and cessation of beating at 30-fold below their sodium channel potency. Class 1A and C, however, fell within 3 to 10-fold of their potency. And what that suggested to us was that not that these class 1Bs were more potent, but rather perhaps it's a function of the substrate. We know that stem cell cardiomyocytes have a depolarized resting membrane potential when grown in this platform. And so perhaps it's that voltage dependence that's actually causing the sensitivity to the class 1B antiarrhythmics. So maybe that could be used as a property to categorize drugs in the class 1A, 1B, or 1C categories. Now, to gain a better mechanistic understanding of the cardiac risk of our drugs, we evaluated additional technologies. In this case, we looked at the optogenetics platform created by Q-State Biosciences, which uses channel rhodopsins that are fluorescent proteins transfected into cardiac myocytes, stem cell cardiomyocytes. And these proteins serve as reporters as well as actuators or sensors. What we could do then was we could control pacing rate using optical stimulation. We could also measure action potential prolongation, such as with quinidine, the initiating proarrhythmic event, the EAD, and further, we could look at upstroke velocity or rise time of the action potential, which is a surrogate for conduction. Additionally, we were able to use calcium fluorescence from these proteins to simultaneously measure changes in action potential as well as calcium homeostasis, understanding that drugs that block the L-type calcium channel, for instance, can cause a change in contractility as well as in hemodynamics. And so this could serve as predictive of changes in contractility and hemodynamics in large animals as well as in the clinical setting. So again, providing more mechanistic understanding of the overall cardiac physiology and pharmacology. So next, I'm just going to follow up a little bit on what Dr. Lickiss just presented earlier today. We've been evaluating the FlexSite assay developed by Inovitro and Nanion, which replaces the hard substrate or 96-well plate with a flexible membrane. The stem cell cardiomyocytes are grown on these membranes, and you can actually measure shortening and contraction. We wanted to start with some very basic experiments to understand the utility and the limitations of this assay. So what I'll share with you first is a calcium challenge. We know that in adult myocardium, increases in extracellular calcium lead to an increase in contraction or contractility. Normally, our standard endpoint when using a plate-based assay, especially with stem cell cardiomyocytes, is roughly 60 minutes. What we saw was that, in fact, an increase in extracellular calcium did lead to an increase in contraction. However, at 60 minutes, there was some dysrhythmia as well as some calcium mismanagement, especially at the highest concentration of calcium. What we learned was that if we were to capture earlier time points, we could actually recapitulate that increase in contractility based on extracellular calcium. And that helped us to understand the types of studies we would design subsequently for positive inotropic agents. Here we have an example of a drug that's been in the clinic for decades now, digoxin. This is often given to patients with heart failure, and it causes an increase in cardiac contractility. And again, at the 60-minute time point, we see quite a bit of dysrhythmia. We know that digoxin does cause delayed after depolarizations and calcium overload. But if you were to measure intermediate time points, you can actually recapitulate that increase in cardiac contractility as well as show the progression of the dysrhythmia over time. And what we also learned with these two assays was that we had a roughly 60% increase in contractility as the peak bandwidth for our assay. And we found that once we exceeded that 60% increase, we were able to start seeing some of that dysrhythmia or calcium overload or calcium homeostasis. Now, we shifted to looking at myofilament proteins because, again, this is something that we have an interest in and is not related to the classical ion channel pharmacology. And what you can see is that omecamtiv, for instance, which is an enhancer of cross-bridge cycling, produces a concentration-dependent increase in contraction. Mavacamten, on the other hand, produces a very classical reduction in contractile force. So again, we're able to use this medium throughput platform to start understanding a little bit more about cardiac contractility and not just repolarization risk. So then we moved on to adding complexity and looking at engineered heart tissues. So we did this work in collaboration with Terra Biosystems on their BioWire platform. The BioWire platform uses stem cell cardiomyocytes which self-assemble into these engineered tissues, and they adhere to these silicone wires. From this preparation, we're able to measure contraction, as well as fluorescent-based action potentials and calcium transients. We can also stimulate these preparations, and we can do disease modeling as well from patient samples. What we published a few years ago was that these engineered heart tissues actually do retain mature signaling, including protein kinase A pathways, calcium homeostatic proteins such as L-type calcium channel and SR calcium ATPase, as well as a myofilament activity. So we've studied mavacamten, omecamtiv, and levosimendan. So again, the engineered heart tissue was able to recapitulate more complex physiology for us. But what I've shown you thus far is basically a recapitulation of what we've always known about acute cardiac physiology and pharmacology, especially at the myocyte or tissue level. What we wanted to do was understand whether we could exploit these technologies to take things one step further, not just look at acute effects of drugs, but rather chronic treatment-induced effects. For example, we use the optogenetic platform and the channel rhodopsins to understand whether pentamidine, a known hERG trafficking diminisher or hERG trafficking modulator, would cause a change in action potential duration. And in fact, over 20 and 44 hours, we can start to see a prolongation of the action potential duration, as well as the genesis of the early after depolarization. So again, we could mimic a chronic treatment paradigm. Next, we use the BioWire platform to look at molecules such as insulin growth factor 1, and found that over several days of culture, we saw a positive increase in contractility. We then tested doxorubicin, which, as you know, is an anthracycline often given to cancer patients and has a myriad of cardiotoxic effects. We were able to see sort of a bell-shaped response over time to contractility. We know that doxorubicin is a mitotoxicant and causes all sorts of issues related to reactive oxygen species and energetics. And so again, this system was allowed us to culture tissues for longer periods of time and to really interrogate these chronic treatment-induced effects. Now, something we've been digging into lately are alternative uses for our stem cell cardiomyocyte platform right from the MEA. So can the MEA serve as a surrogate for additional toxicities, not just electrophysiological risk? So what I want to show you here is an example where the MEA platform actually allowed us to investigate mitochondrial toxicity and energetics. So in the top left, you see a characteristic response to difetilide. Difetilide is not a mitotoxicant, but what you see is an increase in field potential duration in a concentration-dependent manner, here in the blue. What you also see is a dissociation between that field potential and an increase in the beat period, so a slowing of the spontaneous beating rate. Now, we've known that as a sort of fingerprint for hERG blockers in this platform. Interestingly, we tested rotenone, which is an uncoupler of the electron transport chain, and found that rotenone in a concentration-dependent manner actually increases that beat period or slows down the spontaneous rate in a manner that's dissociated from any change in field potential duration. That's a very interesting phenotype. We didn't see the precursor sort of hERG-dependent prolongation, which led to the change in rate, but rather a slowing of rate on its own, independent of field potential duration. We were able to also recreate that with a ligamycin, again, a mitotoxicant, over time. So over 24, 48, and 72 hours, we see a prolongation of that beat period. So what we're doing now is identifying this type of phenotype, one where either you see the field potential duration increase first, or you see the beat period slow initially. And then we're suggesting whether these molecules should be further tested for mitochondrial toxicity. So in summary, I think it's really important to understand cardiac physiology from a viewpoint that is more all-encompassing than just hERG. So measurement of cardiac conduction and contractility, and thereby hemodynamics, is recommended in ICH-S7 guidelines, but really far downstream. The emphasis is placed on the large animal studies late in development. And little emphasis has been placed on these studies early in preclinical development in comparison to hERG. Most of the automated patch clamp systems and these technologies have been developed around repolarization risk and translation of hERG potency. But we know that conduction and contractility are both associated with increased mortality, especially drug-induced. While patch clamp electrophysiology remains the gold standard for ion channel pharmacology, tools such as in-silico modeling, the ex vivo preparations I spoke about, as well as these cardiomyocyte utilizing technologies, can bridge that translational gap from preclinical to clinical outcomes. It's the creative use of these stem cell technologies that will allow us to shift from just looking at acute exposure-driven toxicities to chronic treatment, which really is the case for drugs such as oncology targets. And finally, the future of non-clinical cardiac safety will incorporate these tools to produce safer medicines for patients. So I want to thank you for your time and for listening to me talk about how we've evaluated novel technologies at GSK. And I'm happy to answer any questions.
Thank you very much, Khuram. This was really amazing and wonderful overview. I liked specifically how many different technologies you were actually using in your research. And since we're now waiting for a couple of questions, maybe one from me to start. When you showed so many assays, and what is, in your opinion, what would it take for an assay to be adopted in the preclinical drug discovery and safety paradigm?
Well, there's really two parts to that question. One, as an overview, I know recently there has been quite a bit of discussion in ICH on validating novel assays. So clearly that discussion is happening. Mm-hmm. But on the one hand, it's important in the pharmaceutical industry for us to make the appropriate business decisions to develop the highest quality drug. And that's more of a resource issue, right? We want to reduce costs, we want to efficiently get into the clinic, we want to predict these toxicities early on. Secondly, there's a regulatory component. So, you want to ensure that you're doing the right studies as part of your regulatory submission or your follow-up studies to reduce cardiac risk. And so it's really a matter of understanding both that early development issue and the business decision, as well as reducing attrition late in development. I think all of these technologies have utility. One thing I haven't talked about are things like the use of primary myocytes. That's a hot topic as well these days. And again, it's really a fit for purpose type of paradigm. Whatever you need to ensure that you're producing the highest quality drugs is what you should be implementing.
Thank you. Maybe a very short one before we continue. The experiments that you've done with mitochondrial toxicity in MEAs, and you tested rotenone. So did you also take a look at if there's some cellular toxicity directly on the cardiomyocytes, if the cells are affected? I mean, in neurons in Parkinson's disease, for example, rotenone is the #1 toxin, so to say.
Yeah, it's a really good question. So there's a few ways to look at this. We have a more comprehensive way of addressing these issues that falls under investigative safety, where we look at high content imaging, we look at cellular structure, we look at mitochondrial toxicity using energetic assays, ATP assays. But what we found was that when we were studying company compounds and saw this very unique profile, and then looked at that investigative work to see if there were any links to either screening studies, off-target effects, or even some of these additional toxicities, including mitochondrial toxicity, there was a link between mitotoxicants, and this fingerprint that we saw. That's when we went back and looked at some of the reference compounds and found that depending on which complex in the electron transport chain you tinker with, you get a very unique pattern in the MEA assay. And so we're not suggesting that we're going to use the MEA to predict mitotoxicity, but it is a surrogate that gives us a signal that we reproducibly see that relates to mitotoxicity. And so it might be the trigger to do further studies downstream.
Thank you so much, Khuram. Thanks a lot. So I think in the sake of time, we will continue now. So if you can stop sharing. Thank you very much. Now we have our next speaker. So this is Dr. Ravi Vaidyanathan. So Ravi is coming to us now from FCDI, and he has obtained his PhD in pharmacology from State University of New York Upstate Medical. And then he continued his work on cardiac ion channels as a research fellow at the Center for Arrhythmia Research and also as a postdoc at University of Michigan. And after that, he also worked in University of Wisconsin in medicine, where he basically did research and development of iPSC-derived cardiomyocytes. So that was a very nice transfer to his current position, where he is the product manager for cardiac portfolio at FCDI. Ravi, the stage is yours. Please share the screen. Then we see if all is working, and then I'll leave you to it.
Can you see my screen?
Yes, we see you. We hear you. Have a nice talk, Ravi.
Perfect. Thank you very much, Elena. Good morning, everyone, for the people who are in this part of the world. And then, of course, good afternoon to everyone who is in the other parts. Thank you for the organizers for giving me this opportunity. And today I'll be talking about physiologically relevant and predictive 3D triculture cardiac microtissue, how to build them with isol cardiomyocytes, isol endothelial cells, and primary cardiac fibroblast. So a brief outline of my presentation is I'll go over the cardiac portfolio, how to create and culture 3D triculture cardiac microtissue, the characterization that we have done of these microtissue, some compound effects, and then finally summary and conclusion. So as you know, Fujifilm Cellular Dynamics was built on iPSC technology. We can use peripheral blood cells or skin samples from healthy and patient-derived patients to and use episomal reprogramming vectors to reprogram the cells to iPSC-derived iPS cells. They can also be genetically engineered. This is a service we offer. Then these can be differentiated into multiple cell types. Today, we are going to focus on mainly the use of cardiomyocytes and endothelial cells. The other aspect that we do is to build application protocols that can support the use of these cells in various aspects of basic science, drug discovery, cardiotoxicity, and cell therapy to relate them back to how patients actually respond to drugs and such. Overview of the cardiac myocytes. We have the first generation cardiomyocytes, which we have apparently healthy donors, disease donor lines, and isogenic controls, and also genetically engineered disease models. We also have the cardiomyocyte squared line, which is the second generation of cardiomyocytes that is currently being used extensively for cardiotox, and then for customers interested in cardiac development, we have the cardiac progenitor cells. Our cells have been part of the SIPA study right from the beginning. We have been working with the FDA, HESI, and also with the Japanese SIPA, I would call it, where our cells are kind of the gold standards for safety testing. Jumping right into what I wanted to talk about today. So as you know, the heart is not only made up of cardiomyocytes, there are other cells too. What we decided was, why don't we take some of these cells, put them together, and how they behave? So that's what was done. So you can buy isol cardiomyocytes, isol endothelial cells, and primary cardiomyocytes, thaw them right out of the vial, combine them in the ratios that is shown here. Cardiomyocytes at 75%, fibroblast at 15%, and endothelial cells at 10% in these prime surface spheroid plates. You can use any plates, but these work really well. And combine them in the eye-cell plating media, and you start seeing that in a few days, these cells merge to form these microtissue. For feeding them, you need to use a combination of eye-cell maintenance media and the promocell endothelial media at 80 to 20% ratio. And you can do half feeds every 2 to 3 days. And we saw that after 14 days in culture, these cells were ready for assay. And I'll talk about that a little bit. So what you see here are photomicrographs of these spheroids in culture, days in vitro 4, 7, 10, and 14. And if you see, they're nicely growing in size. That is shown here in panel B. They range anywhere from 350 to about 450 micrometers in diameter. You can make them in different sizes depending on the plate size. We have created them with total 5,000 cells or 10,000 cells. And then the diameter, of course, is not significantly different between the two, the spheroid sizes. What you see here in panel D is a H&E stain of one of these spheroids. This is a 10,000 spheroid made out of 10,000 cells total. And you can see that, that it doesn't have a necrotic core. So it's actually a completely viable spheroid in this case. We also looked at the calcium dynamics of these cells using the cardiotox dye from molecular devices. The 5 and the 10K triculture spheroids or cardiac microtissue did not have any significant difference between them. Also, the breed rate was similar. What we did is we wanted to compare them, the performance of these cells with spheroids that were created with eye-cell cardiomyocytes only, and compare them with the triculture spheroids. So that was created using this protocol that was a collaboration between FCDI and molecular devices, where Sirico et al. Actually, what she did is combined, just took eye-cell cardiomyocytes and they formed spheroids in 4 to 5 days and can be used for assay. When we did that in comparison with the triculture spheroids, we saw that the signal strength of the triculture spheroids were stronger, the amplitude was bigger, and this is summary data shown in panel D, where you can see that there is significant difference in the signal strength in amplitude compared to pure cardiomyocyte spheroids, which is shown in black here. And this was done for 5,000 and 10,000, and they were significantly different. We also looked at the positive inotropic effect of these 3D triculture spheroids and compared them with pure cardiac spheroids. So they did show chronotropic effect. With increasing concentration of isopaternal, you can see that the beat rate increased. We see that with cardiac spheroids too, but the effect was much more prominent in the triculture spheroids. The most exciting part of this study was the inotropic effect. So if you look at the pure cardiac spheroids, they tend to have a flat line. So they don't tend to have the chronotropic effect, which is an increase in amplitude. But with the triculture spheroids, when you put these 3 cell types together, there's some changes that are happening to the behavior of the cells where they tend to respond nicely to increased concentration of isopaternal. This was done using, again, calcium imaging, but there was a paper that was published by Burham et al. In 2020 where they created these same spheroids, but what they did is they also co-cultured them with gold and iron nano beads. So what they could do is they could, these spheroids tend to float, so they could position them using a magnetic plate to the bottom of the cardioexcite system. And when they challenged those with isopaternol at 100 nanomolar, which is shown in the dotted line, you can see that the signal strength increased. And when they normalized it, they also saw that there was shortening of the the impedance with the force, which is the impedance is the surrogate for force. So they saw that too. So what I wanted to show is that it is not only in our hands, but also others have shown that by combining multiple cell types, you get this benefit. So in summary, what we have done is we have combined isol cardiomyocytes, isol endothelial cells, and primary cardiac fibroblasts directly from thaw, and we can generate 3D triculture cardiac microtissue efficiently. These 3D triculture microtissue demonstrate spontaneous beating and a robust calcium signal using the early tox cardiotoxicity dye. The 3D triculture cardiac microtissue show positive inotropic effects, suggesting that the isol cardiomyocytes are further maturing in the 3D environment. And what we have done is we have also built an application protocol around this data that I've shown you, and that is available on our website. And you can also contact our your sales representative or FASs from FCDI to get access to these if you don't have access to them. So I'm ready to take any questions.
Thank you, Ravi. Thank you very much for this talk. So let us then go through the questions. So far we have 2 now for our listeners. From Andre. Hi, Ravi. With spheroids greater than 500 micrometers, did you observe a hypoxic necrotic core?
No. So the maximum size that we tried were the 10K total cells, and the image that I'd shown was for the 10K, so the largest spheroid that we could have, and we didn't see any necrotic core, at least from the experiments that we've done. Okay.
And Chloe, she's asking, with what kind of analyzer can we use the spheroids? I guess you touched base already, but maybe.
Right. So yeah. So of course, any type of imaging, but also with some modification, it can be used on the flex site. Also, I believe it can be also used on the NANA and cardiac site. I think there might be a little bit more work to be done, but if it works on the cardiac side, it can be worked on most NMEA platforms too with some modification. But I think the easiest one is, of course, imaging and what I showed you with the cardiac, because there's data already available on how to do it.
You mentioned imaging. Would that mean that that they would work in higher throughput, like 384?
Right. Yes. So we have done some experiments. So all you have to do is decrease the total number of cells. So on a 96 well plate, we recommend anywhere from 5,000 to 10,000 total cells. But if you're going, say, 384 well plate, you can go down to 2,000, 2,500 cells total.
Perfect.
Yeah, and still keep the same of cardiomyocytes, fibroblasts, and endothelial cells.
Perfect. Thank you, Ravi. So now we will switch to the next talk. So I will welcome Stephen Jenkinson. So Steve is the Director of In Vitro Pharmacology at Pfizer. And today he's joining us for a talk on where he will focus on stem cell cardiomyocyte models in predicting clinical cardiac endpoints. Stephen got his PhD in the University of Leicester, and he was also going back and forth over the pond during his career. So after his PhD, he then moved to Scripps Research Institute in La Jolla in California, where he was actually in the lab of Nobel laureate Jared Edelman. And after that, he transitioned into large pharma. So he was at Pfizer, at GSK, at Mitsubishi Tanabe Pharma, before rejoining Pfizer within the safety pharmacology group. So as I mentioned, currently, Steve is director of the in vitro safety pharmacology group, and he's responsible for all in vitro safety and secondary pharmacology profiling within the company. So welcome, Steve. Thank you very much. Maybe we try to share your screen and hear you speak, and then I'll leave you to it.
Hi, can you hear me okay?
Yes, yes.
Let me know if you can see my screen. I'm not sure if it's sharing properly.
No, not yet.
Okay. Is that better?
Yes, it's getting better. Perfect. If you go into the presentation mode. Excellent. So we see you, we hear you. Thanks a lot. Enjoy your talk.
Okay, thank you. Yeah, so thank you very much again for the invitation to speak today. I always love working with you guys at Nanion. You've been very helpful to us in the past, and look forward to continuing to work with you in the future. So today's talk, as you can see, is really focused on using the stem cell cardiomyocyte model and trying to understand how we can predict clinical endpoints. And obviously this is very important for us within the drug discovery field. So our main focus is really looking at developing these sort of stem cell models. Importantly for us, they have to be predictive of the clinical cardiac endpoints, and the important word there being predictive. And what we've really been focusing on over the last year or so is generating assays that can predict multiple endpoints. And really we're focused on two main endpoints, or two main sort of aspects here. And this, in today's presentation, I'll kind of show you two different models that we're doing or evaluating to address both of these endpoints. And one is the electrophysiological endpoint, as you've heard before from other speakers, really focusing in on, looking at QT, QRS prolongation, for example. But we're also looking at the functional aspects as well. And again, the speakers before me have already touched on this, and looking at, for example, contractility. And our goals within Pfizer are really to replace the older systems, as you heard earlier on as well with regards to Langendorf, very low throughput systems relying on animals, and questionable, clinical translatability as well. So what we're trying to do is develop, these models to replace those models, and again, higher throughput, more clinically translatable models. And again, also touched on earlier on is also, the impact that potential modifications in ICH S7B could have to the way we do things. And so any assays that we plan to develop, we really want to make sure those assays are validated, and will address the guidance that, the updated guidance that's coming through from ICH S7B. And again, multiple talks mentioned this already, there is this drive to also get these models to be both serum and protein free. This is very important within industry. We need to understand, what the potency of our compounds are in these models in serum preconditions, because a lot of the compounds can have significant protein binding, which can cause a major shift in your IC50, for example, of our compounds. So the need to have serum preconditions is very important. And then long-term effects, whether it be with regards to HERG, as Khuram mentioned, with, for example, pentamidine being a HERG traffic blocker, these are sort of compounds that we would miss if we're looking at them in, very acute assays such as Langendorf, and we would only really find these issues out when we get into longer term, maybe in vivo assays. So again, having an assay that's longer term and allows us to look at those effects over a longer period is very, is basically vital for what we're trying to do within our group. So we'll split the talk into two sort of parts here. One is really looking at the electrophysiological assay that we're kind of developed, and the second part will be looking at the functional assay. But before touching on either of them, I just wanted to highlight this slide, which is really just an analysis we did to look at, what genes were expressed in stem cells versus purified human ventricular cardiomyocytes, and we obtained these from Anabios. And we did a gene expression analysis. We will be doing protein expression analysis, just to look at the differences. And as you can see, there is a large number of genes that are similar between the two cell types. Roughly about 80% of the genes are similar. There are significant differences, though. However, for obviously an electrophysiological assay looking at action potential, we're kind of more concerned at a high level, at least, looking at the ion channels and seeing what ion channels are different, which are the same, and are the ion channels that are present that are kind of really responsible for the majority of the action potential, are they present at the same sort of expression level? And as you can see from across here on the right hand side, if you're looking at NAV 1.5, CAF 1.2, or if you're looking at HERG, the expression level is very similar. Other channels that are responsible for other currents, you can see as expected, are slightly different. So overall, the expression level for the channels we care about the most are intact and are similar between those two systems. So now I'd like to introduce you to the technology that we're using to try and look at, you know, this sort of electrophysiological endpoint. And we've decided in-house to go with a system that looks at voltage sensitive, using a voltage sensitive dye. And this allows us to, using the Bolt system from PhotoSwitch, allows us to look at action potential duration in a very, very sensitive manner. As I say, it allows us to look at readings in a 96-well plate-based format, and with the frequency of recording at 10,000 hertz in serum-free conditions, these are the sort of traces that you typically get at the bottom here. And as you can see, the recordings are basically equivalent to patch clamp quality. So it allows us to really look at small changes, for example, in action potential duration or rise time, and look at it in a very, very high level degree of, you know, the quality is very high degree. So just wanted to sort of give you an overview of what the data kind of looks like before I dive into looking at larger data sets here, just to show you what you know, really again, just emphasize the quality of the data that this system can generate. And so again, the typical endpoints that we're looking at are action potential duration, which can either be APD 90, 50, or any other combination in between. We can look at action potential rise time, so the very brief rise time from baseline to the peak here. We can measure very accurately because, again, of the high resolution of the measurement. We can measure triangulation or calculate triangulation and look at beat rate as well. So on the right-hand side here, just some example traces. So raw traces, just highlighting some of the standard compounds that we would normally profile with regards to this sort of EFS assay. So obviously difetylide, you can see a concentration-dependent prolongation of the action potential with some early after depolarizations kicking in here at the higher concentration of 10 nanomolar. If we look at a calcium channel blocker such as nifedipine, you see a shortening in the action potential duration. And then for pentamidine, this trace here is actually taken at 24 hours. Acutely at 30 minutes, you would not see any effect at all, but you can see at 24 hours, you're seeing this large prolongation in action potential duration. And since we have this 96-well plate format, it allows us to generate a lot of data very quickly. It is really more of a high throughput system than most of the other systems that we've used in the past. And so, and again, because we can use it acutely and chronically, we can use the same plate to look at the effects over both time points. But we can generate full concentration response curves for each endpoint here, looking at difetylide, either EPD 90, 50, or 20. Same for nifedipine, and as you see, pentamidine, no effect, but at 24 hours here, you're seeing more of a robust response. What we also looked at as well was to look at some rate correction. Again, in ICHF7B, there is a discussion, or it is highlighted that it would be preferred if the data could be rate corrected. This is the standard RR plot versus APD 90, and you can see here this is just for control wells on a plate over multiple plates, and you can see the nice correlation there. If you use a Fredericia correction, you do see a reasonable correction. However, as reported by Yamamoto, slightly different modification by that process, you get a much better sort of slope of the curve here. And so moving forward, all the data we present that we call corrected will be Yamamoto corrected APD 90 down here. So another point that came up again in multiple previous presentations was the concept of long-term incubation and mixed with serum-free media. And of course, when you put two of those together, it doesn't usually end well. Cells do not like to be in serum-free media. Usually you have to include some protein to keep the cells happy. That's not what we found with our assay. We've managed to find a system where we can actually keep the cells relatively comfortable for up to 6 days using this Fluorobite media, which is just a regular media that you can purchase from Thermo Fisher. We also added Hebes to it just to like pH balance it a little bit, since plates have been removed from the incubator quite a few times during the study. But this media is serum free and protein free. And as you can see here, if you look at multiple different endpoints, whether it be APD90, corrected 90, rise time, or beat rate, the response that we get is relatively stable across a 6-day period using the voltage sensitive dyes. Now, what I'll say here is that with the CDI media, this doesn't look good, and I apologize to Ravi. This is not a reflection of your media. The media is fantastic. What it is with this voltage sensitive dye, as we wash on day 2, what we think is actually happening is that the voltage sensitive dye is partitioning into the serum or the protein in the media, and that's why we're seeing this decrease in signal. And you know, the one thing that is great about the voltage sensitive dye we use is we load it once, and the same dye is there for 6 days, has no toxic effect, and it allows us to measure for 6 days. So unfortunately, this is a little bit of an issue for us if we were trying to use the CDI media. But again, our aim is to get away from using protein containing media. So with regards to the main emphasis the main focus I'd like to talk about today is really the translation of our data to clinical endpoints, and that's been the main thrust of what we've been trying to do over the last year. And so with regards to ECG effects of compounds, you know, we really split this into three sections. One being hair block, where the clinical effect is obviously an increase in QT interval. With our stem cell model, what we've been looking at with regards to that is an increase in APD90 duration. With regards to channel block with sodium channel, we're looking at clinical endpoint there is QRS that we're interested in, and we feel that the best endpoint there is really looking at an increase in the action potential rise time. And with calcium channel, obviously that can result in an increase in PR interval, and we've kind of focused there on looking at a decrease in APD90. We also looked at our correlation to Torsades de Pointes. This is obviously a low incidence arrhythmia that's linked to hair channel inhibition, but it's difficult to predict just using hair channel data alone. And we weren't sure which endpoint would be the most optimal there, so we did some modeling to try and predict which endpoint would be the most useful to define that. So now I'll dive into the data. And so this first slide is looking at our translation of the stem cell data, the APD90 data, to clinical QTC data. There's been a lot of emphasis on Torsades, but one of the key factors is trying to understand, will your compound produce a greater than 10 millisecond change in QTC interval in the clinic? And so that is our main focus, is trying to understand, can we predict that from our stem cell model? And as you can see here from the graph, what we've taken is the compound concentration producing a threshold increase in APD90 on the y-axis here. And for us, a threshold is 3 times standard deviation of the over vehicle. And we've compared that to the clinical free exposure that produces a 10 millisecond change in QT. And as you can see, we end up with a very nice correlation for the compounds here. And the linear fit allows us to do an estimation of the exposure the exposure that may produce a 10 millisecond change in QT using the information that we're obtaining from the stem cell model. When we performed a 4-fold cross-validation analysis of this to really understand the predictivity, we ended up with an R-squared value of 0.84, basically meaning that the in vitro data can explain 84% of the variability of the clinical data. What was also nice was that the prediction error was not huge. So if you look at the root square mean here, which is actually in log scale, if you actually convert that back into just fold change, the average prediction error is around about 4-fold. So not perfect, but considering we're using stem cells and clinical data in combination, I think that's a pretty reasonable error on that data. Next, moving on from the QT aspect, we did obviously look at the Torsades risk aspect. So for this, we analyzed data from 66 clinical compounds that had defined TTP risk associated with them. And what we used mainly here was Credible Meds and also the SIPA data set. What we were looking at, just so we're clear as to what we class as Torsad positive, that would be in credible medicine known risk, or in the CIPA list, it would be the high risk category. Those were classed as TDP positive. All the other compounds were classed as TDP negative. If we looked at several single endpoints, and I'll show you on the next slide the highest value for the highest AROC value model, but for multiple single endpoints, we showed a TDP prediction with an AROC value around about 0.9, which in itself is quite impressive. However, if we did a random forest model trained on 4 variables, that was found to be the most predictive overall. And what we're looking there was at the maximum corrected APD90 and corrected APD50 within a 30x exposure of the effective free therapeutic plasma concentration at 24 hours. That actually got us quite far. One thing that pushed it a little bit higher for a couple of compounds was actually just to use the maximum corrected APD90 and corrected APD50 at any concentration at 24 hours. When this was run in a 20 times repeated 5-fold cross-validation, we ended up with an AROC value of 0.938 with the sensitivity and specificity, as you can see below here, and confusion matrix here as well for those that are interested. So again, quite a robust translation for this data set. And I just included some of the other models that we used as well, just to show what we also looked at in this analysis. So not only the top model here is the one I just described with the AROC value of 0.38. However, if we included ion channel data, and here we used hERG binding data and CAV and NAV patch clamp data, it didn't actually improve the AROC value at all. In fact, slightly reduced that value. However, if you use a stem cell and just use a single endpoint, and this was the maximum response of the Yamamoto corrected APD90 at 30 minutes, 10-fold over the effective free therapeutic plasma concentration, you actually ended up with a very reasonable AROC value of 0.9117. Ion channel data alone, we used below here, it was significantly lower, an AROC value of 0.819. However, if you took this data and you ran it through the Ohara-Rudy model and looked at the concentration that would generate a 10 millisecond change in that model, and then use logistic regression, you could actually improve that slightly to 0.845. So overall, the stem cell model seemed to be that we generated here seems to be the most robust out of all the models we looked at. One of the other aspects, and Khuram kind of touched on this as well, with regards to QRS interval, it's a major issue moving into the clinic if you have an increase in the QRS interval. And so again, to try and understand if the stem cell model could predict that, we took the compound concentration producing a threshold increase in the stem cell action potential rise time. And we compared that to the clinical free exposure that was associated with a significant increase in QRS. We then performed a receiver-operator curve on the ratios of those concentrations versus the effective free therapeutic plasma concentration. And following a 5-fold cross-validation analysis, we basically demonstrated that the stem cell model was equivalent, more or less, to the patch clamp assay with respect to predicting QRS risk. And again, here we use 34 compounds. If you look at the patch clamp, you can see that we're around about 0.97, 0.98 range. And the stem cell model, whether you looked at 30 minutes or 24 hours, we're seeing a similar sort of AROC value there. Sensitivity was similar across, and specificity was slightly higher with the stem cell model. So again, not just the stem cell model doesn't seem to be just only useful for hair block, but it is also useful for looking at sodium channel block as well. And then finally, looking at calcium channel, we used a subset of compounds here that produced a decrease in APD90, and that was kind of associated with calcium. We assumed this was associated with calcium channel block. When we looked at this data, and again, the problem with calcium channel data is trying to relate that back to the clinic is very difficult because there's not a huge pool of PR interval data from the clinical side. So we tended to focus here more on the translation to the calcium patch clamp data. And again, within stem cells, as you know, if you block calcium channels, you get an increase in rate. So we use rate-corrected exponential threshold for APD90, and this was negative threshold. And again, you can see that there's a very nice correlation here with regards to a calcium channel block, again with an R-squared of 0.87. So I think, you know, what I've tried to show here is that with the voltage sensitive dye in this model, it's a very good model for predicting and assessing, you know, clinical QT prolongation, TDP risk, clinical QRS liability, and also calcium, and helping you understand calcium channel activity as well. So now I'll switch gears slightly and look at the functional endpoints. And for this, we're obviously the second component here is really contractility. For us, we use a surrogate endpoint of impedance, and this is using the cardiac excite system from Nanoan, where we can again use a higher throughput with 96 well plate-based format. As mentioned before, this requires maturation of the cells. If you use naive cells, you do not pick up the correct pharmacology. So we use a sort of proprietary 14-day electrical pacing protocol. It's different from the protocols that are out there at the moment. It's much more simplified. It's much more amenable to what we, you know, working in industry. With regards to assay validation, we compared the results with data from ex vivo clinical samples. This is literature data. We utilize data from isolated human tissues such as trabeculae and papillary muscle. The reason for doing that rather than looking more at clinical data is that human clinical data may be confounded by secondary effects. So if you have, for example, autonomic reflexes, then you would never expect your data to match up with what you see in the stem cell model. So with regards to cell maturation, it was quite interesting what we were seeing. So, you know, with the naive stem cells, you do not tend to see an increase in contractility in the presence of positive inotropes. In fact, you tend to see a decrease in contractility. And that's what we're really kind of showing here. This is us looking at isopaternal in the unpaced cells. And you can see in the green here, this is just vehicle control. But you have a dose-dependent relaxation or decrease in impedance, if you want to look at it that way, with increasing concentrations of isopaternal. In the paced cells, however, you see this large decrease in the baseline impedance. Vehicle still has no effect, but you now start to start to see the increase in the positive inotropic effect with isopaternal. What's also interesting is when you look at the force frequency relationship in the cardiac excite, when you look at unpaced cells, there's absolutely no change in the amplitude of the response across a reasonable range of frequencies. But when you look at the paced cells, you get a nice force frequency response to those cells. So in order to validate this system, again, it's one thing to see an increase in contractility, but again, we want to relate that back to the translatability to what we see in the clinic or what we see in a human tissue. We took a series of compounds where we managed to find literature data again in a human ex vivo tissue system. And what we tried to do as well is try to really look at multiple different pharmacologies here. So not just picking a single sort of beta agonist kind of response, but looking at multiple different endpoints. And again, just to emphasize, this is all run in serum-free conditions. And so in the paced cells, you can see here that we see directionally correct inotropic responses. So nice response to isopaternal, endothelin-1, for example. And in nifedipine, as expected, we produce a decrease in the response, and disipramide again, decrease in the response. But what's also nice to see as well is that the potency correlates well, not perfectly, but it correlates well with ex-human vivo data. And so in the slide here, we're just correlating the PEC/IC50 values for the human data with the stem cell data. You can see for both positive and negative inotropes, when you put them on the same plot, you get a nice correlation. You will emphasize that the potency the slope is not 1. The potency for that stem cells is a little bit more potent down here for these compounds. For those that might have noticed that, these are actually mainly peptides here. So the concern for me is that in the human ex vivo tissue preparations, that there may be more binding to the glass in those tissue bath systems where we may have less here. So, but overall, we have a very nice correlation there. So in summary, combining the stem cell model the two stem cell models that we have really recapitulates the key endpoints that we need within our organization to look at ex vivo, that we'd normally look at in an ex vivo cardiac model such as Langendorf, and so it allows us to replace those. More importantly, the main impacts for us are it improves the understanding of clinical translation. We really now have a good handle on the clinical translatability of the data. It also allows a significant increase in throughput and significant reduction in turnaround times, which is key for our chemists, and a reduction in resources as well. And I think you would agree that what we're presenting, especially for the ECG assay, is really, it does address the proposed ICH S7B guidance with regards to stem cell models. And what's very nice as well is the assays are amenable to this long-term effects of compounds in same-free conditions. So it really allows us to look at that as well, which is something that we've struggled to do in the past with some of our ex vivo tissue preparations. So with that, I'll stop there. And if there's any questions, I'll be happy to try and address them.
Thank you very much, Steve. This was wonderful. Thank you so much. Amazing data there from you. And we also have some questions coming from the audience. So from Ron, great talk, Steve. Your stem cell APD model predicts the QDC changes very nicely. He was just curious if your in vitro fluorescence APD data correlates well to in vitro electrical field potential data sets for published or in-house compounds.
So we haven't looked at published data and compared it, to be honest. No, we have not done that. We haven't done any field potential. So our group does not run field potential duration studies. We just focused purely on looking at voltage sensitive dye endpoints, because we find this is the most reproducible. It's a very robust assay. This system works extremely well. So it's we're kind of focused on that.
Okay, maybe one more from Mike Morton. So he says, you mentioned using hERG binding data in your predictions. Does hERG patch clamp data offer improved predictivity? Binding hasn't really been considered in the SIPL paradigm.
Yeah, so it's kind of interesting. What I didn't show, we'll be publishing this data shortly, but what we didn't show here is that if you actually use just the hERG binding data alone with regards to QT, the correlation is just as good as you get from the stem cells. So the hERG binding data, at least in our hands, is equally as predictive as the stem cell data. We actually have less with patch clamp, because of the high throughput systems and some of the issues with the small volumes and sticking to the plates, we don't get as great a correlation with the actual patch clamp data from those systems. So the binding's actually been more robust for us.
Okay, thanks a lot, Steve. Due to time, I will just switch to Yin now. And maybe if you stop sharing, Steve. Yeah, perfect. Thank you so much. Hi, Yin, welcome, and thanks for accommodating the time difference. So you're the last but not the least. And just for your short introduction, so you have actually spent some time in Europe already. You have been doing your bachelor and master's degree in Switzerland, in Lausanne and in Zurich. And then you returned to Korea and you joined Nexel. So today you will talk about challenging our understanding of iPSC based cardiac safety with physiologically relevant and assay-ready cell culture media. So maybe say something. So I see if I hear you. Perfect. So we see you, we hear you. Go Yin.
Thanks, Elena, for the kind introduction. And thank you, everyone, for still being here, despite it being the last session. Just to give you a bit more introduction to me, because there are a lot of you who are seeing me for the first time. I've been working on Nexel for 3.5 years now. I've been working on neuronal differentiation and cardiac differentiation, and then transitioned away into electrophysiology focus. and am now completely focused on the business development of our products, along with our other businesses here at Nexcel. Today I'll be presenting some work that we've been doing on cell culture media, in particular trying to stay physiologically relevant and assay ready. Now these are big words compared to what we're able to do, but I hope that I can raise some questions and some thoughts. I think a lot of others have also touched on some aspects, but I believe there are some interesting things we could discuss. I will be starting with a brief intro to our company because not everyone knows who we are. And then I'll touch in about cardiomyocyte media and how it's being prepared currently. I will then present some very brief data that we have. It's not extensive because we're still getting ready to publish all of it. And then I'll conclude with some thoughts going forward concerning cardiac safety and what we have to offer. So Nexcel is a company that specializes in iPSC technology. We started off as a spin-off from Korea University, which is a university in Seoul, South Korea. We have since moved on twice, actually, into bigger facilities and have completely separated from the university. But we are proud to be the first Korean company to license iPSC technology from iPS Academia, Japan. To tell you about this in a bit more detail, we started off quite small, and we are still quite small, in 2015. When I joined the company, we were about 15 to 30 employees during that year. We are now 50 employees large, and we finally have dedicated manufacturing and R&D facilities. We are always investing into scaling up and many different projects. But in one sentence, we have 3 human iPSC-derived products, along with a cardiac safety service that we offer, and we have 2 drug development pipelines. So as I mentioned, we specialize in differentiation technology. What this means, I mean, everyone here is quite familiar, but we have hepatocytes, neurons, and cardiomyocytes. We have named them respectively the hepatocyte S, the neurocyte S, and the cardiocyte S. We have recently focused a lot on cardiac safety, not only because there's quite a lot of demand here locally and domestically in Korea, but also because we realized that to offer a proper cardiomyocyte product, we need to be able to do the cardiac safety ourselves. So that's our product. It's nothing different to what you're familiar with. These are frozen cardiomyocytes, which we sell along with the custom media that we provide. Now, this is something I want to point out. When we started the business, we contemplated selling just the cells and not the media, and have now gone completely the other way. And we believe that the media is part of the product that we offer, and which is why we, as a cell manufacturer, need to be able to produce and provide a cell media with the right components, which is why I'll be talking about the media today. So when we look at the origin of modern cell culture media, and you compare which mediums are used, you can see from the pie chart here, which is simply out of a Google Scholar search from that paper, which is referenced here. Half of all papers use DMEM, and then about the other half grossly use RPMI 1640. Now, these cell culture medias were developed over 60 years ago. I think DMEM was developed in 1959 to be exact, and were not made for cardiomyocytes purposes in mind. So the biggest issue with this is that the salt composition does not match plasma concentrations. What this means is that, like, if you look at the table on the right, underlined are the concentrations which go over plasma concentration, and then in italics are the concentrations which are lower. If you look at RPMI 1640, for example, you can see that calcium and magnesium are quite low, are too low actually, whereas phosphate is too high. And now this is one of the big issues. And then the other issue is when we discuss cell culture media in the context of cardiomyocytes, a lot of people talk about maturation. Now, I don't like the term maturation, and not because this is something we do. We also call our factors in our supplement maturation supplements and factors. But basically, this boils down into what can we supplement the media with, and popular choices are FBS. People have tried to use knockout serum replacement. A popular option was B27. Now, B27 is quite unique in that it was developed for neuronal applications, which are also electrophysiologically active. So it was kind of a natural transition to use it in cardiomyocytes as well. But in the end, from the work that we've been doing, there are doubts into what exactly it provides and does. Now, a popular choice in the past few years has been T3 supplementation with dexamethasone sometimes, and then fatty acids, along with changing the sugar source, which are good options, which we will probably include in the future as well. But I think something that has been overlooked is that action potentials are made by a combination of ionic fluxes. Now, these are done through a combination of channels. And when we change the ionic concentrations, such as calcium or phosphate, not phosphate, but potassium and magnesium, we actually affect not just the resting membrane potential, but the kinetics and the dynamics of the interactions of these ionic channels. And going further, you actually also affect the expression level of the different ion channels. So the cell, what has been proven in literature is that the cells can detect calcium levels, and then this will affect ion channel expression. Now, this is on the long term. And then if you look at the short term, a sudden change in ionic composition can actually disrupt specific conditions. For example, you can induce calcium overload, which was shown in one of the presentations before. And this is a big issue. I mean, this has to be taken into account. And there's a stark contrast to patch clamp experiments where they use thyroid solutions or salt solutions, such as thyroids, where each concentration is very specifically chosen, depending on the purpose. And it also involves precise adjustments of the different inorganic salts, depending on which current you would like to look at. The big issue with iPSC cardiomyocyte electrophysiology is that it is yet to reach the same level of control over these variables. And I have a couple of slides which show, which illustrate this issue. So one issue we ran into was that we provide cardiocyte S media, which is basically a modification on RPMI 1640 that we made. And once we provide this media to our users, and they try to use a thyroid solution or their own media, such as DMEM-based medias, for some reason, there were arrhythmia issues. So the beat would become irregular. We'd see arrhythmic events. And at the beginning, we couldn't figure out why. But this actually led us to figure, and the thing was, the reason why we couldn't figure it out was that it was very inconsistent in how the irregularities happened. And we have reason to believe that the abrupt change in calcium concentration is what destabilizes the rate, in particular because our media had a very low level of calcium of just 0.42 millimolar. And then this problem was exacerbated when we started working with Clyde in Scotland. So this came up when we were working on the 28 SEPA compounds, and then a particular one in clarithromycin, and actually in other macrolides such as erythromycin, where we would see a FPD shortening instead of a lengthening. And our hypothesis again here was that this was due to the low level of calcium. And we were able to show by simply adding calcium and magnesium to the mix to reach 1.5 and 1.8 millimolar calcium and 1.0 millimolar magnesium, we're able to reverse the FPD-CF response to a prolongation as we would expect under these settings. Now, we're already at 10 minutes, so I'll try and do this a bit quickly. We believe that there needs to be a deeper understanding of the ionic channel interactions within iPSC-derived cardiomyocytes. We have to be able to predict interactions with ionic imbalances, such as those that we would see in the clinic. A patient which comes in and is suffering from hypokalemia, for example, would react differently to arrhythmic drugs. We also want to be able to potentially differentiate different arrhythmic agents in more detail. So other presenters have discussed this as well. But basically, we believe that iPSC cardiomyocytes have the potential to do this a lot better. Now, what we will do as a cell provider is to provide the full composition of our basal media by the next quarter. We will not tell what is in our supplement, but we believe that it's crucial for everyone to know what is in our basal media, what the ionic composition is, in order to perform the right experiments and to fix the variables. We touched on this a lot, but it was not the focus of my presentation, but we are serum-free. We are also protein-free in our maintenance media. We believe this is quite important for a variety of reasons. I won't go into too much detail, but basically, for our media to stay stable and to not interact with the drugs, we believe that this is something we need to keep and we will maintain. We are one thing I want to add is we plan on providing calcium-free media as well, but this is something I'm open to feedback for. And then one last point I want to bring up is we talked about ionic channel expression levels, but we don't show any data. So there are a lot of acute versus chronic effects that need to be distinguished. And this, of course, is not just limited to our experiments, but all maturation effects. Thank you. And if anyone has any questions.
Okay, thank you so much, Jin. So maybe one short one from Ralf. So he says, nice talk. And when you added calcium and magnesium to the medium, was the reversal of the clarithromycin effect immediate, or did this require longer incubation?
So I expected it to take longer, but it was actually immediate. So this was an acute study where we would change the media just as we would in normal settings, 2 or 3 hours before the assay. And then that was enough to reverse the clarithromycin response.
Thank you. Thank you very much, Jin. And thank you everyone for listening. Thank you for our speakers. Now we will switch to sessions. So when you look at the left side of your screen next to the stage, there is also a little button called sessions. So when you go there, you will see 4 different sessions that we have planned for 2 for today and 2 for tomorrow. And those sessions are based in a way that we will have panelists in session number 1, focusing on the new role of iPSC-derived cardiomyocytes in structural cardiotoxicity. You will meet Ralf Kettenhofen, Elena Mazza from Encardia, and Simon Hilco from FCDI, and myself. And the second room will be focusing on challenges for chronic treatment-induced cardiotoxicity in vitro iPSC-based assays. And the panelists there would be Bettina Lickiss and Peter Linner from InnoVitro, and Ravi from FCDI, as well as Jin from Nexcel. So Jin, go switch there. And Sonja will meet you at that session. So we will meet here again at 16:50 to close the meeting. And I encourage you to join the sessions now. You can join them actively if you have your microphone and your camera on. And you can also just sit down and listen to what other people are talking. For that, please keep yourself on mute because only 20 people can communicate live during the sessions. Thank you very much and see you there.
So, good morning, good afternoon, and also good evening to Australia. And welcome to day 2 of our Cardiac Safety Symposium. So before we get started, I just wanted to read a couple of sentences from a book that I'm currently reading, which fits really nicely, I would say. So listen. The greatest challenge of science, its art, lies in asking an important question and framing it in a way that allows it to be broken into manageable pieces. So into experiments that can be conducted that ultimately lead to answers. And to do this requires a certain kind of genius, one that probes vertically and sees horizontally. So after having a great meeting day yesterday, I hope we can also frame important questions today and stimulate you guys to see bits of information that you haven't seen before. And with that, I would say we sort of vertically go all the way down under to Adam, and let me introduce him just quickly. But first of all, Adam, thank you so much for your dedication to this meeting. We all know it's quite late already in Australia. So Adam is head of the Computational Cardiology Research Group at Victor Chang Cardiac Research Institute and Deputy Director of the Cardiovascular Innovation Center. He received his PhD from Imperial College in London before joining Victor Chang in 2005. In 2011, he founded the Computational Cardiology Research Group, of which he is now lab head as a faculty member. And Adam has received continuous competitive funding support from several Australian research councils, as well as international funding, for example, through the Safety Pharmacology Society. In relation to cardiac safety, a major focus of his current research is using in-silico modeling and in vitro pharmacological assays to better understand drug-induced arrhythmias and to develop improved preclinical screens for pro-arrhythmic risk. So Adam, welcome. And the screen is yours now. Please go ahead.
Thank you, Sonja, for that intro, and thanks to the rest of the Nanion folks for the opportunity to come and speak here, as well as hear about all the great work that's going on around Nanion platforms. I'm very pleased to take part. So what I'm going to be telling you about tonight or this morning is our journey through developing automation of data processing and analysis, and how we've scaled up for the in-house platforms that we started with a few years ago to a hands-off, cloud-based solution that we think has much broader application to large-scale electrophysiology data sets, both for disease modeling and for cardiac safety. So everyone here will recognize the SynchroPatch platform here and the data acquisition screen of data control. And this is a system that was designed very much with drug discovery in mind. So it's good at analyzing things like ion channel hits and measuring dose response curves, or inhibitions, concentration inhibition curves. But more and more, it's being used for more complex electrophysiology projects as we try to analyze things like ion channel gating kinetics or drug binding kinetics. So the challenge that this presents to us is one of automating the data quality control, the processing, analysis, and interpretation of these more complex protocols. So the examples I'll talk to you about today cover some of our efforts trying to achieve this. So first, going back a few years, trying to automate analysis of the CEPA-Mills protocol to infer drug kinetics for the models that Ziwa Li so successfully developed at the FDA. Second, this cloud-based platform that we've now developed around high-throughput functional analysis of variants in the hERG ion channel. So lots of analysis of gating kinetics. And finally, talk about how we're looking at pivoting that platform to safety pharmacology applications. So to start off with the CEPA-Mills protocol, that turned out to really be quite a challenge. This is a paper we published 4 years ago in 2017 with the Ion Channel Working Group, describing application of that protocol to measure the kinetics of the initial 12 CEPA drugs. And the way this protocol was applied in the paper, that there were 5 repetitions of this 25-second sweep, which had a 10-second depolarization step in there to establish a baseline or a control. 5 sweeps with no depolarization step while the drug was washed in, and then 5 sweeps again with the activating step when we monitored the block. And the key to extracting kinetics is to perform a subtraction of the drug band or the drug block state from control, divide that subtraction by the control to get a percentage block, and then fit that with an exponential to derive the time course. So in terms of doing this analysis at scale on data from the SynchroPatch, there's really quite a few steps involving data manipulation and processing that really required robust automation of the analysis. So what does that data fitting look like? Well, in the ideal case, we have a nice time course of block curve that we can fit with an exponential shown in black and derive a tau very easily and accurately. Too often, though, we'd seen this sort of thing with a lot of noise in the first second or two, which tends to occur if you've just got a slight shift in the stability of the cell. So sometimes you get these transient, these times where the drug-bound trace is actually a little bit bigger than the control trace. And that gives these noisy upward hooks at the beginning. So it really throws out the fit. And this is something that a lot of operators in manual and automated patch have seen. But doing this at scale, you realize just quite how common it was. And to get around this, we tried quite a few different techniques in terms of filtering, in terms of denoising the data. But really the simplest approach to it was just truncation of the data. So we can eliminate that part from the fit. But in doing that, you've got the danger of data loss too. So particularly for drugs that are quite fast, where a lot of the information you want to capture is in this first part, it's potentially quite a dangerous thing to do. So it goes without saying that the data analysis and fitting is pretty straightforward if the data quality is good. But these stability issues are relatively common. This really reinforced for us the importance of robust quality control, not just based on the patch clamp parameters of the type we can get in data control, but also these kind of more advanced post-analysis quality controls that I'll talk about in the next few slides. So what did that QC look like? So here are the parameters we used. Some of them related to standard patch clamps, such as seal resistance, whole cell capacitance, some specific to the protocol, like current amplitude, baseline leak, and stability, and some of these post-analysis quality controls. So, you know, is the percentage of block sensible, or how good is the fit to the time course? Now, unfortunately, for the practical application of this protocol, the QC really does decimate the data. So on the left here, excuse me, we're looking at the data from 12 plates, 4,500 cells, and what you can see is that we end up losing about 93% of the data based on our QC, and most of that, over half of that, is through this stability quality control, which unfortunately we know is really important in terms of getting sensible percentage block curves. If we take out stability, we do start to see other quality control measures, such as the percentage block or the goodness of fit, eliminating data. We gain maybe another 20%, so it makes it a little more usable. But even so, we're still losing a lot of data based on these quality controls. And for comparison to what we'd see in a more conventional measure of potency, through QC, we maybe lose about 20%. So there's really something about this protocol that makes life very difficult. So in the next few slides, I'll show you data from both of these scenarios. So first of all, we're looking at percentage block measured using that Milnes protocol. So for each drug across the top here, I'm showing you 3 different concentrations, and I'll show you data from SynchroPatch with and without the quality control based on stability compared to the gold standard manual patch clamp. The first thing that you can see in terms of the percentage block is that the dose-dependent trends that we see, by and large, are comparable to manual patch. And in fact, the mean percentages are not significantly different to the manual patch. But there is a lot of variability, particularly in those cases where we don't have the stability QC. And in some cases, that ranges from 0 to 100% block. So it's a real problem. In terms of the time constant, this is potentially even more problematic. So bear in mind, these are log scales we're using now. And in all cases, for every drug, there's a significant difference between the time constants measured on SynchroPatch relative to manual. And what we also noticed is that there appears to be a trend for either over or underestimation of the time constant, which is drug specific. So for Bepredil, for example, manual patch is always faster than SynchroPatch, whereas for Cisapride, manual patch is always slower. And I really haven't got my head around understanding that, so I'd be interested to hear what people have to say. But the point is there's really some extreme variability again, particularly in the absence of those stability QC. So it's so important. The stability is so sensitive in this protocol. So just to summarize that first foray into automation of analysis of a more complex protocol, we'll start with the positives. I think we were successfully able to automate the analysis and processing. But it really showed that the Milnes protocol is very inefficient for data acquisition on automated patch clamp. And to make that point again, we're losing between 70 and 90% of the data compared to 20% that we normally see for conventional protocols measuring potency. So again, it's a real problem. And the significant component of that variability is always down to the sensitivity of the derivation of those time courses of block to the stability of the cell over time. And because it's such a long protocol, that's really something that we just can't get away from. So it's a difficult implementation on APC, as people have realized. So on to the main topic of what I want to talk about today, which is somewhat more encouraging, which is this online platform that we've built for analysis of high throughput screening of ion channel variants. So the project we use as a proof of concept for this is really being driven by Andy Ng, who works in Jamie Vandenberg's lab at the Victor Chang here. And what he's doing is screening variants in the KCNH2 gene, which codes for the hERG potassium channel and potentially causes long QT, by making stable cell lines of these variants, characterizing their function on SynchroPatch, and using that functional data to interpret the variant in terms of whether it might be pathogenic or benign. And last year they published a study in Heart Rhythm, which was essentially a pilot where they looked at 30 variants using this system. All the analysis was done by hand. But what they want to do now is scale up to what is essentially a saturation scale. So they're going to have data for every possible amino acid in the protein, which is essentially a lookup table for a clinician or a genetic counselor to come and look at functional data related to that variant to help them interpret the severity for their patient. So the challenges that poses are obviously firstly around managing data. There's the size of the data, properly cataloging, data quality and security for things that are potentially clinically sensitive. There's automation of the data analysis, which requires extensive quality control, complex analysis of current kinetics, as well as amplitudes. We wanted the pipeline for incorporating the results from the screen into simulations for evaluation of repolarization delay. We want to be able to interpret that data in terms of its significance and whether it might be a more, it might be a benign or a pathogenic variant. And finally, we wanted a mechanism for delivery of that information to diverse users, which might be data scientists, biophysicists, or clinicians. Now, obviously, this analysis of the current profiles is right in our wheelhouse, but the rest of this is the development of the technology infrastructure around this that could potentially support its use by many users around the world is much less so. So we're very lucky to start a partnership in 2020 with a company called Telstra, and with them we set out to tackle this. So for those non-Australians, Telstra are the largest telecommunications company over here. They've got a relatively new digital health subsidiary called Telstra Health. They're already embedded in health and aged care, both in Australia and in the UK. So they're very much across all those issues around security, either for clinically or commercially sensitive data sets. And the final piece of that puzzle was the Telstra Data Hub, which is a new partnership between Telstra and Microsoft using the Azure platform, which is aimed at providing secure, frictionless data sharing between global organizations. And at the time we spoke to them, Telstra, the Data Hub in particular, wants to dip their toes, so to speak, into the health space. So it was very good timing from our point of view. So here's a schematic of the Data Hub architecture that we built our system on. So the data pipeline comes through Ingress. So this is streaming data directly from our Synchro patch into the Azure cloud, managed by Active Directory. There are a series of transformers that does all the quality control, the model fitting, the post-analysis quality control in Spark. There are egress mechanisms where the process data is stored in these Postgres databases, so the users can filter very efficiently by whether it's plate, experiment, mutant, drug, etc. And then from a user point of view, we've got this application zone where we're presenting an interaction point for data scientists using Jupyter notebooks where they can really sort of burrow down into the nuts and bolts of the data, as well as developing these web apps for clinicians which present the data at a much higher view to help them manage their patients. But overall, the key advantages for us are that the Data Hub gives us cloud data storage and massively scalable compute, and it also gives us the ability to securely permission and share with no need to extract or download data. So everything can be done on the Data Hub. So jumping straight into that, the first type of user we envisaged was this, the data scientist or the biophysicist that I mentioned, who really wants to get into the data in some depth. So here's an example of a Jupyter notebook. The example here shows the file explorer with someone searching through the database. You can search on variant, protocol, what type of plot, plate, or even well, if you want to go that deep. In terms of analysis, I'll start with the simplest. This is a protocol to measure the voltage dependence of activation for the hERG ion channel. I should say in each of these next few examples I'll show, this is the raw trace from the Synchro patch. And on the right, the Jupyter notebook interaction that the user on the data science workspace would see. So in this case, we're picking the peaks. We're plotting those peaks relative to the voltage of the test pulse in the notebook on the right here, and fitting that with the Boltzmann equation. And we've got post-analysis quality control around the goodness of fit of the Boltzmann, whether it reaches a reasonable plateau. And from this protocol, we can also extract current density for each mutant. So that's a measure of whether the mutant reduces the expression of hERG, which might be expected to make it cause disease. We summarize that data in heat maps like this, showing the expression levels color-coded for each position in the protein and for each individual amino acid at those positions. And obviously these heat maps are updated as more data is ingressed into the database. Getting a little more complicated now, here's a protocol to measure the rates of inactivation of the hERG ion channel. And if I zoom into this region here, what this protocol requires is fitting of exponential functions to each of these current decays at different voltages as the channel inactivates. The example shown on the right here is a fit to a single sweep, which has been selected through the notebook. We've got quality control around the goodness of fit per sweep, around the current amplitude, because these big currents with these very fast kinetics can lead to poor clamp. And we've also presented the summary statistics where we've got the time constants over a range of voltages, and we've got post-analysis quality control around the voltage-dependent trends in those time constants, as well as missing data. So layer upon layer of quality control to make sure we get really good quality data through. Here's another one more example, this time the rate of deactivation. Again, what we're trying to do is fit exponentials to these current decays, this time as the ion channel deactivates at different test pulses. What we're fitting now is a double exponential. There's some examples here of both an outward current and an inward current. Again, we've got quality control around the goodness of fit, and summary statistics for the weighted time constants as a function of voltage, which again, we've got advanced quality control around those trends, having to see monotonic changes and missing data. So moving on from the data analysis, the pipeline also allows for integrating data from the syncopatch screens into simulations so that we can predict effects on repolarization. So here's a schematic of the myosite. We've used the Tomek model in this case, which is a modification of the O'Hara-Rudy. Each of the channels and pumps is described mathematically in the model, as everyone here will no doubt know. And I'm showing an example of a Markov scheme for hERG here, just for illustration. And what we're doing is taking the current amplitude measures from the syncopatch screen that I showed you a couple of slides ago, feeding this into the model to modify the expression of hERG, and using this to predict changes in the action potential. So again, there's a shot of the Jupyter notebook on the right, where the user's looking at a particular mutation and seeing what that does to the hERG current density and the repolarization duration. And while we haven't done it yet, we're also looking at capturing gating defects from the syncopatch screens and building those into the models too. Now, aside from the data science workspace, we've also made, as I mentioned, this clinician's application, which delivers the information at a bit of a higher level with a view to hopefully helping with patient care or interpreting variant pathogenicity. So here's a movie, hopefully, of me interacting with the web app earlier. This is the splash page, so there's a bit of information about what we're doing, the list of genes. I'm clicking through to hERG or KCNH2. And what the user's presented with here is all the different variants that have been classified, the different protocols and measures that we can look at. And the way it's presented is that your chosen mutant on the left here in red, the variant, is presented relative to known pathogenic variants of unknown significance or benign, to give you some idea of where it sits on the known spectrum. What we've selected here is wild type, so it's got a mean of 1. Everything's normalized to wild type. But for comparison, we can look at a mutant with a significant trafficking defect. So the normalized current density is very low here. You can see it's right on the tail even of known pathogenic genes. So you'd expect, as we look at the prediction for the QT interval, it's very high. There's almost a doubling relative to the normalized control. We've also got gating information in here, as I mentioned before. So here's a mutant R56Q, which we know accelerates deactivation. So you can see here that the normalized weighted tau is down here much faster than existing variants in the database. So hopefully that gives you a bit of a feel for the different types of user interactions and analysis capabilities of both the data science workspace and the clinicians applications that we've developed. So to summarize that section, we have built a pipeline on the Telstra Data Hub that automates streaming from our SynchroPatch, cataloging the data, quality control, analysis, simulation, and reporting. We've already processed about 500 variants through Andy's work, which equates to 40,000 cells or 200,000 protocols through the platform. So it's working very effectively. I think what is demonstrated is that there's a very robust capacity of SynchroPatch to reproducibly measure, complex ion channel biophysics, as well as simple measures of amplitude. And it's a great proof of concept that complex analysis and quality control of SynchroPatch data sets can be robustly performed in a hands-off environment. So lastly, I just want to spend a bit of time talking about how this same pipeline we think could be used in the cardiac safety space. So here's an overview of what I just showed you, where we're taking electrophysiology data related to genetic variants in cardiac ion channel genes, screening them on the SynchroPatch, streaming them into the cloud for processing and analysis on the Telstra Data Hub, and then presenting the data to users through applications. It's a relatively small jump from that to streaming SynchroPatch data related to drug effects on ion channels into the Data Hub for processing, quality control, simulation, and interpretation in terms of pro-arrhythmic risk. So that's a direction that we've been exploring a little bit more lately. So we began processing some initial pharmacology data sets through our pipelines and started thinking about really what kinds of advanced quality control and processing we can apply to add some value to those data sets too. So here's an example of a conventional drug potency experiment on HERG. So we're measuring tail currents and how they change in response to a drug to output one of these drug block profiles on the right here, where the left-hand side is the control measure, and then we've got a drop in amplitude as the drug blocks the channel. This is very similar to the type of thing you might see in data control currently. We're applying the regular quality controls, of course, around seal resistance, whole cell capacitance, and leak. We've also added some in for current density. And we've been thinking a bit about whether quality control around current kinetics might be something else that can bring additional value, because we've already got a lot of that analysis capacity baked in. So it's potentially something that we could add to the process. But more importantly, we've started building in some of those post-analysis quality controls to really clean up the data. And these are the sort of things that really adds a lot of value to our variant screening pipeline. So the first example on the left here is around stability, where we're doing a linear fit to the pre-drug amplitudes and assessing this gradient to indicate whether there's any rundown that might contaminate the drug effect. So here's an example of an experiment that will be cleaned up, and the user can set the thresholds around the gradients of these fits that they want to use to either exclude or include data. What we've also added in is a flag for slow blocking drugs. So this is on the right-hand side here, the green fit at the end of the drug block profile. And what this is assessing is whether the drug block has reached a plateau or not. And we can either exclude or just mark data based on that fit according to what the user wants. So what do these post-quality control, post-analysis quality controls actually do to the data sets, so in this first pass at least? Well, here's a couple of examples from ranolazine on the top, with no post-QC on the left, and inclusion of post-QC on the right. This is pretty good data anyway. What we're doing is ending up excluding 3 points from this fit, which is just moving the slope of the hill curve to something a bit more close to ideal. A more extreme example is shown down below for quinidine. For this data set, there were a couple of real outliers that really threw out the fit, making it essentially unusable. And we've got a slope of 0.17 there, which is obviously not correct. But after the post-analysis QC, we've weeded out those poor quality data points, and we've got a pretty decent fit, which is more or less usable. So this post-QC can certainly improve fits and maximize data usage for these pharmacology screens. And in addition to this, we've also got some post-analysis QC on fits of the Hill equation, much like we did for the Boltzmann in the variant screening. So we've got measures of goodness of fit, the slope of the Hill curve and how close to unity it is, the data distribution relative to the IC50, so we want to get lots of data on this steepest part of the curve, as well as a maximal block, which are all set, defined by the user, and all help add, I think, some confidence to the quality of the data. So what does this look like as part of a more complete workflow, similar to what we did for the ion channel variant? So here's a potential pipeline that we've envisaged with data streaming from patch clamp platforms, whether that's in contract research, pharma, or academia, into the Telstra cloud. And we're considering how we could offer a service based on tiers of analysis. So level 1 might involve data cataloging, quality control, and simple analysis like HERG hits and HERG dose response. Level 2 would be a little more involved, so analysis of multi-channel pharmacology, incorporating that into in-silico modeling to predict effects on repolarization, but also comparing those predicted effects to reference data sets. So on the bottom here, what I'm showing is IKL block relative to prolongation for a library of different drugs, of known drugs, where we can report the new compound in the context of those existing drugs to give a bit of a feel for where it sits in the overall context. And finally, level 3, where we might have a qualified risk prediction model running in the cloud that could be based on QNet or other, where you could stream your data, incorporate it into that risk prediction model, and assign a risk label. And we'd envisage that different users with different levels of maturity of their safety program might have different requirements for these different levels. So if you're a startup, you've just got a SyncroPatch or some other high throughput platform, you might find some value in these early stages around data storage, cataloging, and quality control, where a user with a more mature program who have got their data management in place might find more use for these latter, more in-depth tiers. So just to summarize that last part, I think the Telstra Data Hub pipeline is certainly well suited to an application in safety pharmacology that we're currently exploring. Our short-term focus is expanding the pharmacology analysis capability to more cardiac ion channels. It's very much HERG-centric at the moment, as its background is in the variant screening platform. We're thinking about a set of tiered analysis levels, offering value to users at multiple levels. And in that regard, we're looking for industry or CRO partners to explore the utility of this kind of cloud-based data processing. So that kind of partnership, could range from just some discussions about what the needs are, whether we're wide of the mark in what we're thinking needs to be done, or something more formal, helping out with the development of the pipeline with ourselves and Telstra. And I'd encourage anyone who wanted to have any discussion to contact me. My email address is at the bottom there, and I'm sure you could get it passed on through the organizers if need be. So finally, just to say thank you to some important people at the Victor Chang here. Jessica Farr is an undergraduate researcher in my lab who did all the programming, all the coding around analysis of HERG in the variant screening pipeline, and worked very closely with Telstra, incorporating that into the Telstra Data Hub. Jamie's a long-term collaborator who drove the Data Hub project with me, and together with Andy, those guys have been running with the variant screening process on Syncropatch. And Telstra, Richard Cook is the product owner or the product lead of the Data Hub, and Soren and Hank are the developers and data scientists who really spent most of 2020 developing our application on the hub. You folks at Nanayan who helped us out with some of the proprietary data formatting and streaming our data to the cloud, as well as providing some pharmacology data sets to build that last part of the analysis I showed you on. And finally, the funding bodies who have variously contributed to this, the National Health and Medical Research Council in Australia, the New South Wales Government Health, who have funded Jamie and Andy's functional genomics project, and the Australian Research Data Commons, who gave us some seed funding a couple of years ago now to start this sort of national data set adventure. And thank you all for listening. Sorry, my voice is going a little.
Now you're relieved. Thank you so much, Adam. Very great talk. We have already also a couple of questions here, and I think we have time for only one question. But in any case, all of the questions will get answered. So I'm picking this one here, a pretty general one from Ron, which I think is really nice. Very interesting talk. For those out there who are not HERG voltage clamp aficionados, can you explain why there is a critical need to implement such a protocol like the Milnes protocol, or maybe if there are alternatives to this protocol?
Well, I certainly think that if we're going to undertake any sort of high throughput screening for drug kinetics, which we probably need to be working towards if it's going to be built into any of the next phase of the super pipelines, then the Milnes protocol is a very difficult one to undertake. It just doesn't lend itself to these sort of automated platforms because there's this requirement for, they're really long protocols, really. You're essentially, the problem with stability is you can be comparing your post-drug sweep, which may be minutes, several minutes after your pre-drug sweep, and just a few picoamps of change can really give you all that noise and throw out, your potential to measure the kinetics importantly. So, it's, I think we've probably got to be looking, for alternatives. It's probably a challenge for some of the modelers to say, is the kinetic model of drug binding that we're using correct? Is there a simpler one that we could use where we could use a much simpler protocol, whether that's something like a step ramp or an action potential, and with a simpler model, have enough depth of the data to constrain that model in terms of the ion channel kinetics. So yes, I think the Milnes protocol is very hard to manage.
Yeah. I think a few electrophysiologists here know about that one.
Yeah.
Okay, so great talk. Thank you so much. We can leave the backstage room now, and we can let Jessica into the room. So now we continue with Jessica Veenman-Koepke. Jessica is a scientist and project leader at Encardia. After her education in biology in 2000, she worked at several institutes in Munich in Germany before she moved to the Netherlands in 2006. From there, she started her career in the industry, working for biotech and CRO companies. And at Encardia, her main area of interest are cardiovascular related assays using various cell types and technologies. So welcome, Jessica. Enjoy your talk.
Thank you. I'm just, can you see my screen?
I can see a black screen.
Yes. Okay, I will try again.
Yeah.
No, it's is it entire screen? Which one?
Maybe Jason, if you can jump
Yeah,
in.
so Jessica, if you just open up the presentation.
Yes. Oh yeah, console. Yes, I have. Yes.
Okay, and then depending on what you're selecting, either the entire screen or just PowerPoint as an app, should tell you. And then once you do screen share, then I can see if we're...
Yeah, so I have the presentation open, and then I can entire screen.
Yes, this works as well. And then you just jump into full presenter mode. And then go into the presentation, and then...
Yes, okay.
And then you can click hide for the little thing here.
Okay, but you can't see me, right?
We can't see you.
Oh, I wanted to, okay.
Well, the presenters, the audience would see your icon at the bottom. They still know that you're there, but you can't see your own image.
Okay.
Thank you.
Yeah, thank you very much. So thank you for the introduction, and thank you for the invitation. And of course, thank you all for joining this webinar today. So my name is Jessica Veenman-Koepke, and I'm working as a scientist and project lead at Encadia. And today's presentation will be about multiparametric drug safety assessment in our bioreactor produced inside iPSC-derived cardiomyocytes. So over the 10 past years, we established an integrated iPSC platform, which consists of iPSC resourcing and reprogramming, as well as directed differentiation into the desired cell types. Furthermore, we offer assay and assay development for identity and functionality testing. So all this we do with the aim to support our partners and collaborators to bring medicines to patients faster. If we start with the manufacturing of our inside iPSC-derived cardiomyocytes, so those are produced in large-scale steered tank bioreactor systems with a volume of approximately 3 liters or 2 to 4 billion cells of high purity and post-thaw viability. So if we look at the graphs and start with A, so here we show the purity based on cardiac troponin T expression detected by flow cytometry. And here you can appreciate that over 10 rounds, we were able to manufacture cells of very high purity, above 80 to 90% purity. So this is, so we optimized our production process as such that we obtain robust purities. And the differentiation process itself takes a little over 2 weeks, starting from the iPSC inoculation, which is shown on the top left. There you find the time schedule, until the harvest of the differentiated cardiomyocytes. And once we harvested the cells and cryopreserved them, we perform a variety of functional assays. For example, we looked at the gene expression profile and found that the cells expressed common sarcomere markers, while the iPSC and cardiac progenitor markers are either silenced or detected at background levels. Furthermore, we looked at, we found that the cells showed a ventricular-like profile using manual patch clamp techniques, and found that the cells have a resting membrane potential of approximately -90 millivolts. as well as they showed intact and well-aligned sarcomere structures, as shown in the immunofluorescent stainings on the right side of the slide. So, as you know, there are several assay platforms available to assess the safety profiles. So here we have chosen for 2 platforms. So I would like to start with the impedance platform, where we tested a small set of reference compounds of different ion channel targets. So if we start on slide 6, here we have some examples of isoproterenol, which is an example of a beta-adrenergic agonist. So if we start on the left side of the slide, here we show you representative impedance traces of cells treated with different concentrations of isoproterenol. So here we can appreciate that the more isoproterenol we add to the cells, which is shown in the orange lines, we see expected inotropic effects compared to the baseline levels, which are shown in purple. Furthermore, we observed expected chronotropic effects, as shown in the graph in the middle. And those inotropic effects are characteristic to adult cardiomyocytes and could be attributed to a higher expression of the gene ADRB1 compared to ADRB2 in our inside cardiomyocytes. And as a next step, we looked at 3 different classes of ion channel blockers. We have chosen for mexiletine, nifedipine, and tofetilide. So if we start with mexiletine on the left side, here we can appreciate a rapid decrease in the upstroke velocity at higher concentrations of mexiletine. And if we look at nifedipine, which is in the middle, here we could see the expected increase in the beat rate at concentrations around 30 nanomole and with arrest at higher concentrations. So when we added tofetilide, which is shown on the right side of the slide, here we observed decreased beat rate at higher concentrations with the occurrence of typical arrhythmic profiles at concentrations higher than 10 nanomole. Furthermore, we were also interested in effects from compounds inducing structural toxicity. Here we have chosen for doxorubicin, an example for topoisomerase inhibitor. So here we can appreciate that this compound showed a progressive decrease in the beat rate at concentrations higher than 300 nanomole, which is shown on the left, as well as a similar effect was observed on the amplitude, which is shown on the right over time. So those effects are those effects will cause dysfunction of the cardiomyocytes over time. Another assay platform that we used to assess the safety profile was the analysis of calcium transients using calcium binding fluorescent dyes. So here we used, again, isoproterenol, and we confirmed the chronotropic as well as inotropic effects that we observed with the impedance-based platform. And when we look at mexiletine, nifedipine, and tofetilide, here we observed a concentration-dependent decrease in the amplitude with mexiletine, and a concentration and time-dependent decrease in the PVD50 with nifedipine, and tofetilide reduced the falling slope in a concentration and time-dependent manner and showed quiescence as at higher concentrations. And again, we looked at the set of compounds inducing structural toxicity. Here we have chosen for doxorubicin, ponatinib, and tapsigargin. And all those 3 compounds decreased the beat rate as well as the amplitude, which is not shown and all of those effects are leading to cardiac dysfunction. So, so as a summary of the data we have shown you, we conclude that our large-scale manufacturing process yields highly pure as well as functional iPSC-derived cardiomyocytes. Furthermore, we showed the expected and reproducible responses to a small set of cardiotoxic drugs from different classes using different assays. And based on that, we conclude that our inside cardiomyocytes represent a predictive in vitro model to facilitate drug safety testings. So now we have introduced you to two of our assays as well as one of our products in a nutshell. So if you are interested to learn more about our products or services, please send us a message and we are happy to help you further. So in case you decide to sign a contract with us, so sign a service, you can place your initial inquiry through our website, or you can contact your local business development representative, and in return they will contact you to and bring you in contact with a larger team of different departments to better understand your request and formulate your expectations. So once we have a better understanding of what you would like to achieve, we will assign a project lead, and that project lead will draft a proposal which contains the timelines as well as deliverables. And once both parties agree on the settings, the project will move to the execution phase. So once the project has been executed, you will receive a final report from us, as well as a TC will be arranged to discuss the details or the results more in detail. And after a while, we will contact you again to hear feedback, because for us feedback is very important. So I thank you all for listening to that presentation. So if you have any questions, please feel free to reach out to us.
Thank you so much, Jessica, for this nice overview.
Thank you.
I think we have just time for one tiny question with a quick answer, please.
Mm-hmm.
So Mark Rogers is asking, is the resting membrane potential recorded by traditional current clamp, or were you using dynamic clamp injection with IK1?
These I would need to ask my colleagues, because he performed these. But we can come back to that question.
Okay.
We can answer that in the chat.
All right. Thank you very much.
Thank you.
So before we get continued with Gary Mirams, just one quick info here. We have some polls going on. They will stay for a little while, and it would be great if you participate in these polls. All right, so now let us switch gears and welcome Gary Mirams. So Gary is an applied mathematician at the University of Nottingham Center for Mathematical Medicine and Biology. He works in cardiac electrophysiology and uses mathematical models to understand how drugs interact with multiple ion channels and the consequences of this. And Gary is working with a number of pharmaceutical companies and the FDA to embed modeling in their safety assessment, and develops free open source software to perform simulations. So check out his website for the open source software, just as an info here. His team is currently funded by the Wellcome Trust via a senior research fellowship. And today he will tell us what he found out about examining temperature dependence of hERG gating. I'm very much looking forward to your talk, Gary.
Okay, thanks very much. Can you hear me okay?
Yes, we can hear you, we can see you.
Great. So I'll start with the acknowledgements slide, actually, because most of the work that I'm going to show you has been done by Chonlok Lee, who's now a research fellow at the University of Macau, but did a lot of this work when he was doing a PhD with me and Oxford University. And also a lot of thanks to the other people on this page, particularly the ones in bold that have contributed directly to what you're going to hear about. So how do you describe ion current kinetics? Now, Adam's already given you a nice example of how some models have been used. So you'll all be familiar with things that look like this. An IV curve, an activation curve in this case. So this is from the original Hodgkin and Huxley paper. What you might not be as familiar with is the kind of mathematical modeling of these things. So that might sound a bit controversial, but actually this was from the exact next page in the journal. So this should be almost as conventional as looking at these kind of curves. And I highly recommend actually this, the 1952 Hodgkin and Huxley paper has been updated for modern conventions in terms of direction of currents and voltage and resting potential. So there's a link there, and I'll publish all these slides so that people can download them from my website as well. But that's worth a revisit, especially if you've got students and or colleagues wanting an introduction without wanting to confuse them about how conventions have changed. So what is a model? Well, any simplification of reality. So a lot of people here will work with experimental models, by which I mean cell lines or animal models. You could have statistical models, a regression model. But just thinking about things in words is a model as well. So I might say the membrane voltage changes proportional to all the currents flowing across it, for instance. That would be a word model. Now in physics and engineering, models have got so advanced that we no longer tend to build lots of prototypes and put them in wind tunnels. We'll do all that on computer. So here's a little simulation of a streamlines of flow going over a fighter jet. So are we there in biology? Well, not quite. So what do we want to do? We want to make a mathematical model that specifies all our assumptions about how things work, makes testable predictions, ideally accurate quantitative predictions. A kind of underappreciated aspect is that if you then give someone your model, you've really communicated your knowledge about how something works in a very concise and accurate way. So that might be how a drug affects the hERG channel. It might be how the whole cell creates an action potential. So what's the problem? Well, biological systems are a lot more difficult than physics. So whilst it might look really fancy, that fighter jet simulation is fairly standard and easy to do nowadays, whereas biological systems are still a challenge to simulate. And I really like this quote from James Black, who won the Nobel Prize for beta blockers, I think. And he was quite a good mathematical modeler, which you might not know. And he has this quote in his Nobel Prize lecture: "Models in analytical pharmacology are not meant to be descriptions, pathetic descriptions of nature. They're designed to be accurate descriptions of our pathetic thinking about nature. They're meant to expose assumptions, define expectations, and help us devise new tests." And I really like this because it kind of highlights that we're not going for perfection, but we are kind of thoroughly working through all the consequences of what we think is happening. Now, of course, with things like SEPA, we really are hoping to get close enough to give useful and more accurate than experimental model assessments of pro-arrhythmic risk, for instance. So in a very similar line to what Adam was showing, we've been thinking about using the SynchroPatch for high throughput modeling. What we've done that's a bit different is we've been playing with these high information protocols, we call them. So shorter voltage clamp protocols that ideally contain the same amount of information as running traditional activation protocols, deactivation protocols, recovery from inactivation protocols, etc. So this is the model we've been working with for the hERG channel. It looks a bit complicated. It's actually a relatively simple one. So there's two processes. There's going left to right is the activation and deactivation, and going vertically here is the inactivation and recovery from inactivation. And this is just a Hodgkin-Huxley model when these rates are the same at the top and bottom and the left and right, the K3 and K4. So in terms of complexity of the maths, there's 8 parameters there describing P1 to P8, describing the rates. So what we first did was used a fairly complicated sum of sine waves, Journal of Physiology 2018. And well, thanks to collaborating with Adam and co, we stuck on this traditional activation protocol, which gives you a nice familiar tail current there that we can use to help identify the conductance. And then here we just plot the occupancy of these 4 states in the model over time once we've fitted a mathematical model. So to get this to work on the synchro patch, I'll put this in my nanion feature requests. You can't actually clamp to any kind of arbitrary curved waveform at the moment, like a sine wave. You have to use ramps and steps. So we kind of converted this kind of principle to a series of steps, which we call the staircase protocol for this one. And there's a couple of papers on how we've applied this. So this will run on the synchro patch now, and obviously the nice thing about that is scaling up from being able to get good recordings from about 9 or 10 cells in 2018. We can now do that for 100 in an afternoon, which is quite an improvement. So what we do looks like this. We run our calibration protocol, the staircase, and that's what we'll use to fit the model and those 8 parameters. And the idea is that that will completely capture how we think the gating of the herd channel works. And then we've run some other protocols just before it, which we're going to use as validation. So we'll see if we can predict the results of these traditional protocols, as well as some sort of action potential like clamps, actually comprised of ramps and steps. And we did this at 25 degrees C to start with in a temperature controlled synchro patch, up to 37 and a few intermediate temperatures. Now I'm not sure if the video will work. Let's see. So what you should be seeing is the orange line, which is a simulation, jumping around as an optimization algorithm tries to find parameters P1 to P8 that fit this data nicely. And so this is just to give you a flavor of how the actual fitting process works. So we've actually using simulated data here, which is the blue line with a bit of noise added. We then kind of give the computer an initial guess and use an optimization algorithm which runs loads of different simulations with different parameter sets, hones in on the one that gives the best match to the data trace, which in this case really is as simple as kind of minimizing square error. Is the models of ion channels is using the whole time trace rather than just IV curves and time constants. So that's who that. The way we've ended up working is to do the whole set of recordings, then repeat them all in the presence of E4031, which is a fairly specific and strong hERG blocker. So we record the currents. We've done our own leak just to kind of have complete control over the process. We have a little ramp at the beginning, which we use to fit a leak current. We do the same thing after E4031, and in this cell, it's very tidy. As you can see, you go from currents of about 1,000 picoamps with hERG to about 100 without. So 90% of it was hERG anyway. But we then subtract off the leak on both of those traces, and then again subtract those traces from each other to give a fairly tidy and clean hERG current. And we do this in every cell or every well of the synchro patch independently. Now, very much again along the same lines as Adam, we had to introduce very strict quality control, and perhaps even more strict, because we're fitting directly to that trace, then it really does need to be accurate. There can't be any vertical shift in it or anything like that. So there are more strict quality control criteria which are listed in the paper, and I'll put the references up at the end. But typically the QC1, and looking at the seal resistance, membrane capacitance and series resistance, would be what you'd typically use within the machine and the GUI. We've added in all these extra quality control checks. So this looks really strict and loads of different things, and a lot of those are about checking stability over time, as Adam mentioned, and that things like the estimates of seal resistance are not changing hugely through the experiment. This still left 124 good recordings out of 384 in just one run at room temperature. Those numbers drop a little bit, but are still in the tens of wells at 37 degrees. So here's an example for just a single well of what we recorded. I'll blow up, this was the fit, so we go through the process I showed you in the movie to fit the model to the data. So now we've got our parameters 1 to 8 and our mathematical model, and we use that to predict what we should see in all the rest of the protocols. And so here you see the prediction for this 1 Hertz action potentials, and the red line really is going quite nicely through the blue data. And what we didn't necessarily expect but came out really beautifully was the we zoom in on the initial what corresponds to the upstroke of the action potential. There is a little blip in HERG that kind of grows with use. We call this a kind of spike at the beginning, which is to do with the inactivation, how rapidly that switches off. And so you can see that we actually managed to predict really nicely the size and the time constant of that little spike, as well as the main repolarizing HERG current, which was a nice surprise. And there's similarly good predictions for the rest of the protocols. So we'll get onto the temperature then. So there's a few equations on this slide for which I apologize, but you kind of need them to get into the details of the temperature dependence. So the rates we were fitting looked like one parameter, exponential, another parameter, and voltage. There's no temperature dependence in there. A common way to put temperature dependence in is Q10s, which probably everybody's heard about. So here you'd have this factor, a Q10, and you'd put your modified temperature in here, T, use your reference temperature that, and divide by 10, which is why it's called a Q10. So what you can see by looking at the top and the bottom equation is that the BV term looks just the same, but the A has now got this Q10 and temperature dependence multiplied by an effective A parameter. So what we can see there is that the A is got some temperature dependence, but the B hasn't. What wasn't immediately obvious to me is that this implies that in Hodgkin-Huxley models, there's a temperature dependent scaling on the time constant, but unchanged activation curves and inactivation curves. Now there's a different way of putting in the temperature dependence, which is kind of going to the first principles energy barriers from a kind of physics model of when the protein shifts conformation, it jumps over an energy barrier. And this is sort of entropy and enthalpy, and temperature comes in different places here. So now you can see that there's temperature out the front, but it's just a multiplying factor instead of a power. There's a 1 over temperature here, and there's a 1 over temperature next to the voltage in the exponential now. We actually looked at a more generalized form of this, which is less first principles and more engineering fit, to just have an extra plus a constant times voltage that doesn't depend on temperature. Now, what you expect to see as temperature changes, so this is a plot of 1 over temperature against log of the A parameter over temperature, which gives you some nice straight lines, which is why we use these transforms. And then the same for this is just the B parameter, or the magnitude of it, against 1 over temperature. So what you see in the top plot there is that actually over the highlighted range is room temperature to physiological. You won't really be able to tell the difference between a Q10, a typical airing, or a generalized airing relationship. And we expect them all to follow this decreasing trend. Well, increasing when it's not 1 over temperature. But for the B parameter, there's quite a lot of differences. As we said, the Q10 says the B parameter won't change with temperature. The usual typical airing says there'll be a straight line and it has to go through the origin of this plot, which would be off the bottom left. And the generalized could be any straight line on this plot. The idea of what we did was just to repeat the fitting of these A and B parameters at different temperatures on the synchro patch, and just see which of these patterns they followed. So here we see the fitted models again, this time at 25 up to 37 degrees. And what was quite nice and something of a happy accident was that this protocol that we designed for room temperature actually worked really nicely for 37 degrees. And whereas the activation is so slow that you only saw currents at the end of this protocol at room temperature, they start to happen and you get some quite good strong currents in the first half of the protocol. And again, when we zoom in on small aspects of the protocol, we get quite nice predictions of even the small spikes. And there are sections of this where the Q10 just really can't fit all of these temperatures at the same time, whereas the generalized airing can, and the independent fits the different temperatures also can. So when we plot these parameters against temperature, what we see is that some of them have really strong temperature dependence, much higher than the usual Q10 of 2 to 3, which is in grey. Some of them have lower temperature dependence than the usual Q10 scaling. And a lot of the B parameters where the Q10 says they won't change with temperature actually have some strong trends. And those trends can be positive or they can be negative. So that told us that the typical airing didn't work particularly well either, and we did need the generalized airing relationships. So the punchline really was, if you want all the details of ion channel kinetics to get nice predictions, ideally refit your model at the temperature you're interested in with the exact temperatures data. If you haven't got you can't do that for any reason, then probably use this generalized airing and be a bit wary of using Q10s. So we can then translate back to something you might be more familiar with in terms of time constant and IV curves for activation and inactivation. And what we saw was there was some shift in the activation curve with temperature, as well as the kind of vertical scaling of time constants that you would expect with Q10s. So that's kind of the temperature results. I'll tell you a little bit, if I've got a tiny bit of time, about how we looked at the variability in the data and started to examine reasons for that. So we've come up with something we call the reversal ramp, which I'm hoping is quite widely useful for people. So at the end of our staircase, we've got a big activation step for Herg. So we expect a big tail current afterwards. And during that tail current, we ramp the voltage from up here, where you're expecting the Herg current to be positive, to down to minus 100, where you're expecting it to be negative with kind of usual concentrations. And so what happens is that this big tail current, now the first half of it's positive and the second half of it is negative. And so that crosses zero, and you can have a look at exactly what voltage that happens at by just plotting current against voltage during this step. And so here you see that it's crossing zero at about -86 millivolts in the top protocol. So you're basically just drawing a line up for where this crosses zero into the top protocol to read off the voltage. Now we do this for all the wells in the that pass the quality control. And we get a histogram of these voltages of the crossing to zero. So this is the kind of reversal potential. Now there's an expected reversal potential, which you just calculate using the Nernst potential with a simple formula based on the intracellular and extracellular concentrations and the temperature. And that would have expected us to reverse at this orange dashed value. So what's nice is that these are all distributed around here. But perhaps there is a 5 millivolt shift there, which is difficult to explain. If the current really is reversing at -82 millivolts, that's kind of difficult to explain. But it's sort of easy to explain if you assume that actually the machine was just had a few millivolts of offset in the imposed voltage. So the ramp hinted this reversal ramp. I think it probably is quite a useful thing to include quality control as well, just generally this reversal ramp. But what it hinted at is that there's slightly different patch artifacts in each well, and perhaps those are leading to the apparent variability in the traces and in the inferred parameters we have in our mathematical model. So what we've done is actually built a model of the artifacts as well, which is I appreciate for a lot of you that's really going through the rabbit hole a bit of mathematical modeling. But the usual assumption is that you've got this kind of idealized voltage clamp, and the current it reports really is the current going through the ion channels of interest. Whereas all the manual patch people will realize when they're twiddling all the dials on the front to offset the series resistance, the pipette capacitance, leak currents, etc., you really have a circuit that looks more like this. And that introduces quite a lot of artifacts along the way. So what we did was built a mathematical model of that, and then said, well, perhaps actually the herd currents kinetics are all identical. The conductance might vary, the expression level might vary cell to cell, but the kinetics actually should all be identical. And then we'll try fitting a theory that some small experimental artifacts explain the differences in the data. And when we did this, we inferred that the voltage offset might be might be about, plus or minus 2 or 3 millivolts. There might be a little bit of residual leak current that we didn't perfectly subtract. And with just those assumptions, we can fit the data just as well as saying the kinetics are cell specific, and we can make as good, if not better, predictions. So by just having these 2 parameters that vary well to well, instead of all 8 in fitting the Herg model, you get just as good predictions. And so this kind of makes it more plausible for us, really, that this is a nice way to summarize data from many different wells, get a good consensus of your ion channel kinetics, whilst kind of removing some of the small artifacts and voltage shifts. So that's it from me. If you're interested in any of that, you can contact me on my webpage or Twitter. I'll tweet out a link to these slides shortly and put them on my webpage. And these are the 3 papers that we, most of that work featured in, that we just published last year. And again, thanks to Chon, who's the first author on all of these. So he deserves most of the credit. Okay, thanks.
Thank you so much, Gary. It was a really interesting talk, very nice data that you showed. So we have time for one question. And what do we have here? What is the biggest hurdle to embed modeling into safety assessments, in your view? What would you say?
That's a good question.
Yeah.
I think it has probably been kind of working out how to deal with the data and what the best ways to do that. So we've done quite a lot with GSK, where we've tried to embed simulations, in-house for them, using their screening databases, so IC50s and things. And the most tricky thing really was coming up with a quality control pipeline for their IC50 data. If the hill slope's too low, maybe it was run down in the experiment rather than real drug effects. There's variability in those IC50s, even when you do trust the hill coefficient is good. So it was building that pipeline of when do we use the data, when do we think it's good data, and then we know there is some variability in that data. How do we map it through the model predictions? But it's a great question that I think we'll probably get onto a bit more in the roundtable discussion.
Yeah, right. Okay. Yeah, thank you so much, Gary.
Okay.
So next in line is Teun de Boer. He is an associate professor in the Department of Medical Physiology at University of Utrecht. And Teun obtained his PhD in physiology at the Utrecht University and continued with his postdoc positions, one at Utrecht and the other one at Oxford. Teun then returned to Utrecht and during the last 12 years has been offering valuable insights into cardiac physiology and safety. And today he will talk about a special interesting topic, which is dynamic clamping and how this technology helps build hybrid biological digital twins of cardiomyocytes. So I can see Teun, he's here. Teun, if you like, the virtual stage is yours now.
Okay, very good. Then I will try to share my screen. Or can you already see my screen? Just checking.
No, not yet. We can see you and we can hear you nicely, but we cannot see your screen.
Okay.
No. Yeah. Yes, exactly. We can see your keynotes. Yes, perfect. This looks good. Enjoy your talk. Thank you.
Okay. Yes. Well, thanks, Sonja, for the introduction. And also thanks for the invitation. Happy to talk here about our work on dynamic clamping. So today I would like to share with you our work on dynamic clamping, as I said. And I think it's a useful tool that you can use to build hybrid biological digital twins of cardiomyocytes. And during my talk, I will explain to you what I mean. But I think you could see this as effectively merging computational simulations with patch clamp experiments. And that way, I think we can solve some of the problems with computational models, like the fact that you need to have a model in the first place, and it has to be good, which is not always very easy. So I won't be saying dynamic clamping is easy either, but we're getting towards automated dynamic clamping. I think that makes quite a difference. Right, so to start, as you probably all know, cardiac excitability is an emergent property. So the behavior of the heart, the action potentials you see, emerges from the presence of components that interact, very much like these bees that you can see here. So governed by a few very simple rules, like when your neighbor moves, you should also move. You can see the emergence of these beautiful patterns that are also very similar to arrhythmic impulse propagation in the heart. So this is a nice example of how complexity can generate the behavior in the heart. So if you look at an individual cardiomyocyte, in this case, a cell in the sinus node, the action potentials that are being generated on the left are being produced by ion channels that are all coupled into the same plasma membrane and therefore all experience the same membrane potential. And by opening and closing, each channel also has an effect on this membrane potential. So they're all coupled together. And through this coupling, the complex behavior emerges, which is the sinus-nodal atrial action potential. So if you want to look at the effect of a drug or a mutation on an ion channel and on the action potentials, you have to look at the cellular electrophysiology. And for that, we all use patch clamping. So this is the typical pattern, manual patch clamping, and a picture that you also just saw with Gary, a representation of the way recording works, with a pipette making electrical contact with the interior of the cell, and then you can measure the membrane potential or currents flowing through the membrane. So this is then done in two modes, which most of you probably are familiar with. So either you do voltage clamping, where you apply a voltage of your choice and record the current flowing through the ion channels in the membrane, or the other way around, you use current clamping. You apply a current and record the membrane potential in response. And these two different modes can also be used in different dynamic clamping setups. So dynamic clamping is a technique where you have a real-time interaction between a computer model of a biological system and the biological system itself. So in the case of cardiomyocytes, you would have a real-time interaction between the cardiomyocyte and, for instance, a simulation of an ion channel. Or the other way around, you can have an interaction between a simulated cardiomyocyte and a real ion channel that's being expressed in a cell line and being recorded on a patch clamping device or a manual patch clamping setup. So in the first, in the upper example that I show here in this graph, we look at stem cell-derived cardiomyocytes. So those are not as mature as you would like them to be. And one way, that was already discussed in the earlier talk by Jessica, one way is to add IK1 via dynamic clamping. So in that case, you record and current clamp the membrane potential of the stem cell. And this membrane potential is, it's measured using the patch clamp amplifier, and the voltage is being sent to a computer that's running a real-time model of the IK1 channel. So it can compute how much current should flow at a specific voltage, at a specific time, and that current is then sent back to the amplifier, and the amplifier inserts the current and injects the current into the membrane potential of the cell, so into the cell, and thereby changing also the membrane potential. So this is a closed loop, and this loop is running very quickly. So every time this loop is being done is in about 50 microseconds. And that way, for the cell, it seems almost as if these simulated IK1 channels are really part of the cell. So that's one application, and I'll show some examples of that first. Later on in my talk, I will also show an example of doing this with voltage clamping. So in that case, you would have a real-time computer simulating a heart cell, using, for instance, the Tentution model or the SIPA action potential models. This model produces an action potential, so a membrane potential. And this membrane potential is fed into the patch clamp amplifier, which imposes it on a cell line, on a cell, for instance, overexpressing hERG channels, which produces a current in response. And that current is being measured with the amplifier and subsequently sent to the real-time computer, where this current will be added to all the other currents that are being stimulated and is used to compute the membrane potential. And in this way, the ion channels in the real cell are contributing to the action potential shape of the simulated cell. So these are two ways to construct a hybrid model where you compose the model with a biological part and a computational part. So why is this relevant? Well, in my opinion, there might be other reasons, but there are two important reasons. First, I think we need better models for safety pharmacology. And also there is an increasing need to explain what a mutation does. So people are diagnosed with mutations in genes, and they have a disease, but with unknown mutations, it's often unclear whether the mutation is the cause of the disease or maybe something else that has not been discovered yet. And in both aspects, iPS-derived cardiomyocytes are an important tool, but I think they're not the final answer. And that has to do with the immaturity of these cells. So as you can see here on the left, there's a cartoon of an adult ventricular action potential. And I would say that the ventricular action potential is most relevant. Right? So the largest mass of your heart is ventricular cardiomyocytes. So that's what you should use in a model, also if you want to predict ventricular arrhythmias. And on the right is a cartoon of iPS cardiomyocyte action potential. Well, the differences are large, and the iPS cardiomyocyte, granted there are better looking examples, obviously, as also been shown before, but this is a bit exaggerated. But what we know is that if you add IK1, then the membrane potential, the action potential of an iPS cell will go towards the adult type. So this is a simulation where we've used a model of an iPS cardiomyocyte and added more and more IK1. And what you see is that the cell starts to hyperpolarize, the action potential shape becomes more adult, and for instance, the upstroke velocity gets faster, and at a certain point, the cell is no longer spontaneously beating, which is an unwanted behavior for a ventricular cell. And this also has an effect on, well, not only on how you can test drugs, but also on whether or not you can assess the effect of a mutation very well. And you can see that here. So this is a further part of the same simulation I just showed you. But then looking at the various ion currents. So as I told you, we were increasing the amount of IK1. So you can see that the IK1 current is getting bigger and bigger with an increasing GK1. But also the other ion currents are affected. So for instance, if you look at hERG, in the immature state, the hERG channel is active throughout the whole action potential. And in the adult one, you can see that there are, well, there's less activity. And similarly, the sodium channel contribution is pretty small in the, well, with lack of IK1, and it increases when you add IK1. So if you can add IK1 to stem cell-derived cardiomyocytes, you have a tool to increase the predictive value of the model. Well, and you can do that on a dynamic clamping setup. That's also what we do. This is one of our setups. And if you have a good day, you can maybe do 10 recordings in one day, but that's about it. So that's not very high throughput. But you can also now do this together with the patch liner system. So do automated patch clamping in combination with dynamic clamping. And that obviously increases the throughput quite a bit. It also has another benefit that, so we developed this system together with Nanoion, and they've now brought it on the market. So there's already a couple of labs that have it in their, well, next to their patch liner. And one of the things that we've incorporated now is the seal resistance compensation. So if you have a leak in a voltage clamp experiment, you can do tricks afterwards to correct for it. Gary Mirams also just explained some of the tricks that are available, and that can make a really big difference in the quality of the data. The problem with current clamping, however, is that you cannot do that after the fact, because if the cell is depolarized due to a leak through the seal resistance, then the membrane potential will be different and it will affect the state of all the ion channels, and it cannot be corrected for afterwards. So the correction has to be done during the experiment. And to show how this works, so you have here a cell sitting on a patch liner chip. The cell produces a membrane potential, Vm, and that's the difference between the potential on the outside of the cell and the interior. Well, on the outside, voltage is 0, and this voltage, it can dissipate via the membrane, right? So that's a resistance, but there's also the seal resistance. And coupled to that, you have the amplifier and current clamp. So it's not contributing any current. So the current is either going out via the membrane or the seal. And by calculating this current that's flowing through the seal, you can also add it back. So compensate for this leak by actively injecting current that matches the voltage at each time point during the experiment and the seal resistance. And it helps a lot to get better action potentials, both in manual patch clamping experiments, as was already shown quite a while ago by David Cristini's group, but also in these automated patch clamping experiments. So the system we designed runs on LabVIEW. It's a LabVIEW real-time system. And what it does, it couples with the patch clamp amplifier used with the patch liner. And it can, this is from an older publication where we show that we can do 4 channels at the same time, but the current system does 8 channels. So all the 8 channels of the patch liner are independently coupled with, to a dynamic clamping model. And if you are recording from 8 cells, you can add IK1 to each of these 8 cells independently. And the whole system is controlled via patch control and patch master. So the whole dynamic clamping procedure can be fully automated and be run unsupervised. And it works. So if we add IK1 to IPS cells, we can get nice action potentials. This was already shown earlier for manual patch clamping, but so it also works in this setting. And as you can see here, adding BK induces prolongation of the action potential, and nifedipine shortens the action potential, as you would expect. This has also, in the meantime, been done in another group. So this is the group of Kramer. I'm not sure if I pronounced her name correctly. My apologies. In Dresden, they've also used a prototype of the dynamite system and also were able to record action potentials in combination with the patch liner that have a nice stable resting membrane potential and action potential shape. So then, obviously, the question is, how much IK1 should you inject? And we, in the first study, we had a quick look at that. And what we saw is that if you start to inject IK1, so here the IK1 conductance is increasing, you can see that the action potentials, which are at first a bit longer when compared to injecting a constant hyperpolarizing current, they quickly get shorter. So it has quite an effect on the action potential duration. Also, the upstroke velocity is also affected a bit, but less so. So how to look at this? Because it's an important aspect, because in an automated experiment, which is not supervised, there's no one, well, looking whether or not this is going well, and it has to be done during the experiment. So we decided to do a modeling study to look at different models of IK1, so mathematical description of the current and how this affects the automaticity of IPS cells. So we've compared all the published IK1 formulations that are present in a number of models that you see here. And we've plugged them into a simulation of IPS cells and then tested all different combinations. And one of the things that you can see in this figure is that these different IK1 models, they have quite a different effect on the action potential shape. So the first impression is the models make quite a difference. And that was also the conclusion of the paper. And during this work, we also observed that there's a critical value. And this is a critical value that's the minimum amount of IK1, or the minimum conductance that you need to hyperpolarize the cell and make it stable. So you have a stable resting membrane potential and no spontaneous action potentials. And this critical GK1 that we defined, we also tested it in a whole number of simulations. So these are all different tweaked versions of the IPS model. They're all slightly different, and then we applied IK1 from these various IK1 models to test what this critical value is. And what you can see in this figure is that if you look at the different cells on the horizontal axis, there's some variation between cells, but the largest variation actually is between the models. And this is also in line with experiments that we did in the same study. So what we tested is, can we also look at this critical GK1 in a real experiment? So here you see the membrane potential of an iPS cell that was current clamped, and IK1 was injected. And the amount of IK1 that we injected was gradually decreased. And you can see that you go from the upper left from a very stable membrane potential to, well, it's slightly depolarizing, it's getting less stable, and even more less stable. And at a certain point, spontaneous activity ensues. And if you completely stop adding IK1, then you get the spontaneous beating behavior. And if you look at the cells that we tried, you can see that this critical value is actually quite variable between cells. And I think that's an important thing, because that means that during an automated experiment, you have to address this and take this critical value into account. And in our study, in the simulation part, we also use this to describe the effects on action potential, duration, etc. So I think this is an important start in getting this really very well unsupervised. So are we there yet? No, so far we have enabled higher throughput automated dynamic clamping and identified the best IK1 model and say, but to our opinion, it's the model by Ishihara, and that's also the one that's actually being used in the Dynamite 8 system. And we established this new parameter, critical GK1, that I think will be very important for automated dynamic clamping. But can we do more? Yes. So I mentioned to you also the hybrid model that uses voltage clamping. And that's actually already known. It's not an invention of ours. It was already described quite a while ago by the group of Ronald Wilders, the first author, and they described it as dynamic action potential clamping. So the action potential that's being used to clamp the cell that expresses the ion channel, so IKR in this channel, it's dynamic. It's not static action potential clamping, because the current that's being produced here actually affects the shape of the action potential. So, and I think we came up with the name biological digital twin, because digital twins is a bit of a hype at the moment. So if you want to model the heart cell, you can now include this biology into the simulation. So the way we see it is you have a model of a human cardiomyocyte. Let's say we're interested in HER currents. We remove the HER current from the model and measure HER currents from a cell that overexpresses it. And the HER channels here may be mutated. You could add a drug that happens during the experiment. And the two parts are merged via the dynamic clamping technique. And what you effectively end up with is a biological digital twin of the heart cell that incorporates biological HER current. And what I think is the important part is that there is no need for a mathematical description of this HER current. And there's also another need to describe before the experiment how this drug might interact with this channel. And I think that's a very important benefit. We can just sort of unbiased see what happens. And you can also imagine that when you have a new mutation, you want to test drugs, then you both don't know what the mutation will do, but you also don't know if this will have an effect on the drug channel interaction. Okay, so we have some first results here. So this is an example where we, using manual patch clamping, coupled the Teun model. So that's one specific action potential model for the left ventricle. We coupled it with a cell overexpressing her current. And the current you see here is actively contributing at each of these time points to the action potential above. So there's a constant interaction. We didn't add any drugs in this experiment, unfortunately, but we did do this with IK1 a bit more extensively. So what you see here is, again, a manual patch clamping experiment. And we had, again, the Teun model, and then coupled with a HEC cell that overexpresses IK1 channels. And so here you see the action potential. And this is the current that we record. So this is going straight from the amplifier. And you can see that there is this peak here. So this is the most relevant part of the IK1 current. But you can also see the other part here. And that has to do with endogenous channels in the HEC cells. So I won't go into the details here, but you can, we've characterized cells that, HEC cells that do not express any IK1 or other channels, and we can quite well predict this endogenous current here and subtract it from the signal. And well, we tested what happens if a barium block happens. So you add barium, and then you can see that this peak here is disappearing, and also the action potential is getting shorter. And you can also see that the stability of the membrane potential here is getting less. And we've also overlaid some of these conclusions. So what you see here is the blue line, that's the model itself, so the action potential that the computational model produces. The red line here is when we couple the biological IK1 into the model. And you can see that the cell is depolarized a little bit. And we explain that now because we think that the scaling was not perfect here. So obviously, the amount of IK1 that the cell that you're patching produces is not the same as what's in the computational model. So there's a scaling step to match the two. And the matching wasn't perfect. But when we add a little bit of barium, you can see that the membrane potential becomes unstable already. And the action potential becomes a bit longer. And when we add more barium, well, essentially you end up with sort of a sinus node-like thing with spontaneous depolarization, slow upstrokes, and an altered action potential shape. So I think this shows that indeed we can couple or use this dynamic action potential clamping technique and also observe drug effects. So to conclude, automated dynamic clamping is possible. You can do it with the dynamite system. And the way it's available now, you can readily add IK1 to iPS cardiomyocytes. Currently, we are working on implementing this dynamic action potential approach on the Dynamite 8 system as well, to reproduce the things I've shown from manual experiments also with the patch liner. And I think that by using dynamic action potential clamping, we can, well, it can advance the study of drug and mutation effects, because we do not need a mathematical description of these effects a priori. Okay, and the good news is also that this work, well, we've applied for funding together with NENI and Elements in Italy to work further on this dynamic clamping topic, and it seems like we are being funded. So probably there will be a postdoc position from this fall for 2 years to work on well, further advancing dynamic clamping in my lab. So if you know somebody who could be interested in this, has the computational skills and experimental skills, or you are interested yourself, please do contact me. There's not an official job opening due to admin reasons, but it will come shortly. Okay, thanks for your attention, and also thanks to these people, especially the ones highlighted in yellow, that have contributed to the experiments I have shown. Happy to answer any questions.
Thank you so much, Teun. Very nice talk, very interesting. So we have time for one question. Ron Knox, by applying digital dynamic conductances in the right amount and right time, get a hex cell to fire action potentials. Could you do that?
Well, yes, I think that's exactly, in fact, that's exactly what we're doing with this dynamic action potential clamping. So you could say you're adding the current from the hex cell to a simulated cardiomyocyte, but you could also exactly say the opposite. We're adding all the other ion channels to this hex cell so that the whole ensemble is firing action potential, so also the hex cell. But I'm not entirely sure, but that's answer to the question.
Yeah, I think that was the answer. All right. So thank you very much. We will continue now with a little networking session. We will truncate this by a couple of minutes. So we will come back together with Jim Kramer's talk at 3:05. All right, and Jason will give a little introduction to the networking again.
Hi, Sonja. I'm just going to go into presenter mode now. So for those of you that joined us yesterday, just a little recap about the networking session that we have. So on the left-hand side of your screen, you should see a networking tab where you'll be able to participate in an interactive mixer where Hopin will match you with a fellow meeting participant. So meet someone new or rekindle a familiar face. By default, each meeting is set to a specific interval. Feel free to extend that talk as desired, and then the platform will connect you automatically to the next person. So we'll reconvene here. We have a little delay in the program, but we reconvene at 3:05 for the presentation of Jim Kramer.
Welcome back, everyone. Our last session is ready to roll now. And I hope you enjoyed the networking event and met some nice people there. All right, so our first speaker in session 4 is Dr. Jim Kramer. So Jim started his education at Xavier University and got his PhD in biomedical sciences at Wright State University. Since then, he has been a principal scientist at Gentest in Cleveland for 16 years, specializing in cardiac safety drug testing. Since 2014, he's a principal scientist at Charles River Laboratories, and he is also in the leadership team of the CEPA Ion Channel working group. And today he will share his expertise and focus on a very important topic relevant for everyone in the cardiac safety field. He will speak about the ICH-S7B Q&As, the best practices for ion channel assays. So Jim, welcome.
All right. Yeah, thank you. And thanks for inviting me to talk. It's since COVID, it's been a long time since I've done one of these. And my daughter asked me this morning who I was going to give the talk to. And I said, well, scientists from all over the world. And she said, good luck. So we'll see how this goes. Not a lot of biophysics in this talk, going to talk about the rules essentially. So hopefully this is interesting. So just an introduction, the S7B guidelines were at least the most recent version was finalized in October of 2005. So that's been a long time ago. And for those of you on the call that don't know what these are a set of guidelines. And the main objective is to identify potential test substances that may delay ventricular repolarization. And this flowchart over here really outlines this procedure, how you go about determining the risk of this happening. So the first, it really is composed of two assays. The first two are the in vivo QT assay that's usually done in dog or monkey. And then the second main assay of the strategy is to test IKR, which all of you probably know is the HERG assay. And there's information from the pharmacological class that goes into determining how the assays are, this is really the initial sense of risk by looking at the class. But the data from these two assays will be fed down into this box here, which is the integrated risk assessment. So this is where we'll look at the results from the QT assay, we'll look at the results from the HERG assay, and companies will consider any other information from non-clinical or clinical sources. And also this little box over here, it's relatively small, but this is where a lot of work can be done. This is where other ion channel assays could be performed, as well as other assays like the Langendorf or Purkinje fiber. And this can all be fed into this box. You'll integrate all the data, and then out comes an evidence of risk. And so what I did, and so what I'll do before I go over the new best practices is to look at the old best practices. So I went through the S7B document. I will point out there really isn't a section called best practices for ion channel assays in that document, but there are certain points that were mentioned. And the first one is something very simple. It's just constructing a concentration response curve for whatever ion channel you're testing. And the suggestion was that the concentrations should cover and exceed the anticipated Cmax for that compound. Of course, we all know now that the HERG assay is performed usually very early, usually before these Cmax values are known. So the main objective of our assays is to do a very nice dose response curve so that we can calculate an IC50 or an IC20 or an IC25. The third best practice that they pointed out was talking about the duration of exposure of the test substance. So you can see this little experiment over here. This was performed, oh my gosh, this was performed many, many years ago in our laboratory. It's a manual patch clamp experiment. This is the vehicle period. We're looking at the peak tail currents, and you can see 50 nanomolar terfenadine was applied to the cell, and it takes an awful long time. I mean, this is a manual patch clamp study with continuous flow, and it still took 9 minutes to reach steady state. And of course, in the guidelines, they state that you should, your exposure period should last until steady state is achieved. In the experiments that you perform, you should use a positive control that will establish the sensitivity of the assay, and they suggest that a submaximal concentration, just a single submaximal concentration of a positive control should be used to determine this, and in this case it's terfenadine. The assay methodology and experimental endpoints should be scientifically valid and robust. So what we do in our lab and what we've done for years is before any assay goes live, we validate the assay to make sure that it's working as expected, and usually, and the word robust is completely apt here. We want to make sure that the assay works the same every single time. The preparations and the assay conditions and protocols are standardized, and the results have to be reproducible. And the endpoints of the assays have to be relevant for assessing human risk. So for example, if you look at a HERG experiment, you're measuring the tail currents because that is the most relevant parameter to measure in a HERG assay. They do point out in the guidelines that there could be solubility issues, and this is really an understatement. Almost all of the test articles that we have evaluated here since I mean, I've been doing this here since 1999. Just about all of them have limited solubility because they're very extremely lipophilic and hate our buffers, our aqueous buffers. So you have to take that into account. The way we do it here is we just perform a normal solubility assay. The guidelines also point out that it could be adsorption to the glass or plastic or some kind of non-specific binding. And this is, I'll point this out, this is most often the case. You have to be careful with this. So of course, what this implies is that you should be measuring the concentration of the test article to make sure that your nominal and actual concentrations are the same. And they also point out that sometimes the test substance can ruin your membrane integrity. And of course, this can happen, especially with these. I've tested many drugs that have surfactant-like qualities, and it just ruins the seal between the recording electrode and the membrane, the cell membrane. And so this brings us to the latest Q&As, and this is from August 27th of 2020. And as you can see here, there's actually a section that says best practice considerations for in vitro studies, which begs the question, why? Why do we need better best practices for ion channel assays? And so in the next few slides, I hope I prove that maybe we do need better practices. And so the first thing that I'll point out is that this non-clinical ion channel data is now expected to inform clinical decisions. And this, what I state here on the slide, is an example of this. So a double negative, and what's a double negative? A double negative is a compound that is HERG negative, doesn't block HERG current, and is also QT negative. So in an in vivo QT assay, there's no prolongation of the QT interval. So what's proposed is this information can actually streamline the drug development process, and you can use this data as a substitute for a thorough QT study, and it will inform the labeling. As I pointed out, it's going to help with all kinds of regulatory decisions at the time of the market application. Especially, and it's really going to be useful because some drugs you cannot conduct a thorough QT trial for. So now the bar is raised. Okay, so now what has to happen is this data that we acquired for this ion channel, all of our ion channel assays have to be as robust and as good as possible. And you might say, well, hasn't this always been the case? I mean, we're all trained electrophysiologists. We go to school, we learn biophysics, Hodgkin, Huxley, we learn about them and their equations. What's the problem? Well, if I went back into the literature, actually, I just looked at two papers, one from Will Redfern and the other from Glenn Kirsch. These were in 2003 and 2004, pre-S7B. And if you look at the IC50s, I just recorded here in this table the minimum IC50 for each tested article and each drug, and the maximum IC50 that I found in these two papers, and then calculated the ratio between them. And as you can see, for some of the test articles, there's quite a bit of difference, and that's shown here in the table in the figure on the right. So you can see what I plot over here is just a simple column. At the bottom of the column is the minimum IC50, the smallest IC50 that I found in the literature, and this is the largest. And you can see for difetilide, that is 3 log units difference. 3. And it's not just for difetilide, but bepirodil, terfenadine, cisapride. All of these have at least a log unit difference. And so then you say, well, okay, this is pre-S7B, that there weren't, if you look at the studies themselves, people are using different cells. Many of them are using ventricular myocytes to measure IKR. Solutions were different, voltage protocols were different, temperature was different. Okay, fine. Okay, so all of that may have contributed. But then if you look at another study, this is a study from Laurie Hansen. This is one where when we were at Chantest, we actually participated in, and another ion channel company at the time was Zenus. And we tested a number of drugs and recorded our IC50s. And I'm just looking at the differences here in the last column. And most of the compounds were anywhere from the same IC50s, 1-to-1, or up to about a 4-fold difference, which you can see here on the upper right-hand side. This is terfenadine. This was about a 4-fold difference between our IC50 at the time and Zenus's IC50. But still, here's another drug at the end. This is pimozide. There was up to almost a 44-fold difference between our IC50s. Now, this was done. I remember when we were doing this project, this was done as the ICHS7B guidelines were being finalized, and we're both companies well experienced, and we were well experienced electrophysiologists, and we still had a difference between our IC50s especially for pimozide. That's a big difference. And then getting even more post S7B. This was just published last year. This was a study where we looked at the effects so there were 17 sites that volunteered to do the study, and all of them, and we all used our automated platforms to record currents, the effects of 12 drugs on 3 different currents. And I will notice the word here, volunteered. All of us volunteered to do this. And we wanted to characterize the real world results. Again, all of these laboratories that participated were, I consider, experts in their field. These automated platforms have been around for quite a few years. And if you look, so these are IC50s, log IC50 values plotted for each test article on the x-axis for HERG and for peak nav over here. And so you can look at this green circle over here. This is very tight data for chlorpromazine. And but if you look just a little bit to the right, you can see our friend dofetilide here, and you can still see there's a 2-log difference between the lowest IC50 and the highest IC50 obtained. And the same is true over here for peak nav 1.5. So I would suggest just looking at the variability in the data that's occurring site to site. It's not just platform to platform, but inter-platform variability exists, site to site variability exists. It really suggests that we need to standardize our practices. And I will point out in this last study, with the automated platforms, we did have quite a few controls. The voltage protocols were all the same. All the compounds were blinded, and we still saw some differences. So in the new Q&As, these are just a series of best practices that are suggested to tighten up the data. All the voltage protocols are now going to approximate elements of the ventricular action potential. I think all of you people who do patch clamp know that we could actually feed an action potential waveform into an action potential and use it as a waveform for our patching, but we've simplified it. I was actually on the committee that determined these, and these all approximate a bit of a crude manner in a step ramp kind of fashion an action potential. The guidelines suggest that everything should be recorded at an elevated physiological temperature between 35 and 37 degrees C. I know it's harder to record at this temperature. Gary's talk earlier, you could see that the effective temperature on his success rate was pretty profound. And this is what we find here too. But now we've always recorded at near physiological temperature, but it's usually around 33. But for the last year, we've been doing a project where we've been recording at 37. And in the beginning, it was hard. It's still hard, but once you get people trained up, it's not that bad. And why would you do this? Because temperature makes a difference, not on every compound, but some compounds do have temperature dependence, and that's an example of this is over here. Erythromycin is temperature dependent. If you look at the effects of 1 millimolar erythromycin on the tail current, you can see at ambient temperature, it really only blocks about 30% or so. But down here at the lower panel, when you raise the temperature between 33 and 35, you get close to an approximately 80 to 90% block. And there are other compounds that are temperature dependent, so it seems prudent to use physiological temperature. Concentration verification, as I said before, the original guidelines seem to imply that you should measure concentration because of adsorption to your perfusion apparatus. Now it's definitely in the Q&As, and this is a paper by Jessica Brimacombe that came out in 2009, where we looked at the effect of the loss in the perfusion apparatus at low concentrations and high concentrations. All of these were manual patch clamp studies, about 37 of them, and you can see, at least in there's not, the high concentrations, you don't lose a whole lot. Only 2 studies showed a loss of 10 to 19%. But here, low concentrations are the biggest problem when it comes to adsorption. And one of the studies, we lost greater than 40% in the perfusion apparatus. We also looked at stability of the test articles. And when I say stability, it doesn't mean that the test article breaks down. It just means that it comes out of solution. 10% had a loss of test article within 8 hours. 22% were not stable for 24 hours at room temperature, and 16% were not stable under frozen conditions. So again, this is now in the guidelines, in the Q&A. And as I mentioned before, the S7B, the original S7B, just suggested that you do a single submaximal effective concentration for your positive control. That's different now. Now they suggest that you do 2 or more concentrations, ranging from 20 to 80% block. And now, if you are outside the expected range of values for the inhibition, your study is going to be considered inconclusive. And you have to read between the lines here. So now what you have to have is you're going to have historical data in order to see what the expected values should be. And this is also in the guidelines. It's suggested that you demonstrate basic voltage control. This is actually an experiment on the upper left. This is a typical verapamil experiment where you add verapamil to a Herg cell. So in the guidelines, the Q&A, it suggests that you have manageable leak currents. You can easily measure this by looking at the holding current, which is shown here. And you can see this cell was very stable over time, except at the end when we added E4031, there was an increase in leak. Another way to demonstrate voltage control is to measure the series resistance. It's hard to see. I know it's over here, there's an actual hyperpolarizing pulse, really tiny one. And from this, you can, using Ohm's law, that's the only equation that I have in my talk is Ohm's law. You can actually calculate the input resistance, and that's shown and plotted very easily. And you can see here, little jumping around a little bit, very stable though. I think I would consider it fairly stable throughout the experimental period. These plots that I'm showing here, it's suggested that these be in the report. And the last thing that I have listed here is that a saturating concentration of a selective blocker be added. And we do this anyway. We add E4031 at the end of our Herg experiment, just another way of determining the amount of leak that's in your cell. And then here's the data analysis part. This is what they suggest. We've done this for a very long time. We've calculated our rundown and actually corrected for the rundown. And this is just taken from one of our spreadsheets where we do our analysis. I'm sorry, the cell doesn't have a lot of rundown. Herg doesn't have a lot of rundown, but what we do is we can estimate it by looking at the vehicle period and then extrapolating throughout the duration of the experiment. We do this and we correct for it, but now it has to be reported how you're doing it. The other change is what we usually do is we we'll list the individual concentrations in the table, but what we'll do is we'll plot the average inhibition. And now what's suggested is that you actually plot the inhibition for each individual cell with the standard error of the mean, and plot a 95% confidence interval. And of course, you should report your IC50 as well as your Hill coefficient. These are the two variables in the Boltzmann function that we allow to float. The point of doing this is actually to calculate the IC50 error. If you remember back in, this is a figure taken from Will Redfern's paper in 2003, where he was looking at the safety margins for compounds. Remember I showed you that there's a lot of variability in the IC50s in this paper. Well, in this plot, in order to calculate his safety margins, he used the most potent IC50s. But there's no indication of error. And this brings me to the next part of the guidelines, is this, a lot of people use the 30-fold to 100-fold difference between the hERG IC50 and the free plasma concentration as an indicator of risk. So if your hERG IC50 is greater than 30-fold then your free Cmax, then it's considered to be, it may be safe. And that's indicated here. In these compounds in group 1 and 2, you can see the safety margins were most of them were less than or equal to 30-fold, except for amiodarone, which is a little different. And here in group 5, which are considered to be non-torsades drugs, the ratios were more than 30-fold for most of the compounds. That's no longer going to be the case. What's suggested in the Q&As is that your safety margin should be calculated, but you should also, like I said before, calculate the error, and that's going to come from your from the plot that I showed in the previous slide. And now what you're going to do is compare this safety margin with the safety margin of a group of known reference compounds that have known torsadogenic risk. So now what we'll have to do here, and what we are doing now, is we are acquiring a library of reference compounds, and the effects of these reference compounds on our ion channels, and we'll have to calculate the safety margins for these with their error. You may ask, well, what is this reference compound group that you're doing? Well, we're taking the reference compounds from the SIPA. This is a drug list that's on sipaproject.org. These are the SIPA compounds. And the torsadogenic risk was determined by a group, a separate SIPA group that actually I was on. I was kind of the oddball. I was the only ion channel guy on the group, but the rest were composed. The people on the group were mostly MDs that were that assigned torsadogenic risk to these compounds. And so finally, this is just the slide that kind of sums up what the action items are for Charles River right now. So we're preparing that reference data set, not just for hERG, but for CAV, peak NAV, and late NAV. The most important one, obviously, is hERG, because we're going to be calculating safety margins and comparing and using it for comparison. So this is going to be a very important data set. We're doing it for manual, of course, because that's usually what goes into the marketing application. But we're also doing this for our automated patch. We have the NanoIon Synchro patch. We have 2 of those, and we also have a Sofian Q patch. Both of those have temperature control. So I know it's going to be, I don't know how we would measure the concentrations in the wells because of the volume limitations, but it's very small volumes that we add. But people are going to want to know the effects of their compounds under these conditions. So, and early, not at the end of the drug development process. So we're going to be have these assays as validated as possible for the automated. There has to be agreement between our manual and automated patches, but there also has to be agreement across sites. We are not the only ion channel site at Charles River. There's an ion channel site up in Quebec, Laval, Quebec, Canada, and one in Edinburgh. And we can't, I don't see how it's just not allowed for us to have a large amount of variability in our data, because you or whoever comes to us as a customer wants there to be consistency. So we have an actual subgroup that we're forming to address these issues. And there, I should point out in this second to last bullet point that the raw data for these SIPA assays is sent to the FDA for their review. So you can't cut corners. They're going to be checking on you to make sure that your data is following these guidelines. And finally, extra time equals extra cost. I'm a scientist. I'm not a business person, but all of this is taking extra time. The actual study will take extra time to do because of the elevated temperature. And we're going to have, and it's taking extra time to put together these data sets. And, you know, the cost is going to be transferred ultimately to the customer, but because the bar has been raised on the importance of this data, I think it'll be well worth it. And finally, just thank you for allowing me to talk. And if you have any questions, please, I'll answer anything. Thank you. Okay.
Hey, Jim, thank you so much for this very nice presentation. So we got room for a question here. So Stephen Jenkinson is asking, with respect to the temperature and running at 37 degrees, from your experience, does increasing incubation time at lower temperature produce a similar result to that, say, at 37 degrees?
You know, for the example that I showed for erythromycin, I think, yes, it would have eventually reached, at room temperature, it would have eventually reached the level that we achieved at higher temperature. But that would have required, I think, at least, I haven't looked at this data in a long time. I'm sorry, this was many years ago, but it would have taken, I think, almost 30 to 40 minutes of exposure to reach the levels that we saw at higher temperature. And of course, that I know, you know, it's tough to do that, especially in a manual patch clamp. I've had a few that have lasted 30 minutes, but that's not the norm.
Okay. Maybe I also have one last tiny question. Just out of curiosity, have you guys also implemented any in silico modeling already, or this is not the plan, right?
Yeah, no, not yet. We don't. Unfortunately, we just haven't done that. I think I think the other experts are filling that void. You know, Gary Mirams is helping out with that, and as well as some other groups. The FDA, of course, has their own modeling effort. I'm kind of waiting to see how that all, especially with the FDA model, how that all plays out. I know that the kinetic, that at least the FDA model needs some kinetic information of hERG block, this on rate. And that was using the Milne's protocol. It was very difficult. And even for us, it was difficult. We have done some work with that, but I'm just waiting to see if that's modified, and I'll let the experts handle that.
Good. Okay. Thank you very much, Jim.
Thank you.
Okay, so now we can move on with our next speaker. And yeah, I'm very proud to present Dr. Michael Pugsley as our keynote speaker for today. So Michael currently holds the position as a director of toxicology and safety pharmacology at Cytokinetics. He was the former director of toxicology at Purdue Pharma, where he led the PKDM toxicology department in planning and executing all non-clinic tox and safety pharmacology studies. Prior to that, he had developed broad experience from various pharmaceutical companies, including experience preparing IND and NDA submissions and conducting technical assessment of pharmacology, toxicology, and safety pharmacology data in support of licensing candidates. He was previously at Johnson & Johnson as the scientific director and as a research fellow. He also worked at Forest Pharmaceuticals as a senior principal scientist. And earlier in his career, he was a senior scientist with XOMA in the US. He holds a PhD in cardiac electrophysiology, an MSc in icosanoid pharmacology, and a BSc in human pharmacology from the University of British Columbia in Vancouver. So, Michael, we are all extremely happy that you are here today.
Can you hear me all right?
Yeah, we can hear you. You have a nice T-shirt. I like that.
Yeah,
Safe
that was,
pharmacology.
Yep.
Great. Yeah, we are really excited to hear your insights on your 20-year journey through the continuously evolving role of safety pharmacology. So, Michael, the stage is yours now.
Thank you, Sonja. I really appreciate Nanian asking me to provide such a talk. It's been, something that we've been thinking about the past presidents. So we are going to actually put a manuscript together that kind of takes everybody through each of the presidents from Pete Siegel through to our current president. So, when I started to put this talk together, I didn't think, I knew there were key points that we needed to discuss, to capture, but I didn't realize the totality of the data sets that we have generated over the years. So I'm going to kind of walk everybody through, pre-safety pharmacology, and then one of the remits of safety pharmacology, which, this group knows best of all, is the importance of the method development, method validation. As Jim just pointed out with regard to the ion channels, you can already see how far the electrophysiological aspects of safety pharmacology have developed, but we're still not there yet. And so, that is, I think, an important remit. And so I'll talk a little bit about different assays, and then I'll kind of follow up with kind of where we are in terms of the SIPA. I've got a slide or two on that, and also the ICHE 14S7B.
Sounds really good. And I still like the idea with that paper. We have to talk about that.
Yeah, we do. Yeah, we do.
Michael, the floor is all yours.
Thanks, Jason. So I thought it would be, I'm very appreciative for Nanian to ask me to give such a talk. Really, the society has taken, has been around for 20 years. It's totally amazing. I'll talk a little bit about how the society developed, why it developed. Well, we have physiology, we have toxicology, we have pharmacology, why we need safety pharmacology. Then one of the remits of safety pharmacology really is method model development and validation. And you can see that from Jim's talks and the other talks that are given about the importance of accurate values in order to determine safety profiles for drugs. And that starts early on in the discovery phase, where we have a lot of models that we look at through to the GLP that are involved in IND enabling aspects of pre-phase 1 type studies. So I'll present a little bit about everything. I'll present some models that I thought were really interesting that have been developed over the last few years. Many of you have likely used them. Many of you probably haven't. But let's start. So next slide, Jason. So it was a long time ago and not in a galaxy far, far away. Really was only 20 years ago, actually. But and this is difficult to consider now since we've been around 20 years, is that safety pharmacology really didn't exist as an independent discipline. It was really, you know, safety pharmacology was really sort of an extension of pharmacology. You know, why was that? Next slide. So you have to go back to the really look at drug development through the late 70s and really into the early 80s to get an appreciation of the types of studies that were conducted. Most of the studies types were not conducted, but if you look through the data back then, the majority focus was that a lot of cardiovascular and cardiac drugs were in development, and there was an awareness and data that existed for these drugs to prolong the QT interval and produce a unique arrhythmia, polymorphic VT called torsade de pointe. And you can see in the right side of the screen the you know, typical trace from way back in the 70s, probably the early 80s. That was a clinically relevant you know, arrhythmia that was present. We knew from multiple clinical trials, the CAST, SWORD, there's Diamond, there was a whole pile of studies that were ongoing through the early mid 80s to try and develop better antiarrhythmic drugs. So we developed flecainide, sotalol, amiodarone. They're all characterized according to Vaughan Williams' classification scheme, class 1 being sodium channels, class 3 being potassium channels. And class 4, there was all the calcium channels that were developed, which were less effective against the ventricular types of arrhythmias and more effective against supraventricular arrhythmias, like atrial fibrillation. But the understanding was that CV drugs were, you know, were relatively unsafe. So we knew that. Next slide. We also knew that QT prolongation, you know, produces an arrhythmia called TDP and or, you know, VF and causes sudden death. But there's a whole host of risk factors that were involved. Just the presence of a long QT interval. You know, now we know there's genetic variants that can cause it. Back then there was, you know, we know now that there's a precipitation if you're female to tend to have a longer QT interval. And there's a whole host of other factors, bradycardia, you know, renal issues, like I said earlier, genetics, modifying how the drugs are cleared by the liver and metabolized. One of the main ones was co-administration with drugs that prolong the QT interval. And again, this was still all somewhat circumspect back in the day. Next slide. So we knew that we can measure a lot of parameters, you know, and being a cardiovascular safety pharmacologist, you know, these are, you know, some of the standard parameters that we look at. Both we look at flow function. So in my current company, we look at a lot of ejection fractions and contractility measures since we're developing drugs for heart failure. In previous life, you know, in addition to that, previous life, I was looking at blood pressure. So you can see the parameters that you can look at. And of course, we're all familiar with the electrical activity, both the ECG and then looking at finer details within the heart and specifically at the cardiac action potential. Next slide. So we knew from that data set that, you know, if we pulled the heart apart, we probed it with microelectrodes, we could see this continuing evolving change in the action potential as you go from the SA node to the epicardium of the heart. And yet, in totality, if you looked at the global electrical activity, you get the nice PQRST ECG that we're all familiar with in the QT interval. And if you break that down and look at the components of each of those action potentials, you can see that they're aligned to activation, inactivation of certain ionic channels that are involved in depolarization and repolarization of the heart in order that it functions properly, pumps blood, and we're good to go. Next slide. So when we look down, so we dig down even further and look at just the cardiac action potential and splash a drug on it, you know, starting to discern which channels were involved. There's a multitude of them. You've probably heard tons from the last day and a half of lectures on it, so I'm not going to exacerbate it. There started to get some key factors such as methodologies looking at differences in action potential duration, triangulation, those sorts of parameters. And then on the right, if you look at, if you slowly prolong the action potential sufficiently, you get early after depolarizations. So on the other side of it was the understanding of potential mechanisms that were involved in development of the arrhythmia, and then slowly the link between drugs causing those types of arrhythmias. Next slide. And again, this is just an overview of the ECG itself. As safety pharmacologists, we're really familiar with the typical ECG parameters that we look at, the PR, QR, SQT. You may look at the ST segment as well. We looked at the R wave amplitude. We were developing cardiac drugs back in the late 80s, early 90s, as I think most folks who are safety pharmacologists were also doing. More recently, there's been an emphasis on developing additional parameters, JT-peak, T-peak to Tn, to complement the QT interval changes, and looking at transmural dispersion of repolarization. So we had all this background work. We knew that cardiovascular drugs were problematic. There was concerns. Clinical studies had shown it. Next slide. We knew that drugs were not selective or specific. We know, and I've just put this together for the standard drugs that we know. If you look at class 1 sodium channel blockers, if you look at the lower left is tetrodotoxin, that's highly specific and selective for the sodium channel. If you look in the upper right, you've got dofetilide and E4031, variant structures that are very highly selective for IKR blockade that we all know and use as both positive control drugs and produce torsade de pointe. But you've got a spectrum between that, and this is only 2 of the ion channels. Now we know there's like a dozen. Again, so evolving data showed that there was, even though we wanted it to be specific and selective, a lack of specificity and selectivity of drugs. So in the backs of our minds, it's a matter of dose and response and physiological effect and the reasons behind that relating to ion channels. So the data was evolving, the drugs were evolving, we knew there was issues. Next slide. But it really wasn't until we had non-cardiovascular drugs that were beginning to show development of arrhythmias. They were precipitating, and it was combinations of certain drugs that became more prominent. I remember reading New England Journal of Medicine papers back in the late 80s, early 90s that showed, ketoconazole plus certain drugs could cause QT interval prolongation. Again, in addition to the changes in rates, there's components like stretch, there's changes in electrolytes that can cause it. We definitely knew that the class, 1As, 1Cs, and 3s were specifically unsafe at certain doses and in certain patient populations. But it really wasn't until non-cardiovascular drugs, simple benign drugs, antihistamines, antibiotics, those sorts of drugs, some tricyclics, were starting to show that there were issues, antidepressants as well. So now it was like a wow, and again, the development of the methodology, our understanding of the types of arrhythmias, and our understanding and development of the electrophysiological profiles that drugs can have on the heart sort of were all coming together at the same time. Next slide. But it really wasn't until pharmaceutical companies either had to withdraw drugs from the market, were given black box label warnings for restricting prescriptions or labeling implications, and there's tons of those. And you can see that withdrawn from the market, there were a lot of drugs. Drugs simply like cisapride, it's a prokinetic drug, really. Terfenadine, antibiotics, anti-incontinence drugs, and or prescribing restrictions. That's when safety pharmacology started to come together. Next slide. So this was like one of the really interesting papers that came out in 1993 in JAMA, showing that when you looked at the percentage of patients that had QT prolongation greater than 500 milliseconds on antiarrhythmic drugs, it's between 80 and 90%. and looked at the non-antiarrhythmic drugs, they were not any different. And that was the green light to say, wait a minute, we need to do something about this. And folks within the industry got together, regulators got together. You can see on the right the types of drugs that are listed again that produce an increase in the QT interval. So all this data was coming out, something needed to be done. That's when safety pharmacology happened. Next slide. So I'll talk a little bit about the Safety Pharmacology Society that formed, and a little bit of the regulatory guidance implementation for study conduct that was implicit to the types of studies that were and are carved out and manifest in the safety pharmacology study profiles. Next slide. Ooh, hit one more time, Jason. Okay. So back in the early 90s, there were no guidance documents. There was nothing really confirmatory, regulatory in nature. Pharmaceuticals prior to 1991, really the studies that were done were really pharmacological in nature. If you go back to the different regulatory guidelines, there were concept statements in the US and in the UK. General pharmacological profiles really was what it was. The Japanese were really the first to really distinguish descriptive from safety pharmacology for any type of new chemical entity. And you can see the listing of the types of studies that they began to compile into, and there were lists back in the day, given more of the pharmacological description on the left versus how do we characterize the safety profile on the right. And so that was sort of the template that was taken and used to develop the guidance documents that we have now. Next slide. And so Safety Pharmacology Society was formed in 2000. Right now there's 500 members globally. As you all know, we have annual meetings that rotate between North America and Europe. We have educational webinars. We try to have regional meetings, except for this last year due to COVID. Awards, we have vendors that come to the meeting. You know, the list goes on and on and on and on about what safety pharmacology does. And if you care to look, there's, and you can find it on the website, what the vision is to, you know, lead global safety pharmacology community in the development of safe medical use of biologically active compounds. Next slide. So I guess a lot of the questions that I was asking or being asked back in the day when the society was first coming together is, well, safety pharmacology, isn't that like toxicology? It's like, no, it's not toxicology. So as the guidelines were developed, the S7A and S7B, they were separate from the M3 guidelines, which is a predominant toxicological. Safety pharmacology studies, as we know, they're usually a single dose acute type studies, moderate exposures to look at moderate adverse effects, whereas toxicology, we know we dose very high, very long to determine NOAELs, and we look at organ endpoints and hematological changes and clinical chemistries. Safety pharmacology are short-term, reversible, functional effects, usually recorded from conscious animals. And I'll talk a little bit about the in vivo QT dog study that we use typically. We look at the onset, duration, and magnitude of effects. So, you know, in a toxicokinetic, pharmacokinetic world, it's Cmax driven. It's what's that maximum drug concentration? How's that affecting the physiological parameters that we want to look at? Whereas toxicology, it's AUC. But both sets of studies are pivotal and are conducted under GLP. Next slide. So as I talked about earlier, you know, there's a number of ICH guidance documents. These are a few of them. They're the ICH S7A and B, and then B being potentially supplemented through the ICH E14 S7B draft guidance document developed relatively recently. Really are what shaped safety pharmacology as the discipline that we know now. There are others, you know, that are involved and may have components, but the predominant is the S7A, S7B. Next slide. So where does safety pharmacology fit into this overall drug development scheme? Well, I kind of like to nestle it between pharmacology, since pharmacology is all about dose response, determining efficacy models and your compound compared to, if you were lucky, positive controls for that activity, and toxicology, where we have the short and chronic type of studies, long-term dosing, looking at endpoints that are related to histopathology changes. Safety pharmacology is in that little band between the two, and I kind of liken it to extrapolating between both disciplines, and is composed of, as you all know, the core battery, which looks at CNS. We have respiratory functional changes. We have the cardiovascular effects, HERG and DOG, and a series of ancillary studies depending on what changes you see or what the indications are for your compound or what the target is. And then I've added SIPA, since we've are now tightening up different aspects of and adding more details to understanding your drug effects on multiple ion channels across the spectrum that are involved in heart function. Next slide. So this is just in totality safety pharmacology. Most of the endpoints that you can look at for any of the core battery assays. I have SIPA coming in from the corner here as a little Pac-Man that is now actually part of the whole integrated risk assessment. So this is where safety pharmacology is now, after 20 years. Next slide. So I'll talk a little bit about the current non-battery non-clinical, or the core battery non-clinical models, and some of the additional non-clinical models, assays that are used that you may be familiar with, you may not, you might have heard about, you might not have heard about. Next slide. So back in about 2004, first of all, if you look at how do you define safety for pharmacology? It's the study of the potential undesirable pharmacodynamic effects of a substance in relation to dosage with the substance, therapeutic range, and above. And again, it's to identify pharmacodynamic properties that may be relevant to those first in-human phase 1 type of clinical studies. We want to evaluate adverse pharmacodynamic effects, investigate mechanisms of action if that's possible. Studies are specific with properties and uses of the drug, so you will tailor your program to that. The important distinction between, say, some of the other disciplines and safety pharma is that we use validated methods. That's preferred, and this group, if you sift through the literature that we've compiled since 2004, as you can see on the right, you can really see the marked increase in technologies and development of preclinical methods and refinement of those methods. I was very frustrated prior to 2004 because we started the society was founded. People were starting to develop the assays that we were looking at, they were being used, but there was no literature on safety pharmacology. So myself and Mike Curtis, who was editor-in-chief of Journal of Pharmacological and Toxicological Methods, said, hey, why don't we give an issue each year to the meeting, reports that come out and manuscripts. And so you can see over the years from 2004, and I just updated to 2020, where I'm currently compiling the 2021 issue. You can see on average, and I broke it out into total articles and then the different cardiovascular, CNS, respiratory, how dynamic safety pharmacology has been over the years. And most recently, the cardiac stem cells, papers involving cardiac myocytes, SIPA, and in-silico methods and models have really started to increase, obviously, compared to earlier times. Next slide. So again, as I mentioned earlier, safety pharmacology includes both early or exploratory as well as regulatory studies. And I'm not going to go through this slide. Everybody's familiar with it. There may be earlier methods of models that will help you discern, in lead candidate selection, and they range from in vitro and in vivo versus the regulatory, application of S7A and S7B, as we've, been discussing here, for studies that are mandated to be required and included in your IND submissions. Next slide. So some of the models that I thought were really neat. still do. The zebrafish, early screening model, I remember must have been in mid 2000s, mid to late 2000s. We started to get a flurry of zebrafish papers. Currently it still remains the only vertebrate model amenable to both medium and high throughput in vivo tox. I don't know between now when we were really using it or groups were trying to use it, and now if we've had a better establishment of some of the issues that people or concerns that they had about, metabolism, transporters, those sorts of things, and how they correlate. The data that you generate correlates to mammalian models. The assay was fairly sensitive from the literature that I looked at. There were some differences in margins. If you looked at, say, an IC50, it could be up to 250-fold, but not outside the realm, as Jim nicely pointed out in his previous lecture. But solubility was an issue, and so that was an early interesting screening assay that I think is still being used with some shift in focus. Next slide. So again, I'm not going to belabor the point. There's a lot of high throughput patch clamp and different micro manipulation. As Jim pointed out, we can now record at 37 degrees centigrade. That was physiological. That was impossible with the early types of single patch clamp devices that I was using back when I was a grad student or doing my postgraduate work. So you can see that the fidelity of the equipment, the recording equipment, the technology has superseded, has exploded. Next slide. I thought this was an interesting slide. It was a presentation that was given in about 2009 at the Safety Farm meeting, is cardiac slices. It's a method that I've never looked at, but it seemed physiologically possible at the time. I'm not sure if it's well used or I know it's been characterized, but I'm not sure if it's well used now. And you use the synchro slice to cut your either vertical transmural or horizontal transmural or tangential preparations for electrophysiological recording. Next slide. This is the wedge preparation. I think everybody's familiar with this, and developed by Ncelevic back in 2005. Very well validated. Multiple groups have looked at many compounds in the model as a nice means by which to evaluate epicardial, mid-myocardial, and endocardial effects of drugs, and trying to get a handle on the ion channel profile and potential for action potential heterogeneity and proarrhythmia development. Next slide. The next one that has been used for many years is the female rabbit Langendorf isolated heart. You can measure both perfusion pressure, flow, left ventricular pressure. You can measure pH, electrolytes, coronary flow, contractility, ECGs, all of it. I did my master's thesis using isolated hearts. Still tried and true, looking at contractility and electrical effects. Next slide. So these are, the two means by which the core battery safety pharmacology cardiovascular system profiling is completed. The in vitro voltage clamp HERG assay, and we've heard a lot about that, so we're all familiar with that, but also the telemetry dog method, which allows you, 24-hour beat-to-beat recording, high resolutions, stress-free now that we have implants. QT correction methods, and a lot of us have been working on modifications to the assay, tightening it up, looking at through the ICH E14S7B Q&A documents. Next slide. And I put this slide in just because it's something that's always been near and dear to my heart is blood pressure. And we are putting together a group to investigate blood pressure, since it's a key parameter that if you look at the Lindgren 2008 survey that we did, everybody that looks at drug effects early on even does blood pressure measurements. But we want to make, and we know the complications involved, and you can see it depends on intrinsic reflex, hormonal, renal, and microvascular control systems. But we are gearing up to look at, Mike Foley and I put together a proposal with the HESI group to examine drug-induced changes in blood pressure using a standard in vivo safety pharmacology model, i.e. the CV dog. Next slide. So this is a slide that I've always loved. It's sort of just, it's a big screen snapshot that tells you all the cardiovascular methods, models that you can apply at different stages of advancing a candidate all the way through to first in human. And it involves looking at, you know, like I said, all the ion channels and any other parameters that you think are relevant to your molecule. Next slide. So the second set of core battery assays is the respiratory assessment. I've only put one slide in on it. It's unfortunately, I think, almost become a checkbox exercise for a lot of people, where there's been somewhat of a Safety Pharmacology Society review of that. We are trying to compile data sets to look at that. You know, we look at respiratory rate, tidal volume, and there's multiple methods. You can use plethysmography just to look at chamber pressure changes, lung airflow, tidal volume, breathing rates. There's face mask, depending if that's, you know, more relevant to the species that you're looking at. We've done studies. I've done studies with colleagues in monkeys looking at inductance straps or impedance electrode measurements. You know, not much more to be said about that. Next slide. Now, what has been dynamic over the years in safety pharm has been CNS evaluation. You know, not only looking at, you know, the Irwin and the functional observational battery, and we've got a whole series of papers over the last 15 years at least, and a lot last year in our SPS issue of JPTM on CNS studies and diversification of those, and looking at neuronal preps and the likes. There's also been an upsurge in looking at the risk of seizure thresholds, and you know, we're looking at, there's been a series of studies looking at proconvulsive potential and even the effects of the drugs on cognition using the, you know, standard models like the Morris Maze and the passive avoidance type models, and a lot with EEG, like I said, EEG methodologies as well. You know, so really advancing the CNS aspects of drug safety pharmacology. Next slide. And as a component to that, and I have her book, our dear friend Carrie Markgraf, who was really her and Mary Jean Coleman involved in the abuse liability guidance developments. You know, the importance of that related to physical dependence, withdrawal, you know, considering drugs that affect the CNS system, and there's a whole list of them on the left. Looking at and considering, you know, high doses, IV administration, those sorts of parameters that you may be looking at with your compound, and then breaking out, you know, what the non-clinical safety assays are, looking at the early types of studies, binding and the structure, and relating that to any of the known drugs of abuse, and then developing on, you know, physical dependence, drug administration, and drug discrimination type of models, which are very well defined now to address you know, safety concerns, public health, and provide a basis for regulatory actions of drug control. Yes, abuse liability, you know, didn't, as far as I was aware when Safety Pharm started, didn't really exist, and now there's a very defined profile that, you know, needs to be followed. So well done again for the society in implementing this component of CNS aspects of drug development. Next slide. And then what we've seen over the last years is, rather than just single studies that look at central CNS effects, respiratory, cardiovascular, I haven't seen recently an integration of models, but there are, if you go back through our literature over the last 16, 17 years, I think we have now, you'll see one-offs of CNS and respiratory or CNS cardiovascular where there's been a very clever design study, changes to standard study designs to optimize the animal models so they assist in NC3Rs, as well as you know, again, just optimizing the model to best characterize the effect in more than one aspect of the core battery studies. Next slide. And again, this is, you know, I'm not going to belabor the point. In totality, this is, you know, as of 2005, what the non-clinical testing strategy was. I have a few slides that will sort of illuminate that this has really changed a lot and will change a lot, especially with the implementation of the new ICHS7 E14S7B Q&A documents. Next slide. So that's kind of where I'm at right now, is sort of we progressed from why do we develop safety pharmacology, why do we need to do it, give you a snapshot of the types of models that we're looking at that we developed and validated through the years. There's way more than the few snapshots of slides that I've put together. And now, you know, these are sort of the two areas that we've really worked on. Majority of folks for at least for SIPA. I was trying to go back to see what the original date was, and I see around, you know, 2010, 12, 13 is when that sort of those changes became considered, and then we moved to where we are now. And then the ICHE 14S7B started in 2020. So I have a few slides on these two aspects, and that'll be it. So next slide, Jason. Yeah, so this is SIPA. Everybody's familiar with it. You know, it's to provide a more comprehensive assessment of proarrhythmic potential. And again, back in the day, was to not obviate conduct of the non-clinical in vivo core battery ECG assessment, which is what we're updating now. And you know, the remit for SIPA was to focus on proarrhythmia, not erg-centric or QT prolongation in and of itself. And it's a composite of in-silico models and the effect of the drugs on stem cells, which there's, and again, no time to overview any of the good work that's been done in evolution of and use of cardiac myocytes derived from stem cells. But you know, again, to feed into complementing and providing you know, another series of ECG biomarkers for phase 1 clinical studies. Next slide. And so this was I found a couple papers that, well, on the left, that Gary Gintant published in 2020, which is just a beautiful overview of the different recording methods for stem cells. I thought it was just, I was just rereading part of the paper the other day, and again, revel in the creativity of scientists in adapting and developing models for evaluating, you know, looking at drug effects on stem cells. So you can look at, if you look at panel A, there's transmembrane recordings. So you've got either electrical or optical types of methods. And to look at drug effects on the QT, you can look at extracellular field potentials. And there's an example of cisapride for MEA type of assays. More recently, there's contractility in motion, which is, you know, both either looking indirectly at calcium transient changes in terms of modulating contractility or impedance measurements. And there's even Sony pixel movement that looks at arrhythmias by monitoring pixel movements. It's just amazing. You know, some of the best practices that are been discussed and involving stem cells are the, you know, the electrophysiology maybe vary, and that depends on the genetics behind the cells. There's karyotypic stability. Efficiency of differentiation protocols was called out. You know, I think a lot of the issues that were early on have been much resolved using standard protocols. As Jim explained, we're using we used to use pulse protocols. Now we use ramp or pulse ramp protocols, which better reflect what is happening physiologically. We're getting better at defining assay sensitivity, and you saw that through some of the electrophysiology that Jim presented. And then on the right-hand side, this is a paper that was just published by Hugo Vargas and his group, looking at actually omecamtiv mecarbil, one of the drugs that we're developing for heart failure. And they did a beautiful in silico simulation using 118 myocytes, and looked at and compared it to standard drugs like flecainide and that, and just showed that in silico models complemented what they had found when they looked at the electrophysiological profile. And they used the virtual assay based on O'Hara-Rudy and looked at the integrated response to at least 9 ion channels, and actually could from that data prescribe a proarrhythmia value for omecamtiv mecarbil of 0, versus you can see in the lower right, flecainide was 0.115. So again, just a nice use of computer models to substantiate what our other non-clinical models can provide in terms of safety profile. Next slide. And this is what we have ongoing now, is updating the integrated proarrhythmia risk assessment. This just came out. This is in Hugo Vargas's paper with a lot of safety pharma colleagues, that was published in 2020. And I'm not going to go through most of this, but you can just see how we are now, modifying the standard integrated risk assessment by implementing both the the in vitro types of study designs that Jim nicely outlined, with modifications to the in vivo QT dog assay that is currently ongoing. And there's going to be a series of manuscripts that will be coming out to kind of pull apart the CHE14S7B Q&A document and provide sponsors, I guess, with strategies and what the components are and how they're going to be needed in order to optimize that non-clinical model. And again, we really want it to substantiate the non-clinical data derived from the assays that we do, and to better merge the non-clinical and clinical data to provide a better comprehensive clinical risk assessment strategy. That's the ultimate goal of safety pharmacology. Next slide. And I'm going to end with this slide. I was going through trying to find all of the meeting titles from where we have had the meeting. We've had so many since its inception. Most of them I couldn't find, and just to kind of give you an appreciation of the breadth and depth of safety pharmacology as a discipline now, it's really remarkable, I think, and it just shows that the dedication to those members of the society who are involved in drug safety. And I think I'll end it with that.
All right, Michael, thank you so much.
You're welcome.
Thank you so much for handling these difficulties. In the end, it was a really great talk now.
Oh, good. Yeah, I mean, it's just, there's so much, with safety pharmacology is there's so many aspects of it. It's very difficult to capture it. And, I think the best thing is, if people are really curious, is to look at the Safety Pharm annual issues of the Journal of Pharmacological Methods, because you'll really see, or at least even the editorials, because we try and write those to give a snapshot of all the manuscripts that are involved in that article, and you can just see how it waxes and wanes. Sonja, you can see the early cardiovascular models, they kind of decline because they've been done, validated, and now people are using them. Then CNS came along, and then that declined, and then now you see all the stem cell work going on, from validating the cells to all of the different recording methods to, you know, trying to standardize all the parameters. And now we've got, like I said, the, you know, increase in, or not increase, but we're really trying to nail down, and I think it's good, because I think Jim's data was amazing to show, you know, we all know the variability in the response, but we're all willing to tolerate it. Now we're not, because, and these ICHS-7, E14F7B guidelines will really, I think, tighten up, you know, all the components that we're
yeah, definitely very interesting times these days, and we have to see what comes up the next 20 years or maybe 5 years or whatever. Yeah, I see a lot of things going on in the chat here. So Rodolfo Heido says, what a wonderful educational story, Michael. Bravo.
Oh, thank you.
And thank you. And we need more of this. The big picture is also illuminating. Sometimes we go so deep into the details and forget our purpose. And Damien Bell has also info. Thanks for sharing your experience in such a broad and deep overview of safety pharmacology. Looking forward to your specialty safety pharmacology issue at JPTM, also from Damien. So the chat is going on here. I think very nice overview. Thanks.
And Damien actually just submitted a really nice article on HERG, so I really recommend people go and read it. I have read it myself, as I do with all the manuscripts that we get. Just a really nice article. It complements what Derek Leachman had published last year on sort of HERG over 20 years. It just takes it a little bit more, you know, into today. So yeah, great article.
Fantastic. I will read that. Okay, now let's go to our final speaker for today then. So thank you again, Michael. Thank you for your time. Great talk. And our next speaker, our final speaker for today is Alfred L. George. So he's a professor and chair of the Department of Pharmacology and director of the Center of Pharmacogenomics at the Northwestern University Feinberg School of Medicine. Al started his education at University of Rochester and continued to develop his rich career through postdocs, fellowships, and residencies in the US and Europe. His work was contributed greatly to understanding the mechanism by which ion channel mutations cause a variety of inherited disorders, including cardiac arrhythmia and epilepsy. So, welcome, Al.
Thank you.
Nice to see you.
Thank you very much. Can you hear me okay?
Yes, we can hear you. We can see your slide perfectly. Perfect. So the stage is yours.
Thank you very much. Really, thank you very much for the opportunity to participate in this symposium. I'm grateful to the organizers and my friends and colleagues at NANION for the invitation. What I'm going to tell you about in the next 20 or 25 minutes is our experience using automated patch clamp to assess sodium channel blocker effects on human iPS cell-derived cardiomyocytes. And this could be viewed as sort of a technical talk introducing the audience to one way to assess one class of blocker on human cardiomyocytes as a way of entering into the cardiac safety field. The background here, of course, has probably been covered multiple times over the last day and a half, is there are multiple mechanisms and phenomenon associated with cardiotoxicity related to a variety of antiarrhythmic drugs. The ones highlighted here are, of course, the class 1 sodium channel blockers, which are well known for their proarrhythmic effects, especially in some situations, such as in ischemic heart disease and heart failure. They also have negative ionotropic effects and affect conduction delay. And very recently, there's been attention paid to the fact that class 1 sodium channel blockers and other drugs with shared mechanisms of action may be responsible for some cases of drug-induced Brugada syndrome. And Brugada syndrome is generally viewed as a genetic disorder of increased susceptibility to sudden cardiac death. Now, the original discovery that sodium channel blockers of some class 1 agents were associated with increased risk of ventricular arrhythmias and sudden death came from, of course, the cardiac arrhythmia suppression trial, or CAST trial. And here is the original publication from 1991, and the original epidemiological data showing that there were excess deaths in the treatment group. And we think now, in retrospect, this was probably due to sodium channels affecting conduction in the ventricle and ventricular myocardium, and during ischemia you might get unidirectional block and predisposition to reentrant arrhythmias. So moving on, we've adopted, with the help of colleagues here at Northwestern, a robust program to use human iPS cell-derived cardiomyocytes to do pharmacological experiments. And so the real question in the beginning was, can we use these cells for studying the pharmacology of sodium channel blockers? And that really came down to, could we use these in a high throughput assay such as automated patch clamping? And the work I'm going to show you really came out last November in Molecular Pharmacology, and the first author, Franck Potit, has since left my lab and joined Icogen. But the work I'll be showing you is the majority of the work was his. We have a collaboration with Paul Burridge, who's a member of my Department of Pharmacology at Northwestern. Paul's been a pioneer in developing streamlined and efficient methods for differentiating cardiomyocytes from iPS cells. And what's illustrated in the upper part of the slide here is simply the initial cardiac differentiation protocol that he developed and published back in 2014. We adopted this method to determine how to process the cells and how to disperse the cells so they had optimal qualities that were amenable to automated patch clamp recording. And so just as a summary of the method that we had developed and published in this paper last year, was we differentiate the cells and grow them until day 20, in which case we dissociate them and then replate them. And this seems to be a critical step for allowing the cells to grow at the right confluence and preserve their function such that we can get good and high fidelity seals on the automated patch clamp. We actually grow them in a little bit of fetal bovine serum for another week, and then they're detached and dissociated and resuspended for placement on the automated patch clamp system. Importantly, cells differentiated in this manner have a high expression of the NAV 1.5 cardiac sodium channel. There are some other sodium channel messenger RNAs, but we're pretty confident in saying that the majority, well over 85%, probably closer to 100% of the active sodium current in these cells is due to NAV 1.5. We use the SynchroPatch 768 platform from Nanion to perform these studies. All the work I'll be showing you used 384 well plates with a single low resistance hole. And we use the whole cell broken patch whole cell configuration for these studies. We used internal and external solutions as illustrated here to try to isolate sodium current and block potassium currents. We can see some endogenous calcium current in these experiments, but we have different solutions that we optimized for recording calcium current. For the study I'm going to show you, this is a relatively typical screenshot illustrating the 384 well platform during a recording experiment with the green shaded wells highlighting wells that have a gigaohm seal. And in this experiment, we were able to capture approximately 70% of the wells with cells and approximately an average of 0.6 gigaohms. That's actually the average for the entire study, I should say. On average, we're able to get, for all the experiments that we've done to date, we get an average of cell catch ranging from 22 to 84. The 22 is sort of on the low end that was early in our experience. More recently, we're really up in the 80s most of the time, if not higher. Average seal resistance can be close to a gigaohm. And then we've quantified the percentage of cells that we've captured and recorded from that have sodium current. And that tends to average around 50%. If we eliminate cells with smaller currents, less than 300 picoamps, and those cells that have the best voltage control, if we include those, we get about 25% of the cells at the end of the day. So, you know, we get close to 100 cells or more per experiment, and that's a robust number of cells even on a bad day for doing pharmacological studies. And this just highlights some examples of some of the better wells. So summarizing some of the data, this is just average data over the first several or over several plates of experiments. This is sort of our average typical whole cell current that we can see, and this is obviously a beautiful voltage-gated sodium current, which has a relatively canonical current voltage relationship, as well as voltage dependencies of both inactivation and activation. And so we were pretty confident that we were recording a robust sodium current. We can compare these data also with heterologously expressed NaV 1.5, and the data align very closely with one exception, and that is we do see a little bit of a shift in the voltage dependence of inactivation in cardiomyocytes compared to hex cell expression of the same channel. We think that's due to the use of high extracellular calcium transiently to acquire the high seal resistance in the automated patch clamp. We've been working on methods to try to eliminate that, and I think we've got some newer protocols that virtually eliminate the difference in the voltage dependence of inactivation. So now turning to pharmacology, we initially set out to do a comparative study of 5 drugs illustrated here, including some classic local anesthetic-like drugs, lidocaine, and some newer agents that were purported as being selective blockers of late sodium current, including ranolazine and the experimental compound GS967, and aleclazine, which was clinically trialed for type 3 long QT syndrome. We also added in lacosamide. Now, lacosamide is not a cardiac drug. It's an anticonvulsant drug, but it works by an unusual mechanism. It works by stabilizing the slow inactivation state, and it really was purported to have very little effects on the fast inactivation of the neuronal sodium channels. And there has been some emergent reports of lacosamide cardiotoxicity, including some case reports of lacosamide inducing Brugada syndrome. And so we thought this might be a relevant drug for comparison. And so we first looked at tonic block of the channel, and here we simply applied the drug and tested with single pulses at low frequency, as illustrated here, to just determine what is the level of block of the peak sodium current in these cells. And so here, just as an example, we show for lidocaine, the black trace is what we record in the absence of the drug, and then the red trace is in the presence of the drug. And then for ranolazine, you can see at even a lower concentration than lidocaine, you get substantial peak block. These are extremely high industrial strength concentrations of ranolazine. And then the other drugs that we tested have seemingly less degrees of tonic block at the concentrations we selected. And we can use the power of automated patch clamp to do concentration response relationships. And so here's just an example of some of the data for lidocaine, where we looked at a range of concentrations. I think there's 7 or 8 concentrations here, with each data point representing somewhere between 25 and 45 cells on average. And we were able to actually get nice concentration response curves that we can fit to determine the IC50. And the IC50, or the concentration response curves, are shown here for all 5 drugs. This represents a tremendous amount of recordings. And I should say, just overall, this study that we published last year represented data from more than 2,500 cardiomyocyte recording experiments. So this gives you an idea of sort of the diversity of potency for use-dependent block. If we rank order them by IC50, GS967 was the most potent, followed by leclizine, ranolazine, lidocaine, and lacosamide. GS967, again, it was one of the 3 drugs we tested, including leclizine and ranolazine, that were purported to be selective late sodium current blockers. And this would give you sort of a different take on that, which is at some concentrations you get fairly high peak block as well. Now we turned our attention away from tonic block and studied use-dependent block of, again, the peak sodium current. And we used 3 pulse protocols. The first protocol, which was designed really around kind of low frequency, short pulses, is shown here. We then ratcheted up the frequency, but using the same pulse duration. And then to mimic the cardiac cycle, we lengthened the duration of the stimulus, and this limited us to only doing studies at 2 hertz. For all these studies, we went out to 30 pulses and then looked at the residual current after 30 pulses and compared that to the baseline. And so we get data that looks like this for lidocaine at different concentrations, including a control in the absence of drug. You can see most of the concentrations have very little blocking effect at the low frequency pulsing with the short duration stimuli. That's enhanced with going to 10 hertz and similar block when you go to a 2 hertz at the 400 millisecond width pulse. At 400 milliseconds, you are starting to see some endogenous decay in the current, even in the absence of the drug, and we think that's simply a representation of channels entering the inactivated state and not recovering in the interval between pulses. But in all cases, the drugs enhance use-dependent block. Here's another example for ranolazine. Again, characteristically, it was referred to as a late sodium current blocker, but under some conditions, you can see impressive use-dependent block. Now, right around the upper limit of the therapeutic concentration, 10 micromolar, you get a fair bit of use-dependent block, and you can accentuate that at supra-therapeutic concentrations. And here are the concentration response curves determined for all 5 drugs under different concentrations under these 3 pulsing regimes. So again, a lot of data collected in this one experiment that would be virtually impossible to do without an automated patch clamp instrument. So again, at low frequency, short pulses, most of the drugs have very modest use-dependent block, but as we get up to the protocol that simulates tachycardia in a heart, 400 millisecond duration, 2 hertz, we get quite a bit of use-dependent block, and we start to see again that GS967 is sort of emerging as the most potent of these drugs, followed by leclizine, ranolazine, and then finally lidocaine. And this was sort of news to us that we used to think lidocaine was a fairly potent use-dependent blocker, certainly one of the classic use-dependent blocking drugs. But certainly it pales in comparison to the potency of these other these investigational drugs. And so just as a sort of a summary, if we look at the IC50 values for tonic block and all in micromolar, use-dependent block using the 10 hertz short pulse protocol, you can see that there's quite a diversity of effects. If you make the ratio between tonic block and use-dependent block, you can get a sense that ranolazine seems to be the one that has the biggest disparity between peak block and use-dependent block, and on the other end of the spectrum is GS967. If you compare the 2 investigational drugs to lidocaine, they're very impressive. They're upwards of 2,000-fold more potent than lidocaine for use-dependent block, and for GS967, and over 200-fold more potent than lidocaine for eleclizine. And these drugs were originally developed as potential antiarrhythmic drugs, and you can see that perhaps their antiarrhythmic effect might be tracking with the potency and use-dependent block. Now, we are interested in understanding a little more about why particularly the investigational compounds, GS967 and eleclazine, were so potent at use-dependent block? What was the underlying mechanism there? So we turned our attention to looking at the kinetics of block, and to do that, we adopted two protocols. The first is this protocol to assay recovery from inactivation. Recovery from inactivation specifically evoked by a 1 second duration depolarizing pulse that will drive channels not only into the typical fast inactivated state, but will begin to engage slow inactivated states as well. And then we induce a variable duration recovery pulse, and then a final test pulse to determine the residual available current after this regime. For example, lidocaine here shows that the recovery from inactivation is slowed by the drug and in a concentration dependent manner, and that was observed for all of the drugs that we tested to different extents. But in addition to the phenomenon that we observed, we can also infer from these data the drug dissociation rate by comparing the recovery in the absence of the drug versus in the presence of the drug, and actually calculate the kinetics of drug dissociation, or the K-off. And when we did that, we can actually, again, rank order the drugs by their dissociation rate constant. So ranolazine has the fastest off rate, followed by this cluster of GS967, eleclazine, and lidocaine, which are all clustered together. And then the slowest off rate was observed for lacosamide. And I mentioned before, this was the drug that really has unusual properties of stabilizing the slow inactivated state. We also used this protocol to evaluate kinetics of block, and that is the entry into slow inactivation. And this was really designed to look at the on rate of the drug effect. This protocol is designed to give a variable length pre-pulse to a depolarizing potential, going from 1 millisecond all the way out to 1,000 milliseconds. Again, we would expect to see at the shorter pulsing frequencies primarily engaging fast inactivation, with the longer duration pulses engaging more and more slow inactivation. Now, to remove the component responsible for fast inactivation, we introduce a short recovery interval that will then will allow channels to recover from fast inactivation, and then before we do our final test pulse to assess how much residual available current is left. And so some representative data for lidocaine at low concentrations. You see there's the minimal block at short durations, and that gets enhanced as you get longer and longer block. And certainly at high concentrations, you get block across the time spectrum. There's quite a diversity. If you look at GS967, much lower concentrations are required to induce this entry into slow inactivation. And then lacosamide really has almost no effect on the channels at short pre-pulse durations, and really only becomes active at the longer pre-pulse durations. Again, evidence suggesting that this drug has a predilection for interacting with the channel in its slow inactivated state. And again, from these data, we can actually fit the decay curves with an exponential function and derive a time constant, which we equate to the on rate of the drug. And so here are the calculated, the reciprocal of the time constant plotted versus concentration gives us a slope that we can equate with the apparent on rate of the drug. Now, only shown here are 4 of the drugs. The reason we couldn't really calculate ranolazine by this same mechanism is, you may remember a few slides ago, I showed that the off rate of the drug was such that you could imagine the drug coming off during the 20 millisecond recovery interval. And so we couldn't rely on that, on this protocol for that. So here we had to make some assumptions and back calculate the on rate from this equation. But at the end of the day, we had these really unusual and unique collection of data on the kinetics of block of this group of 5 distinct drugs, and we actually could plot them and look at their comparative kinetics. And so what's shown here is certainly the drug that had the most potent effect in our experiments, GS967, has this sort of unique blend of very rapid on rates and modest dissociation rates, dissociation rates that are not dissimilar from lidocaine. And sort of the rank order potency that we were observing for use-dependent block seems to follow more of the on-rate kinetics than the off-rate kinetics. Ranolazine is a little unusual. Again, the off-rate is well out here. The on-rate, which we've estimated, is sort of in the ballpark of eleclazine. And then lacosamide, which is this unusual drug with a very slow on-rate, but a modest or moderate off-rate, is probably more specific for the slow and activating channels. So this is just sort of an example of some of the data that we were able to collect using our automated patch clamp system to record sodium current from human iPS cell-derived cardiomyocytes. I hope the take-home message here is that is feasible and a robust assay. We did have to go through quite a bit of optimization of the cardiomyocyte differentiation and cell culture and dissociation methods. I spared you of those details and just kind of gave you the state of the art at where we are now. These are really essential things to optimize and have been continuing to improve in our hands. And so again, just to sort of take home message, we've been able to show that it's possible to do mechanistic and molecular pharmacological studies using automated patch clamp on these human cardiomyocytes. And then one of the real take-home messages for the impact on these 2 experimental drugs is they are very potent use-dependent blockers of peak sodium current and cardiomyocytes, and that perhaps underlies their in vitro experimental antiarrhythmic properties. Just to thank the people that did the work, again, I highlighted Franck Petit at the beginning of the talk. He was really sort of the master behind the electrophysiology in this study. And then my collaborator, Paul Burridge, and people from his lab who contributed to generating and optimizing the conditions for the iPS cell-derived cardiomyocytes. And then the last picture of the lab in Chicago. So anyway, thank you very much. I appreciate the opportunity to talk and would be happy to entertain questions if there are any.
Yes, there are questions. Thank you so much, and very good presentation. Nice data. So let's see. We have a couple of questions here. Jérôme Monard, do you have an idea of cardiomyocyte, non-cardiomyocyte ratio?
Yeah, so in the cultures, I didn't emphasize this, but these cultures from Dr. Burridge's lab are from iPS cells that have stable integration of a selectable marker. And so these cells are placed under selection to get rid of all the non-cardiomyocyte, or at least enrich in cardiomyocytes. And so we don't have a number, but by flow cytometer readings, it looks like there's well over 90% of the cells are cardiomyocytes.
Mm-hmm. Maybe one tiny question as a follow-up from Ralf Kettenhofen. Have the drug experiments been conducted with cardiomyocytes derived from a single iPS cell line? Yeah?
Yes, they were. Yes, so these were all derived from a single iPS cell line. It's a control line that was that's used widely in the Burridge lab, and that was what we started with. And if I'm not mistaken, it's derived from a male.
Mm. Okay.
Not a very diverse array of cardiomyocytes.
Yeah. Okay, so for the sake of time, we continue now. All the other questions will be answered for sure. So, wow, yeah. I mean, this was just another amazing session has been completed now. Our safety symposium is almost finished. Not yet. We still have to do or can do some really nice roundtable discussions. Before we go to the roundtable discussions, yeah, I would like to thank all the speakers from today and from yesterday. Really amazing talks, and also the audience for taking part and participating in the discussions. And of course, also the organizational team here at Nanion. But this is not yet the end. So we have roundtable discussions going on. We also have a poll going on where you can vote if you want to have these this kind of symposium again next year or maybe in 2 years. So that would be interesting to know from you guys. And we come back to the stage at 5:35 for a final recap and results from the poll from Jason. Yeah, and now we can get started with the roundtable discussion. So Alison will be greeting you in room number 1, together with Jim Kramer, speaking about or discussing about automated patchland best practices. And Gary and me will be in the second room, focusing on cardiac Bitcoin progress in silico modeling. So see you later.